Oop, one of me is going live on, oops, actually. Let me find the right page. I'll try to go live on my real Facebook account. Live. What's quite good is when we restart it, it pings people new push notifications that there's a live stream.
Yeah.
Cool. Live on Twitter. Waiting for live video on the Facebook ones. Oops. Oh, no, hang on. That's okay. Cool. Okay, we're live on Facebook, live on YouTube. Soon we'll be live on Twitter. Yeah, cool. We're live on all. Right, cool. Just going to pin the new videos to each of the pages. Let's hope Hunter joins us soon. Oh, we have Hunter. He'll be with us in a sec. Right, let me just do 2 final comments and then we're there. Mm, 4 minutes till we begin. Ah, that's why I need to make this one public. Great. Right, okay. Let's bring in Hunter. And cool. Hello.
Hi.
Hi, Hans. How's it going? I think you're muted at the moment, so let's unmute, and then we can hear you.
All right, how's that?
Yeah, perfect. Great. Cool. Okay. So our next guest is Hunter N. Hines, who's a PhD from the Harbor Branch Oceanographic Institute, Florida Atlantic University. But you also run the Instagram account at Microbial Ecology, which is another reason why you're here. And yeah, thank you for joining us in the Microscope Hour. In a second or two, I will enter into the Zoom call on my phone. I have a microscope here, which you might be able to see. If I do this, maybe you can just about see. Oh, that's Eleni and my microphone. Here we have a small, but functional microscope, which we can use. And yeah, we can kind of see and delve into a bit more detail about how to use microscopes to communicate microbiology. Yeah, so I have a few questions for you.
All right, I'm here.
And the first one is, so you now have this Instagram account, which, in case people haven't seen it, takes like short and interesting videos of microbes living in pond water and samples you find, and you post it as an Instagram, and you've amassed a huge number of followers. But what got you started doing this? Why did you think, oh, I know, I'll film these things and I'll put them on Instagram?
Right. Well, first of all, hopefully all your followers right now are following me at Microbial Ecology. They're on their phones, busy.
Yeah, find it.
They've done that, and they can pay attention to my explanation. So basically, I started this during my PhD. So back in 2017, I started the account. I was generating a lot of images for the project, and I was really kind of interested in what I was photographing, and I thought that I should maybe record some of these for myself just to look at later or to use for lectures. And I thought, well, why not just try to share these? And Instagram was a really obvious way to do that. Just super easy, a couple of clicks, you can share it directly from the microscope. And I think that's why it got such an interesting, and a large number of followers, because not many people at the time were doing that. And it's kind of this, unseen world. And I had access to such amazing, research grade equipment. Why not share it? So luckily it took off.
Yeah. And it's, probably brought microbiology and microbes to like so many more people than almost any other method I've seen.
Yeah, hopefully.
Because you've got now nearly 140,000 followers.
Yes.
So within that, who do you think what's your main audience? Like, who are these people, and is there any general trends, or is it just a mixture?
Yeah, well, Instagram gives you some analytics. So the majority of my followers are between 18 and 34 years old. At least that's how they identify. I'm sure you can type in anything you want there. So yeah, it's generally it's younger people. That's who's on the app primarily. But interestingly, and I've read about this in a couple of papers. A lot of it's kind of a worldwide amount. It's not just 100% Americans. It's all over the planet, and that's what's kind of exciting. It's people in areas which may not have readily available access to this research grade equipment or stuff like that. So it's great as long as they have internet access, they can just click and take a look down a $50,000 microscope essentially.
Yeah.
So
I think that's one of the most amazing things is like, if you are a microbiologist or you're working close to microbiology, you get so used to just looking at things down the microscope and being quite blasé about this massively amazing world that exists down there. But if most people have no connection to a powerful microscope, so when they see these things, they honestly are surprised and stunned by what they see, aren't they?
Yeah, and that was something I noticed, giving just in-person tours of the lab for different donors, prospective graduate students. They always had this awe and this amazement when they looked down the microscope. So I thought, how can I extend that beyond just 5 to 20 people a month? And social media is a great way to do that. You never know how many people are going to watch, what might go viral, but why not put that out there for anybody to see and incorporate into their lives?
So like, what happened when you started this? Did it quite quickly become big, or were you, low key for a while, and did it all suddenly change in a year? Like, what was the kind of...
Honestly, it took me a little while to figure out how to really use the app properly. Once I optimized that, it probably got to 10,000 within a few months, which was kind of slow. And then once you hit that point, it just kind of went a lot more viral. The different larger entities can pick up your profile, and for them, a lot of the online media, it's very easy. Just here's a couple links, here's an interesting microbe, boom, 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 and that shows their whole audience. So that's how it kind of got a lot more viral, and that's exciting too, because these are people who for sure wouldn't maybe Google ciliate symbiosis. They're not looking for this, but it's kind of thrust upon them and they still find it interesting. So that's a great way to kind of bring in people who aren't necessarily going to sign up for a microbiology conference, but they are interested in the natural world.
Okay. So then, I mean, you said that you've now honed your presentation and stuff a bit over these years compared to your first effort. So what advice would you give to any scientist who, they might have some microscopes, they might want to start using it to communicate? What are the main things they should consider when they're starting this?
Yeah, well, Instagram is so visual, isn't it? I think the most important thing is just getting a really nice looking image. So that really goes with having a nice adapter for the microscope and using the best images you can. Obviously, something really clear and bright is what everybody wants to see. And for me, I've had the most success just being very brief in my captions. I think people have enough having to slog through textbooks in Bio 1. They're done with that. They don't care about it. But seeing a paramecium as a living organism is really interesting to them. Perhaps they've only looked at a diagram previously. So it's kind of bringing that to life. And in my posts, I make sure to not only keep it brief, but just highlight a couple of the key factors. And then if somebody's interested in that, they can then Google those further. So I'm not giving them a Wikipedia page. It's just, here's the creature, here's something to look at, do your own work essentially. And I think that's kind of a great way to learn. It's self-motivated.
Okay. So I just wanted to know, do you have any of your videos preloaded, perhaps, that we could have a look at, just to give someone an idea of what kind of, because it's, these are all very bright, colorful, well-framed images. If you can't see the Instagram right now, no worries. And so there's a certain approach that works well, because you have to make sure that you're seeing stuff in a nicely focused, clear, and sort of like detailed way.
Right. So I'm trying to see how I have a few of the videos on my desktop, how I can
Okay.
upload them to you.
So virtual background's pretty good because then you're in the video.
Yeah, it wouldn't actually work with my computer wouldn't work on my machine. It's a
Wasn't
Mac.
playing ball. Well, maybe you can share screen and then actually that will probably be able to show at least some of the images.
All right, it says host disabled attendee screen sharing.
Oh, okay. Yeah, we're in a new call. So there we go. That should now allow you to do it. Yeah, we've been live streaming for like 7-hour chunks. So we just begun a new one. So it's all back to default settings a bit.
Right. All right. How does that look?
Yeah, we can see your desktop. Some beautiful underwater scenes.
Oh, yes. It's the Florida snorkeling view. All right, here we go. Here's a video. Is that playing for you yet?
Yeah, it is. So we've got some kind of ciliate.
Yeah, this is a ciliate blepharisma, so it's naturally pink. This is in a really kind of bright phase image. You can tell it's phased because the bacteria in the background are showing up as really nice and black. And to the upper left is kind of a worm which has got itself kind of stuck in the air bubble on the slide, and it's kind of wiggling around. I like this image quite a bit because the top part of the ciliate, you can see really obviously these hair-like projections, which is what makes it a ciliate. That's how they move with cilia. So yeah, it's a super common, lab rat organism, but I think it's nice just to show it, moving and interacting with a multicellular organism, which is this ciliate's actually much bigger despite being a single cell, which is kind of fun.
Yeah, that's something we looked at earlier today. We were looking at different scales, and there's amoeba the size of dust mites, even though one is single celled and one's multiple cells. They're the same size. Some
of the ciliates get to be well over 1-2 millimeters in size, so you can actually see them with the naked eye, which is really cool. This is always popular. This is a tardigrade, obviously. So this is a water bear. It's multicellular eukaryote. It's an animal. You can see the head is towards the bottom there. It has 8 legs, claws. You can see the cyanobacteria. It's ingested in the center, that kind of yellow mass. All the little circles are storage cells, which are kind of fun. And you can see the 2 little black dots. Those are the eyes, and those eyes are each a single cell, which is kind of interesting. These are these, indestructible creatures, which of course are quite fragile, but they are resilient to a lot of things. They can survive ionizing radiation in space, all that kind of thing. This is just found in a freshwater spring here in Florida on the microbial mats. They're found everywhere, but they're not as easy to find as people hope they are. But there's a few really great sites here in Florida. Tardigrades are always popular, I think, because they have a really interesting, upsurgence in media these days. So people are kind of familiar with the term and they want to actually see them alive, not just a TEM or an SEM image where it's just frozen in black and white. To see it crawling is kind of fun, I think.
So you've actually, in the past, you shared one of these videos with me for a video I made on a tardigrade, a little piece of footage. And we actually, when we were testing all this live stream software, looped it for 24 hours on our channel just to kind of have some video playing. And 888 people watched this looped tardigrade wiggling around. And actually someone sent me an email being like, is this meant to be on the channel? I'm so confused. And I was like, no, don't worry, it's a test.
I think tardigrades should be used more often for 24-hour loops. I mean, why not,
Yeah.
Let's see, play this one. This is a kind of fun ciliate I just took out of a saltwater aquarium. You can see how kind of plastic these things are. It left behind a bit of algae it ingested. It didn't need anymore. And this is another phase image. Just see the amazing detail of the cilia around the mouth. And these really are complex creatures, despite being just a single cell. You can see the huge mouth at the top of the screen there.
Yeah, it's huge.
Yeah, and you can see all the these are just natural color. It's just brought to life with the polarized light. So these are all different types of protists this thing is ingested. So you can see a diatom in there, various species of algae, all that kind of stuff. So it's really nice kind of looking at this. These are the foundation of food webs, obviously. So to see these interactions at this level is quite interesting. This being obviously a predator of algae, and then larger things would eat this cell. So I always find that fascinating.
Are all of these from samples that you have found in Florida, or do you ever get someone to send you something from further afield?
Yeah, it's a little bit tricky receiving international samples to a laboratory university in America. A lot of awkward rules with that. So most of the stuff I post are things that I've personally collected here in Florida. And obviously for my PhD, I was looking at ciliates in freshwater and terrestrial systems. So I did a lot of sampling, took a lot of images, and that's where all this content came from.
Yeah, very cool. Okay, one of the things I'm going to do now is I'm going to join this call again on my mobile phone, and then we're going to try and see if we can do some live microscopy. Ooh. One of the things I need to do is mute the volume. There we go. And I also need to start video. So there we go. You can see me a bit blurry from beneath. Yeah. Beautiful angle, but we need to flip this round. Cool. Okay. So this is the setup we are in today. We're in the studio room with many screens. And then we've got Eleni, our science communication assistant here, who's doing some backstage stuff. And then here's our...
The thing kind of froze. Hopefully you can still see me though.
Yeah, just about.
I can hear you, but yeah, it's frozen.
Ooh, okay. It's having to adapt to many streams. I'll try and just stabilize this in the adapter then.
Are you still there, Hunter? Ah, now you're moving.
You guys still there? Can you still hear me?
Yeah, I can hear you now. Can you hear and see us okay?
Yeah, I can see you. Yeah, just kind of froze a little bit.
Cool. I think it was just adjusting to...
yeah, it's Zoom at 10 a.m. in America, so everybody's logging on right now. It's never stable.
Yeah.
Yeah.
Well, it seems like a good time is actually sometimes not a good time. Right, okay.
Yeah, it's incredible, the images, Hunter. Like you're saying, you get a lot of interest from non-microbiology people who don't understand it. But I was doing bacteriology research, and this is something that I never saw in my research at all. And it's you forget sometimes that these things exist and how fascinating.
Yeah, and I think it's nice. Just the more microbiology you watch as a professional, the more you kind of learn and incorporate into your own studies. You know, I got an email from somebody who was a professor in UK, and he said, "Man, I didn't know these ciliates ate diatoms." I said, "Well, what do you mean? Of course they do." He said, "Well, I just never thought of it." And so I had posted a video, and it's full of these diatoms, which is what he was working on. So it was kind of interesting little just collaboration there, just to kind of expand his realm of thinking. This is a PhD scientist, so I think it's great.
Mm. Okay, I'm going to just, spotlight, I'm afraid, the microscope video. So we're going to just appear above the top. If I just check what the live stream looks like from anyone else's perspective. Yeah, okay, so we'll appear when we're speaking in the top right-hand corner, or maybe they'll just hear us. I'm not sure. Let's have a look.
I'm sure they're more interested in seeing the microbes than me.
I think so, or than myself. Right, let's do a test because we have some just sort of like less animated slides that are just, you know, things secured here. So let's start off with something historical. So back when, you know, the first microscopists were looking at stuff, we obviously had Micrographia by Robert Hooke, and one of the famous illustrations was that of a flea.
There it is.
Here we have a flea. How's that coming through? Yeah, okay, cool.
Brilliant.
So I'm on, I think, the lowest setting.
Yeah.
So it gets a bit dependent on how much you can fit in between the slide and the microscope, but we can zoom in a bit further and see some more detail. Cool, okay, so here we have some flea legs. You can see some nice hairs.
Yeah, beautiful image.
Yeah, so I gave the microscope a big clean using one of these dust cleaners before today, and it did really help.
Are they sponsoring the video?
Sadly not.
No, pity.
Okay, so that works well. Another cool thing we can look at, I've got a few set slides, and I do have some live samples we can look at as well.
All right.
And then we can use your expertise, Hunter, in trying to guess what we see on camera. Although it won't quite be as clear as what you've produced because it's not a £50,000 microscope, sadly. Here we have a mosquito on the way. So yeah, there we go. Here's the tail.
Nice.
And the body of the mosquito. And you can see kind of the digestive tract,
Sure.
which I guess is probably full of blood when it was fixed because it's very dark. And then you can see some eyes. One of the fun things as well, although this isn't very scientific, is I can put colored filters on to try and make it look a bit cooler.
Oh, nice. I think it's worth noting too how, you're a professional, but how easy it is just to kind of get a live image from a microscope. So I think a lot of researchers, especially students, should maybe think about just how easy it might be to share their work.
Yeah, especially nowadays, you just need a smartphone and Zoom and
Yeah,
there's
super.
Wi-Fi and you're there.
Yeah, I mean, Twitter, Instagram, it's all super easy. So why not post what you're doing? Otherwise it just remains private. You do all these years of work, and who's really going to read your dissertation or your papers? Not many people. But social media, you can bring in everybody, which is kind of fun.
Well, this is something I think about in communication. Is like you have to think about the cone of communication. There's like the initial field who might be 5 people who will read your research, but is there any reason to favor them beyond the rest of anyone else you could reach? If the research becomes more communicable, you get a huge number of people then understanding what you're researching.
Yeah,
Okay,
I think recently I posted a bunch of silly videos which were from a chapter of my PhD, and they were, I did publish a paper on it recently, and I shared that paper. But I think people are a lot more interested in just watching the thing living. If they want to go look at the genomics of it and all that stuff, they can read that paper. They can read my methods if they really want to, but they just want to see the creature and see it for something unique. And I think that's what social media brings to the table.
So here we have coming up next, there we go, an ant, which I really enjoy this slide.
Oh yeah, nice.
Yeah. So one of the things that I guess you might not know if you haven't used a microscope is you're kind of also focusing at a point within the slide. So you can kind of move up and down through the animal by focusing on the top and then focusing through the animal. And you kind of like move down in layers, which is kind of strange to think about, but cool.
Yeah, a lot of people, they ask me in comments or messages, why is this 2D? And I said, well, no, the microbe's not in 2D. It's just, either very thin. Well, that's kind of, think about how a camera works. I look like I might be in 2D, but actually I am in 3D. It's just the way these images work. So the camera of a microscope is similar. You can go up and down through the focus. And that's what people kind of lose perspective on. But interesting point to make, yeah.
Yeah. So I have also got some live samples, but I'll just do one more of these fixed ones just to see, because we also have, these were insects, but it can also be quite good to show plant life.
Yeah.
And then we can see kind of the inside of a cotton stem somewhere over here. Which way am I going? Am I in the right direction? I think I would be told off for not keeping my slides very clean if this was a professional lab. Here we go.
Oh yeah, nice.
Yeah, so here you can see kind of the cells inside a plant. And actually they've stained some of them. So I think you've got kind of the xylem are stained, maybe a bit darker.
Right. Yeah, pretty.
Yeah.
Very nice.
Cool, okay. Now, the next one is kind of a collection of different weird things
Perfect.
that I put together in this little box. Although one of the things that happened was, if you can kind of see it here, I had a beautiful collection of salt crystals and dried moss. And then last night I put some pond water in this one. What happened is all the moisture kind of evaporated, moved down the chamber with the lid on, and then it's dissolved the salt crystals and rehydrated my moss, which isn't ideal, but we'll have a go. Okay. So there are a few mosquitoes down here, which are real mosquitoes in the sense that they are from my flat from this summer.
Oh,
and
nice.
Yeah. So you can also illuminate from above.
Oh yeah.
Yeah. So now you really get a sense of the kind of 3 dimensions, I guess, because you can kind of move down the animal.
Yeah, and how terrifying mosquitoes really are. I mean,
yeah.
Oh, my screen's has gone dead. Just getting it up. Here we go. So yeah, I mean, obviously your research has helped your Instagram page, but has there been any reverse relationship? Has running this account helped your own research in some ways?
Yeah, I mean, just, self-servingly, it was, a chapter section in my PhD. So that was nice, kind of talking about social media outreach. But I think just overall as a scientist, it helped me just kind of understanding the relationships between these different trophic levels and almost just identifying what I was looking at. I mean, I was very specialized in these freshwater ciliates, but finding out what else was in the water, what else in the community, and how they kind of interrelated to explain that in the posts, I think was something I would not have done had I not been posting, daily on social media. So I think overall it kind of rounded me out better as a microbial ecologist.
Oh, that's good. So then, as a microbial ecologist, if there's one thing people should know about microbial ecology, what do you think it should be?
Oh, I think it's that microbes are everywhere and they're incredibly important for the survival of life on this planet. Microbes tend to get kind of a bad rap. They're kind of like sharks here in Florida. You only hear about them when they hurt or kill somebody, but really they're here all the time and very important in ecosystems and within us, microbes. So I think it's good to remember these things are a healthy part of environments, and they're incredible.
They definitely are. Right, okay. I think we've seen now enough of this kind of thing. I think it's time to try and find some microbes.
Ooh.
So this might take a second or two because I have to prepare a slide.
All right.
Yeah. But do you
We'll judge your slide preparation skills then live.
Don't. A long time.
No pressure. How did you get into specifically this area of research, Hunter?
Oh, so I was actually doing a master's degree at Bournemouth University in England, and my supervisor there was Dr. Genevieve Esteban, who's kind of a world expert in freshwater ciliates. So I just did my project with her and just really kind of fell in love with the microbial world, and that continued on to the PhD. And I've been looking at microbes ever since, since 2013, I guess. Yeah.
I'm just going to show people roughly how you set a slide. And you can judge me as well. So I've made it the spotlight.
Always.
Yeah. So we have, I'll just show people what we've got here. So this is a sort of collection of gloopy pond water that's
nice
been
color.
sat on my desk in anticipation of today for probably 3 weeks, 4 weeks. So a lot of multiplication of microbial life has gone on in here.
Nice.
And we're going to try and see if we get on a slide. So one of the other things I have is a little pokey stick, which is very helpful. So we're going to take a drop. Shall I give it a stir?
Is that a weld slide or a regular, just flat slide?
So this is a flat slide, and then I've got a little glass cover slip like this, which will go. I mean, it would be nice if I had little divots for a bit more of a to capture, because this one does have the tendency to overflow. But if we put a drop like that, and then, the trick is to not get air bubbles and not get fingerprints. I'm failing at both. Okay. Right. So now we have, hopefully, some microbes trapped beneath the pieces of glass here. Right. Now switch back to the microscope.
Maybe we should call this video Hunter Hines Reacts to Microscopy Setup.
It's a shame you can't join.
Definitely overflowed the slide. Let's not mess up.
It's a shame that you can't access your own microscope on this auspicious day.
Yeah, sadly, no. My lab is fairly shut down now. Coronavirus is fairly serious here in Florida, so we're minimizing everything.
Yeah. I never realized, until I had a little look, how many people live in Florida. It's just huge. So many Americans, like a fifth or something, live in Florida. It's ridiculous.
It's a big state, yeah.
Yeah. Right, okay. So I have got a bubble under the slides. That's already lost marks.
Oh man, 0 for 2. Yeah.
Yeah. We do have some debris, although it's not quite as thick and rich with microbial life as at the time I did it yesterday. So maybe I'll try and doing this again, but let's have a bit of a search because we might find something cool. I think I need a bit of a gloopier drop of water. Do you know what I mean? I think I was taking it from a bit too near the top. So I'm going to set this one again because we need some more greenness.
Yeah, try to get maybe the very bottom, layer. See what's
I think if we get.
This is kind of the fun thing of, sampling for microbes. It's almost like fishing. You ever go to the ocean and you never catch a damn thing. There's fish there. Very similar for microbes. They're all over in this system, but whether or not you're going to get the targeted microbe is always the question, which we're demonstrating here.
Okay,
So you
this
must
one's
spend quite a lot of your time out there, Hunter, like looking for kind of sampling and doing field work and stuff like that.
Yeah, a lot of my time, and I did. I had 4 major sites for the PhD, which I sampled a couple times weekly. So it was a lot of water samples. Yeah, recording presence, absence, all that kind of thing. Describing new species. It's good fun, but a lot of time on the scope. Yeah.
Yeah.
Oh, there's something. There's
There you
something
go.
alive.
Something alive.
Let's go in one more level. See if we can find something. Something is moving there, isn't it?
Yeah, there it is.
Yeah,
It looks
I think
like
it's
a little rotifer, I would say. Yeah.
yeah, I think you can also see it filtering the food.
Yeah, so it has that pinwheel-like mouth, which is ciliated, and it brings in the current, and it can also use that to really jet across the slide, as well as its little hook foot.
Ooh.
That might
So one
be
of
a
the
little,
problems here is
yeah, I
that
think that's too close. You've hit the
is
slide.
too close, yeah.
Yeah.
And it's now formed a drop on my microscope's
Oh, that was so
lens.
bad. It's froze the screen now. Hopefully you can still hear me.
Yeah, I can still hear you.
So a murky amount of dishwater. Okay, let's go back to the previous one. There's a lot of movement now within the water, but we have now got, a denser mat of microbial life. So there's some other things in here. So there's some more filamentous stuff.
Oh yeah.
Those, yeah. What do you think those are going to be?
Yeah, probably some type of cyanobacteria, the filaments. It also could be maybe like a Vegetoa, a filamentous bacterium. A little hard to tell from this image, but I would go with cyanobacteria based on the coloration of the water.
Yeah.
As long as you say it confidently, people will trust you, I think, as well. So.
Yeah, it's true, isn't it?
Yeah.
What I like is that because there's so much debris, you can see the flow of water into the rotifer's mouth.
Yeah, exactly.
And you can kind of see the flow patterns, which is kind of cool.
Yeah.
Right,
This is from your backyard pond, or where was this sample from?
So this sample is actually, this is kind of weird, grown from like some disgusting murky plant water that was sitting on a windowsill.
Oh,
I just kept it in this jar for some reason, and it wasn't very full of anything particular, but there must have been a few things in there, and now it's full of life.
Yeah. Yeah, rotifers are pretty hardy. I mean, they'll take over a lot of abandoned cultures. Certainly in my lab, you'll have them full of rotifers eating all the bacteria matter, all the algae.
I mean, I haven't ever found a tardigrade in any samples I've been looking at yet. So do you have any advice for where we should go for them?
Yeah, I mean, they say you should look in moss, and then just put a little bit of water in there, and then tilt the dish, and then just take the water out, and hopefully the tardigrades will be in there. From my own work here in Florida, I have several ponds I sampled for years and never once found a tardigrade in. And a couple of springs I go to, and every single time it's just full. There's hundreds of tardigrades per milliliter essentially. So it just depends on the habitat, really. Even though they can thrive everywhere, it's kind of like the fishing analogy. You just never know if you're going to be able to catch one.
Yeah.
But I would try if you're somewhere are you calling from somewhere in Northern Europe? Are you in UK? You're in Netherlands?
So I'm in the Netherlands and Ben is in Sweden.
Oh, okay.
Yeah.
So I would definitely try some woodland moss then and see what's in there. Yeah.
Because they are in every continent, right? Tardigrades. So wherever you are, you can have a go at finding them.
Never far from a tardigrade. Yeah.
Yeah. It's like a much nicer version of a mouse or a rat, really.
Exactly. Yes.
Okay, so here we have some more spherical-like organisms, which are also moving.
So I would say those are probably some type of little ciliate, maybe a flagellate. I can't really see too clearly, but
yeah, it's not quite
the one in the middle is probably some kind of a little ciliate cell. Yeah. And then there's the single-celled algae. Obviously, the different circles there, which aren't moving. Ooh, big rotifer.
Look quite monstrous, don't they? When you get up close.
It's like a whale, yeah.
Yeah. Brilliant.
So we've had some cyanobacteria, we think some ciliates, definitely some rotifers.
Yeah. So when I did my project, I only worked on the really large ciliates, these flagship species, because there's so many tiny ones. They're very hard to identify, hard to manipulate. I looked at the really large ones. So it'd be kind of fun if we find one of those, but nice rotifer, yeah.
One of the problems is that this magnification, the lens is so long, it can bump very easily into the slide.
Especially when it's overflowing.
Yeah.
The shade.
I'm very sorry, guys. But yeah, here we have some, I don't know, they're kind of paired into little groupings. Do you see?
Oh, yeah.
Yeah. It could be colonial algae. It's a little. It's a little blurry on my screen. That's my defense. If I get this totally wrong, or some expert is looking at their screen and saying, "What the heck is he talking about?" But yeah, very nice.
But you can kind of see how everything moves if I tried to
Wow.
zoom too much in a certain direction.
Yeah.
Just applying pressure. Right, I'm going to move this back to another microscope. Standard. And we can come back to this one, but there are some other things we can look at. And one of them is I have been growing this bizarre biofilm sludge.
All right.
There's been some peppercorns in here, like, you know, just black peppercorn. And it's been sitting in this jar for, again, a couple of months. So we'll see what these kind of, gloopy, biofilmy things look like. But I'll try and do a smaller drop this time.
All right. Describe the fragrance when you open it.
Yeah. Let's have a smell. I'm the only one who can experience that, so I'm the only one who has to suffer it. But we'll have a go.
Yeah. Well, miles away.
Right. Very professional saliva clean, and let's have a go. Where's my other pet gone? Somewhere. Here it is.
Can you culture any of these larger ciliates, Hunter? Can you actually have cultures going in your lab, or do you have to sample them every time?
Yeah, I did culture a few of them. There's some old methods which still work. The only thing is for culturing, especially these large cells, is you have to kind of pick them individually, and you get some contamination if you're doing an environmental sample, and then something else will bloom like a rotifer, and then you have a pure tube of rotifers, which you're not trying to work on.
Right.
So I have given it a smell, and it just smells like pepper.
Oh, okay.
Like fresh pepper.
Nice.
So that's not too bad. But if we can have a look, you can kind of see in here. I don't want to pour it on my computer. Right. There's like lots of film that's formed.
Oh yeah.
Yeah.
A lot of life in there. Okay, let's take a look.
Trying to get some of the thicker sediment because that's the more interesting.
Yeah. To your other point, there are some microbes which I just cannot culture, and that makes them a lot more difficult to work on. You're trying to get the molecular sequences, you're trying to really image them. You maybe only find 1 or 2 per 500, milliliter bottles, and so those are very difficult to work on. And then sometimes, there was one I was hunting for a long time. I saw it once, then I didn't see it till the following year, and then I had a bottle just full of them. I mean, you couldn't look through the bottle; it was so dense with this particular style. So you just never know. This is kind of the fun.
Cool.
Video. Okay, so this one I've put less liquid on the slide, so hopefully we'll get a better picture.
All right.
We'll start maximum out, zoomed out. Okay.
Okay.
So we have some weird gloopy mats. I'm not sure if I find anything as big as a rotifer because it's been fairly anaerobic in there. There's not been much oxygen for a long time.
Yeah, it might be just a bunch of, bacterial slimes, something like that.
Yeah.
Microbial mat. Yeah. Yeah, it looks like a really dense accumulation of bacteria, which are just breaking down those peppercorns in there.
Yeah, because there's different sugars in the peppercorns, which some favor and some do not.
Yeah.
One more. Can we go in one more? Maybe just about, because what's annoying is we can kind of see that at this resolution, this zoom, we're actually getting such a small amount of light, it becomes quite dim. Right. But we can just about see some things. What is quite fun is if we use different light filters, it kind of shows you different things.
Very nice.
Because it picks up different, blue's a bit confusing. Let's try. Pink is quite helpful. So yeah, this is kind of pushing the resolution of this microscope, because these, I think you're right, are individual bacteria, and we can just about see them, but
Yeah,
it's quite hard to get much detail on them.
but quite cool. These, you can see how tiny these cells are, and then you look at the jar, it's just this kind of milky substance. So kind of amazing.
Yeah.
They make so many cells, you can see it so obviously.
We can also see if we, yeah, purely light from beneath.
Oh,
Yeah, it is just a mass of cells, isn't it?
Yeah, very nice.
There's a bit of green in here.
Yeah.
There's some darker objects as well, so there's some weird stuff in there. But do you have any idea what kind of bacteria?
Zero idea.
Zero idea. Because I mean, is this going to kill me if I get too close, or is it something that's perhaps
There's
fairly?
one way to find out, I guess. Yeah. How
have
much
a
longer
nice-
do you have on the live stream? We'll see maybe in real time if you're going to.
Yeah, if I make it to the end, it's a question.
Right, right. That's great TV. You know? Who would turn away from that? You've been poisoned at 10 a.m. Yeah.
Yeah, let's eat random microbes. At least it proves our faith in microscopy and in microbes.
Right. Next year.
I'm just cleaning up the slide that has the pond water on, because I think there's more moving things in there than on this
All
one.
right. Let's look.
Yeah.
Yeah, there's just so many different species and so many phyla. You never know what you're going to run into, which is the kind of exciting part. Especially with bacteria, so many of these things look similar. You really these tiny little cells.
So other than tardigrades, have you found there's any life forms which people respond to best on your channel?
Yeah, I think it's based on kind of interesting coloration is always popular. Something's bright blue or bright pink, or different, forms of motion. Some of these big flatworms, they move such interesting ways. They're so kind of elegant in the water. Those always draw people's attention. It's something they haven't really seen before. That's a nice, really good image there, showing all the water currents being created by that rotifer.
Yeah, it is good, isn't it? Let's see if we can get in one more level.
Yeah, really nice.
I think I've just ruined it, I'm afraid. Sorry, guys. I was going for too zoomed in. Here we go.
That happens. Yeah, I think that lens is probably too close to the glass. It must be hitting it.
Yeah, it's a shame, because we were getting some good shots yesterday. I'm not quite sure how it managed to focus, but we'll go back one level.
It's crazy.
Yeah, everything's a bit suspended in some flow.
It's fascinating to think. I mean, so I'm really inexperienced with these kinds of stuff. I was working on bacteria, but these rotifers are such, just like hungry little beasts. You just kind of see them creating the air current and filtering. And the way you're talking, Hunter, it's like, yeah, these are like the contaminants. This is not even the interesting stuff for what I do. Like the world, of kind of ciliates and in these kind of samples is incredibly diverse.
Yeah, there's a huge diversity, and rotifers are kind of weedy species. They're able to move through the air. They can form these kind of resisting stages. So if you have just a backyard bucket, within a week you're going to have rotifers in there, pretty much no matter where you are in the world. And so that's kind of interesting. These things are always common, especially in natural environments too. So they're readily, they reproduce, they survive really well. So yeah, they're kind of annoying for me because they'll either eat or they'll just mechanically break into the ciliates I'm working on. So rotifers and I don't get along so well, but they're amazing to look at. Yeah.
You must be like, not another one. Because, I mean, so what's
a lot of pure cultures of rotifers.
Yeah. So what are the more rare kinds of microbes that you're hoping to find then when you're looking for something to post on the channel or even as part of your research?
Yeah, I mean, for my research, I'm always interested in these large cells, these really big ones, up to 1 millimeter in size, which is so cool for these single cell creatures. For the PhD, we discovered 5 species new to science. That was really fun because they were at that millimeter or larger size points. I wasn't looking at these little 5-10 micron cells nobody can even see. We're finding really obvious things which are very hard to miss in a sample. So that was cool to document those as new species, because it was clear that nobody had seen them before, despite them being so common here in Florida. So that was nice for me, just to see how kind of open the microbial world is, and how much discovery there is to be made at these really obvious cell levels. So you imagine the little tinier zone, how much more discoveries there are to be made here by anybody with a microscope.
I mean, this is probably a stupid question, but when you do a sample like this, how do you know you've got a new species? What's the first moment you kind of think this has never been described before?
Yeah, well, luckily I had immediate access to my supervisor, Genevieve. Even though she was in England, I would just WhatsApp her a video. And so if she hadn't seen something, that was always a really good sign. But when we're looking at these cells, which are so large, there's very few of them, especially in the given genus. So you can almost rule out right away. For instance, we found one which was about 1.5 millimeters in size, and everything else in that genus was maybe 200 or 300 microns. So, this thing is over 10 times the size of the known species. So that was almost right away. This is something new. So that was really cool. You can just tell right away. Yeah, and there must be tons of them out there, really. I mean, I just was looking at freshwater, warm ponds here in Florida. The diversity of the planet is such that there must be an incredible amount of species out there waiting to be discovered.
In fact, there was a talk earlier this morning which still fascinates me, and they were looking at Yeah, you back there? We froze for a second.
Right. You got me back on. Sorry about that. You guys froze.
No worries.
As I must have missed the question. Go ahead.
No, I was saying, yeah. I mean, this morning we had a talk about multicellular magnetotactic bacteria, which blew my mind. And I just never heard of such a concept. So that was pretty fundamentally amazing.
Yeah.
And they were new species, yeah, or at least relatively new.
Yeah.
So in your process of filming, have you developed any special techniques for kind of making stuff look good? Are you just very adept now at using the different filters and so on?
Yeah, it's kind of a bit like driving. The more you do it, the better you get. A lot of my stuff on Instagram is DIC filter. So it's that differential interface contrast. And that really kind of brings out the 3D, makes it really pop a little bit. It also takes out some of the background noise. So you could almost see the gray to blue scale. And it really shows the detail, especially for these single cells. And I think that's really fun. And just showing these things, here's a rotor for moving, just as living, not just here's a boring black and white, drawing in a textbook you have to memorize. I think it's a lot more meaningful for people to see these things living, whether or not they've heard of them or not.
Absolutely. So here we go. We've got a nice rotifer stuck in between some cyanobacteria. So I'm guessing it's trying to eat loose bacteria or something within the cell.
Yeah, it'll pull in any kind of particles that, like bacteria, different algae cells, all that kind of stuff. It'll eat ciliates as well. Yeah, a bunch of them in there. Nice.
Yeah, let's get this focused. I quite like this view because it gives the kind of impression that you're. Let's try which one of these is best. Yeah, that you're looking at. It's no longer a cross section. It looks a bit more real under this.
Yeah. Definitely. And that kind of view, that imaging is nice if you're picking out cells which have different colors in them. Maybe you can just look at for the gold or the blue, and if you're picking out individual cells for research, this kind of black scale is very helpful.
Because we also have here, I think these are groups of algae, kind of bigger spherical cells. Can you see my mouse? Or is it? Yeah, no, maybe not. But and then we've got the cyanobacteria wrapping around it. So this has just been growing on my desk. I'm not quite sure what to do with it now, but it is fairly good.
Yeah, I think keep it going, right? Why not?
Yeah, I mean, I think for me, the holy grail is if we find some tardigrades and start videoing those.
Yeah.
Because that would be fun. Because actually, do I have it on this computer?
Take a look. Yeah.
Because there was, actually, I think I might. I'm just going to see if I can find it. There was so we did this a small series called 52 Microorganisms, where we made a video a week about a new microorganism. And I think you just published a paper in Fem's Microbiology Letters about using Instagram for...
yeah, last year, yeah.
Yeah, and at some point, I think I was in contact with you, and you lent me a piece of tardigrade footage, which made it into the video.
I did, free of charge.
Yeah, you didn't at all. Very generous.
No royalty fees or anything.
Yeah, okay. So if I now...
Oh, nice. Yeah, so there's the the electron microscopy image of a tardigrade, which people have seen, and then hopefully the video will bring it to light. There you go.
Wow, here we go. So I'm now amongst tardigrade. So this is the one we had on loop, but you can kind of see all the cells inside his body, which I find very strange. And then you got the mouthpiece.
Yeah, the mouth, a little brain up there. Yeah.
And this is his belly. Part. And this stuff here it's food. Hang on, where yeah, this is a belly full of food. Not food. Any...
Yeah. And it has those nice storage cells, those little circles. Yeah. Very cool.
And then here's the mouthpiece, which is made of calcium carbonate. So incredibly strong and powerful.
Yeah, it's a little tube and it just ejects out. Really kind of freaky looking organisms, really. The closer you look, the less cute they become, but isn't that true for everything?
No, and I was reading a paper about the mouthpieces of tardigrades, and there's, a tardigrade for every job, and there's a tardigrade that eats other tardigrades. So it has a particularly wide and capacious mouth because it has to swallow a whole tardigrade.
Yeah, and some they'll actually come over and they'll just kind of latch on and they'll just kind of pierce the skin of the tardigrade and kind of suck out the juices, which is something I posted. And that kind of had mixed reviews. Some people are really interested by it. Some were just horrified and they just didn't want me to load that kind of stuff. So, yeah.
That's kind of brutal, isn't it? Yeah.
Yeah.
Yeah. And I posted a video of a tardigrade, dying, because as you see on the slide, the cover slip can flatten anything. So I had a tardigrade like in that image there, just kind of flat, and eventually the slide crushes it, and the tardigrade kind of explodes, and all the cells come out of it. And it was slightly gruesome, I suppose, but it was just showing these things aren't indestructible. Everybody thinks tardigrades can't be killed, but really they're quite fragile microorganisms. They just happen to be resistant to a lot of things which kill humans. But you can squash one.
Mm. Absolutely.
You should, but it can be done.
How did that go down on the channel?
Very poorly. Yeah, very poorly. Yeah. The point was made, nonetheless. They're not immortal. They're just, they're quite resilient. That was the point.
So, what do you reckon? These tardigrades, they got spilled out on that mission around the moon, right? So they reckon that there's tardigrades there now, kind of dehydrated. What do you think the chances of them being rehydrated
or resurrected?
Yeah.
I mean, there's always a chance. I don't think there's probably much food for them on the moon, but when these tardigrades enter this kind of cryptobiotic state, they have to do it slowly. It's kind of like, you and I can't just be thrust outside in the winter. We have to get our jackets on. We have to prepare for it, similar to tardigrades. So they can't just go from being happy in a tube to then crash landing and being at, a freezing temperature with all this radiation. So it might have just killed them instantly, but it'd be interesting to check it out, right? Go sample the moon.
Because then you get to the idea that what if this piece of moon rock was smashed into by an asteroid, and then you spent these tardigrade, what are they called again? What's the name of the dried ton? Is it a ton?
Ton, yeah.
Yeah, and these dried tardigrade tons then spread across the planet, the universe, and are spread to other planets slowly over time. That would really be, a consideration for.
Maybe that's how they got to planet Earth, right?
Maybe they do come from space. Actually, I wanted to show you and share with you. This is my laptop background, which is an amazing image of a tardigrade.
Little nose. Yeah.
And you can see its mouthpiece. Where am I going to point? Over here. Yeah.
Nice.
Used for piercing bacteria. And then I guess there's calcium carbonate pincers stick out of the mouthpiece into its prey.
Pierce cyanobacteria as well. They'll eat any of the juices in the algae or they'll pierce the cell walls of these things, and they're really interesting. And those little claws are great because they can kind of hook on, and so they like the video you showed that I took earlier, the tardigrade, it was kind of just crawling kind of slowly, but that's because it was on just a wet piece of glass. He put it on a cyanobacteria strand, and I posted this before. They're a lot more agile, a lot quicker, and that's kind of how they're more naturally moving around. So that's a great image. Yeah.
I mean, obviously, sadly, this tardigrade will be dead because it will be coated in gold for this electron microscopy image.
It's a great way to die if you're going to do it, right? If you coat it in
Yeah.
gold and immortalized in images. Yeah.
The thing that amazes me are these claws, these incredibly sharp bear-like claws here.
Yeah.
It's got four. Yeah, I mean, they just look so mammalian. It's such a weird.
Yeah, that's crazy. And that one's probably maybe 0.5 millimeter or less in size. I mean, the average tardigrade is not huge. I very rarely see them above 800 or 900 microns. So yeah, they're really tiny little creatures. I actually have a couple of videos of some of these ciliates I worked on, which are 1.2 millimeters in size, swimming around the tardigrades, and tardigrades are smaller. So it's kind of neat to see this single cell, which is larger than this really complex creature with all these limbs.
Because parameciums, which I do have a video of here, are also quite large-celled microorganisms. I think this one.
Yeah, maybe between 100 and 300 microns for a paramecium.
Yeah, I move over this side perhaps. Then I think in this next section there's a few bits of video.
All right.
Yeah, so here we go. There's paramecium. I think the next one you see some mating conjugation. So this is one of the this is the original drawing of paramecium from a German scientific paper. And yeah, they're very complicated cells. They seem to just have.
They are, vacuole, little feeder canals. Yeah, brilliant.
Wow.
I think,
so these ciliates you work in, they were very, very big, and now I'm learning that these water bears are actually a lot more aggressive and kind of gruesome than I realized. So what would win in a fight between a water bear and one of your ciliates then?
Well, I've never seen that fight, but I think the tardigrade would, if it were to be ingested by one of these large cells, I think it would be able to claw its way out.
Oh, really?
Yeah, I think so, because it's kind of a slow-acting process, the digestion of these ciliates. I have seen some of these cells, a species called Bursaria, which is basically like a big mouth that's shaped like a pumpkin, and that just goes around filter feeding, kind of like the rotifer, and it digests everything. And I've seen that Bursaria eat rotifers before, and these, rotifers, they have an eye spot, they're living creatures, and they just stuck inside the cell, and that's where it dies. So quite interesting, yeah.
Wow.
But I think the tardigrade would. But if it was small enough tardigrade, yeah, I'll look for that. Look to make one.
Yeah, cool.
Yeah.
So I was thinking before when you're talking about how rare they are and sampling them. Do you have to do anything special when you're sampling? Do you have to do a lot of filtering or kind of have nets or anything like that? How does that work?
No, so I just take natural environmental samples, and then that kind of the sediments form in the bottle, and I don't shake them up or anything. I just keep it still, and I'll sample that water sediment interface layer. That's where a lot of the cells I work on like to be, in that kind of depleted zone. And that's where a lot of the tardigrades will be as well, feeding on the microbial mats. So I'll just take a little bit off the microbial mat, and from the right site, they will be in there in large enough numbers. Yeah.
One of my goals then is definitely to capture a tardigrade under a microscope. So I'll go looking for some moss when I get home.
Right. Or if you said you're in Europe, so in Northern Europe, if you find any of these kind of woodland ponds which have these microbial mats, so it's like this layer of either green or blue, just like filaments on the bottom, that would be a nice spot for a tardigrade to be feeding on. So I would take a bit of that water and take a look too.
Okay, I will search hard and far for these tardigrades myself.
All right. Good quest to have.
Indeed. So one other thing I wanted to ask you just before we run out of time is now you have 140,000 followers. Have you ever found yourself recognized in public by people who follow your account?
Well, I was sampling with another Dr. Sally Waring at a microbiology conference in Canada, and we were doing some videos for Instagram, and the people there, they recognized us, they followed us. That was kind of fun, but that's a very specialized audience, people who are present at a microbiology conference. I've given lectures before to larger audiences, at ASM, that kind of thing. And
Mhm.
when I start talking, they have no idea who I am, and I'll put up a slide of, here's my Instagram, and I'll see kind of in the audience, oh, they recognize, and they might follow me, and that's kind of fun. But yeah, I think also it's nice that a lot of my account doesn't have many posts about me. It's more about the science. It's more about just showing off these cool creatures. It doesn't really matter who I am so much. It's just, here's something interesting. You guys should all, take a look at this and hopefully respect the planet and the world a bit more, knowing there's all these creatures everywhere. So that's kind of been a fun part of it.
Well, that's a good experience. I sometimes find scientists can be very scared that, if they put too much online and they become in any way internet famous, it will ruin their anonymity and their sort of like chilled out life. But it seems that you can have hundreds of thousands of followers and also live an ordinary life as a scientist.
Yeah, and if you recognize the scientists, what's the worst that can happen? They're going
Frozen. Something has managed to stop.
You see me?
I can. I'm just going to jump out from the microscope for the meantime. Okay, cool. I think we should pull this to a close because we've got a big group session after this with several speakers, and I imagine that they will try and go over time. So in order to keep them to time, we'll give them a few extra minutes.
Can you tell how many people are watching right now? Any idea?
I don't know. It's on several places. I think at the moment it's not huge. We've probably got, let's have a look. Where are we on? So on one of the YouTubes, we've got, just about 23.
Frozen again.
On a few others, we've got 5 here. This one's interrupted. It should resume shortly. So I can't see how many people are on this one.
Hopefully you guys can still hear me. A little bit frozen.
We can hear you now, yeah.
I reckon we've got 50 simultaneous viewers, and then we'll see at the end, because all of these talks can be rewatched, and then we're going to put them all online. But I could have a look back and see the individual peaks. So when did we start? We started around 4. Yeah, so I'm not looking at all the stats, but on one of the YouTubes, we've got between about 20 and 30 simultaneous viewers. And then on the other ones, I'll have to add them up at the end of today. But we're in about 6 places at once. Oh, I think I'm muted.
Oh, I can hear you.
Cool, there we go. No, I meant Hunter was muted. But yeah,
All right, sir.
Let's move on to the next session because they're going to take their time. But it's been fantastic having you, and thank you for showing us your videos and helping us to identify things under the microscope. It's been great, and I hope that any scientists who are thinking about communicating with Instagram and microscopy have learned a bit and can do it themselves from home.
Definitely do it. Why not? Especially in today's times. Yeah. Best of luck with your tardigrade search. Looking forward to hearing about it.
Thank you very much. And hopefully you'll get back in the lab soon. We'll see. Yeah.
Take care.
You too.
Bye.
Yeah, so next section. And let me just check. We're going to be having a COVID-19 talk and round table. So let's get everyone in. Great. Okay. Stop my video.
Okay.
Great. Nice to see you again, Isabel.
Hello, hello.
Hi. Hi, everybody. Miguel from Lisbon as well. Hi, João Paulo. Hi, Fernando, Ben, and...
Hi, Fernando from Brazil.
Hello,
From Brazil,
For me, it's something like good morning, but okay.
we can speak in Portuguese.
Good.
Boa tarde, bom dia, bom dia.
You will not be able to follow us.
Sorry, Ben, we are in Portuguese, okay? And by the way, let me tell you, Ben, it's great work. I've seen you yesterday. I hope you have slept a little bit during this journey, okay?
We have been working. We have been working all day.
Great job.
I did. Thank you. I did sleep a little bit, and coffee is going to see me through to the finish line, I think.
The same with me.
Yeah.
So we have, let's say, well, can we speak in Portuguese among us? So the session will be 1 hour sharp, because there will be another one afterwards. Okay, so we have to be very strict with the presentation, with discussion, with everything.
Yeah.
Okay. Sometimes we have questions coming from the Twitter and Facebook, etc. And if we have questions, we will address the questions. But obviously, I think that perhaps we can start the discussion, the panel discussion, following the presentation of João Paulo Gomes, because I think that could be a good start, if you don't mind.
Ótimo.
It's fine, Pedro. You are mute. Okay, no problem. It's not a problem. It's not a problem.
Hello, everybody. No, I'm fine. No, thank you. I thought I was unmuted.
We have 1 minute. Joe, is Joe with us?
Yeah,
or Ben?
although, begin whenever you want. You've got a free hour now.
We can begin. Okay, so
Yeah.
I think I'm going to start. So welcome to this session, this new session on these 40 hours of microbiology. And this session is about the new coronavirus pandemic. And the idea is just to discuss and the lessons from Portugal and from Brazil. So we have made an effort to highlight the positive roles of microorganisms in our lives and in the economy, but obviously it was not possible to ignore this virus pandemic. And the session will start with a presentation, will be followed by a panel discussion. I will introduce the panel afterwards. So the presentation will be given by Dr. João Paulo Gomes. He is affiliated to the National Institute of Health, Dr. Ricardo Jorge in Lisbon. And he has been coordinating a very important work on the diversity of the new coronavirus, SARS-CoV-2 in Portugal. So please, João Paulo.
Okay, let me share the screen. Okay, first of all, I would like to thank the opportunity to give this talk to the international community. And I would also like to say good morning, good afternoon, and good evening, depending on the global location you are listening to this. So I was asked to present the example of how Portugal is using the whole genome sequencing data from the new coronavirus to monitor the COVID-19 infections in Portugal. And that's why the title of this presentation is Genomic Diversity of the New Coronavirus SARS-CoV-2 that causes COVID-19 in Portugal. So I would say the first question is why? Why studying the genetic diversity of SARS-CoV-2? Well, several reasons for that. I would point out the first one, and probably this is shared by most study groups worldwide. We use whole genome sequencing of the new coronavirus to monitor transmission chains and to identify novel introductions in Portugal. Regarding transmission chain, the concept is like this. So the whole genome sequence of the coronavirus is like its identity card. So if I become infected with a specific coronavirus, and if I pass the infection to my relatives or to people I'm working with, then it's supposed that the sequence of all these coronavirus are similar. So that's exactly what this figure is all about. So if this one is infected and if it passes the transmission to this lady over here, and then she goes to the family and passes the family to all the relatives and so on, and during at her job, at her job work, also it passes the infection to other people, and then we have a transmission chain. And how do we identify the transmission chain? If we sequence all these viruses, we will get similar genetic sequences. So the goal is to use the whole genome sequencing data to inform the health authorities that an outbreak, a transmission chain, is occurring, for instance, in a school, in a household, whatever, and try to block it. And if we block a transmission chain, we will avoid multiple infection, multiple transmission events. Regarding the identification of novel introductions in Portugal, I'm saying Portugal, but it's the same strategy every country is using to identify the introductions during the early epidemics. The concept is basically the same. We identify the virus that are circulating in our country, and then we compare the genomic sequence with public databases that are available worldwide. And if we find matching sequences, then we ask the public health authorities to provide us with information regarding the metadata, regarding travel history, local of putative infection, contacts, date of infection, date of symptoms, and so on. And that's the way we will try to identify how many introductions we had during the early epidemics in Portugal. The second goal of studying genetic diversity of SARS-CoV-2 is to measure the impact of the containment efforts on transmission chain outcomes. We all are aware that probably every country closed schools, restaurants, airports as strong measures to contain the infections and to contain the new introductions. However, we must find out if these measures were taken in due time. So we will use the data generated regarding the genetic diversity of SARS-CoV-2 to find out if the transmission in the community occurred after, during, or previous the most of the occurrence of the novel introductions, and above all, if these measures of closing schools, closing restaurants, and closing airports were effective to stop the transmission. The third point I would highlight is the determination of the genetic variability of antigens and targets of antiviral drugs with impact on the vaccines and therapeutics. And I'm saying that because the sequence of the new coronavirus has several proteins, and at least one of them is pointed out to be crucial for the development of the vaccine and for the development of antiviral drugs. It's designated as the spike protein. So it is believed that mutations occurring in the spike protein is here represented in yellow, is a protein highly abundant that covers the entire surface of the SARS-CoV-2 and is responsible for the virus attachment and entry into our cells, and is also a powerful antigen. So we develop an immune system against this protein. So the rationale is that if we find mutations in this spike protein, then we may have problems with the vaccine effectiveness. And also we may have problems in the development of antiviral drugs, because the conformation of the protein may change and the protein may no longer recognize the antiviral drugs. And finally, we use the whole genome sequencing data to determine possible association between mutations or groups of mutations that are here designated as genetic profiles with clinical outcomes. I mean, disease severity. So it's possible that while most whole genome sequence of the circulating virals are associated with mild infections, and we know this is the case all over the world, we may find out that some genetic profiles, some mutations may be associated with fever or with low disease severity cases, but some mutations on some groups of mutations may putatively be associated with more severely disease groups, like patients needing ventilation and so on. So this is pure speculation, and there are a lot of groups worldwide trying to find out this association between mutations and disease severity. So the second question is, how do we study the genetic diversity of SARS-CoV-2? Well, in Portugal, we started by creating a consortium enrolling more than 60 hospitals and labs that are spread throughout the country. And the role of these labs were to diagnose COVID-19 and pick up the positive samples and send the positive samples to the National Reference Lab in Lisbon, designated as the National Institute of Health. And then basically using classical wet lab procedures, we pick up the genetic material of SARS-CoV-2, and we use several equipments, heavy ones and light ones. This one, by the way, fits in the palm of the hand. It's very, very, very short, to perform the next generation sequencing. And then all the work is performed by using computers. Okay, basically what we get is a huge amount of small pieces of information, short sequences. And if you pay attention, all these short pieces somehow overlap with each other. So what the computer does is it puts everything together, it joins the overlapping regions and create a massive contig that we name as the whole genome sequence of the new coronavirus. And it's with this sequence that we work here with. And we pick up this sequence and we put it in the publicly available databases. And most important one is designated as GZ. And finally, we have created a website to the scientific community. All results are available in this website. The access is completely free, so anyone can enter the web page. It's regularly updated with the data that we obtain in a real-time fashion. And also very importantly, it's user-friendly. So anyone, regardless the degree of knowledge in genomics, can try to understand information that it's contained on it. The web page is this one, and I'll make a short guided tour right now. So the web page works exactly like this, and it opens like you're seeing now. It's a bilingual. We can change it to English, okay? And for the ones that do not want to navigate throughout the web page, you can just go to the latest situation report. It's a PDF. We have several of them disposed in chronological order. And this PDF basically tells you the story of what is most important according to the new data that we have obtained. But if you want to have a little bit more work and to navigate a little bit more, you have the goals of the study here, just clicking in. And some notes, tips for enhanced data navigation for the ones that have expertise in the field, and several reports with all the highlights disposed in chronological orders, the teams involved, the methods. This is very important because we believe that this procedure may serve as an example for other countries to do the same. So we have all the rules available for the wet lab procedures, and also for the bioinformatic parts. And finally, obviously, the acknowledgements and the contacts. Okay. So I'll show you a couple of examples how to navigate this and how to extract information from this. So far, we have almost 2,000 sequences of the new coronavirus that infected people in Portugal. So I can tell you that Portugal is the third country with more sequences available worldwide regarding the population that we have, 10 million. So basically, this is a phylogenetic tree. The colors, each dot represents a sequence of the virus, and the colors represent the location where the virus was isolated. So basically, the colors represent small villages, cities, towns, whatever, in Portuguese mainland and in Portuguese highlands. And basically, the concept of interpreting a phylogenetic tree is that the virus at the end of a branch, this is a branch, all of them at the end of one branch are more closely associated in terms of genetic similarity than if we compare these ones here with the others at the end of a different branch, okay? So if you want, for example, to identify a transmission chain, an outbreak, for example, an outbreak in a school, we have to choose.
Oh, sorry. May I interrupt just for a second as moderator? Just you need to share the PDF or the website. We are not seeing the web page of InstaFlu. Okay, you have to. We are only seeing the presentation. So please share the browser.
Okay, I'll try to find out how can I do it.
Sorry for the interruption.
Okay, sure. So you're not seeing?
We are only seeing the last slide from where you have the web connection, the web link for InstaFlu, InstaPET, COVID-19.
I'm sorry for that. Let's try.
Ah, great.
Oh, I'm sorry for that, okay?
So if you don't mind coming back a little.
No problem. Will we have time to do that? Like 5 minutes more?
I hope so. I hope so. I'll try to take it from the moderation.
Okay, I'm so sorry for this, okay? I thought I was sharing the web page as well. Okay, so this is how the web page looks like. This is the downloads, the PDF I was talking about for the ones that do not want to navigate. It's just clicking in. The PDF is open and with the latest news, and then you close it, okay? And I'm sorry, I have to put it in English again. Yeah, it's in English. The goals of the study, you just have to click here and click again, okay, to hide it. Here, just have to click for the ones that have a huge expertise in the field and want to navigate deeply in the website. Okay, just click in and click out. All the reports disposed in a chronological order, all them in PDF for simplicity purposes. The teams that are involved, the methods I talked to you about, the wet lab methods, and the bioinformatic procedures for any country that wants to do exactly the same procedure as Portugal is doing, and acknowledgements and so on and so on. So again, as I was telling you, we have analyzed almost 2,000 sequences for the new coronavirus. As I was telling you, Portuguese is ranked third in the number of coronavirus that are sequenced and that are released in the public databases, mainly in GZ, which is the major public database available for the whole worldwide scientific community. And this is what I was talking about, the phylogenetic tree. So the colors, the dots represent the sequence of the virus with a metadata in this black box, okay? And the different colors represent different locations where the virus were isolated from, villages, towns, cities, whatever, in Portugal mainland and also in the Portuguese highlands. And the viruses are placed here according to the genetic similarity and to the number of mutations. Take a look at the scale here. This is the number of mutations that each virus has, each virus reveals when compared with the first coronavirus that was sequenced in December by the Chinese groups, okay? All over the world, the procedure is the same, regardless the lab who is doing that. The phylogenetic tree is basically the same. The scale is always the number of mutations compared to the first virus that the Chinese people sequenced. Okay, so what we can see here is that at the end of a branch, the virus within this branch are more similar among each other than if we compare all these viruses with the ones at the end of a different branch. So this is the principle to analyze a phylogenetic tree. So, as I was telling you before, if you want to identify a transmission chain, a mini outbreak, for instance, occurring in a school, you have to try to find out the virus at the end of one of these branches and try to find out if they are similar. And I will give you one example. Here, I'll click here and I'll forget the rest of the tree and I will just zoom in everything that is at the end of this branch. Okay. So this is a zoom in of everything and we have now much more information. And as you can see, we have a lot of small branches, okay? Let's take a look. Let's walk through this branch. First this branch and then this branch. And let's take a look at the green dots. Here we have 3 green dots and also 3 green dots here. The green dots mean that this virus was isolated in neighboring regions or eventually the same region. And when they are disposed vertically, it means that these 3 share 9 mutations when compared with the Chinese one. And these 3 share only 10 mutations when compared with Chinese one. However, when we compare these 3 with these 3, only 1 mutation is revealed. So if we take a look at this, we have a virus isolated from someone infected in Trancoso. Here, Guarda. Here, Guarda. Celúrico da Beira, Trancoso, and so on. And I can tell you that all these regions are neighboring regions. And these 6 viruses have almost matching sequences. So I can tell you for sure that we have identified the transmission chain. And so next step is to provide this information to the health authorities so they can know exactly that this specific variant of the virus is spreading in that specific location, and they should establish and identify the contacts to stop this kind of transmission chains. The last information I would like to give you within the site is, let's go back. Again, take a look at the colors, and if you take a look at this color, violet, I can tell you that the violet corresponds to all virus sequenced in the Atlantic pearls. This is the nickname, the touristic nickname of the island Madeira, okay? A beautiful Portuguese island. And all the virus sequenced in Madeira have the violet color. And if you take a look at the whole tree that has about 2,000 viruses, you see that the violet is spread throughout the whole tree. This means that the coronavirus in Madeira represent a huge, a massive genomic diversity. And as you can imagine, this makes a lot of sense. This means that Madeira have a huge amount of introductions through the flights from other airports to Madeira. So this makes a lot of sense, and probably each of these violet points yielded small transmission chains within the highlands of Madeira. So that's all for the website. I'll go out. I hope I'm doing the right thing. Okay, yeah. You're seeing the screen now?
Fine, fine.
Okay, let's proceed. So finally, I'll give you a brief example of one of the most important results we have obtained so far, because at this stage, we are analyzing the data that we can extract from this massive genomic tree. So what can we extract so far from this study? I can tell you that the first conclusion is that the early COVID-19 epidemics in Portugal was characterized by the massive spread of a SARS-CoV-2 genetic variant. This genetic variant contains this specific mutation DE39Y in the spike protein. What does this mean? I told you about the spike protein, the yellow one here that covers the full surface of the virus and is responsible for the attachment and entry into the host cell. And 839 is the position, the amino acid position in the spike protein, where these amino acids was replaced by these amino acids, okay? A glutamate was replaced by a tyrosine. And according to literature, the mutation from the glutamate to the tyrosine at this specific amino acid position in the spike protein, in this protein, is believed to impact the virus entry. Take a look at the spike protein here and take a look at the entry process. It's the spike protein that is involved in the connection with the host receptor. And it's believed that this amino acid position, as well as others, is crucial for this entry process. So we speculate that maybe there's a conformational change in the spike protein that may impact the entry rate in the transmission. And curiously, this variant, D839Y variant, was introduced in an industrial area in the north of Portugal after the staff attendance to an international trade fair in Milan, Italy, Lombardy, okay, in mid-late February 2020. And what is funny about this is that it means that it circulated undetected in Portugal before the first COVID-19 confirmed cases, because you probably do not know that, but the first reported cases were 2 cases on March the 2nd. So this means that for at least 10 days, we had a massive transmission chain that we were not able to detect. And this is the scenario we observed. On March 7, take a look at the red portions, okay? The red is the proportion of the infections caused by this genetic variant. On March 7, the scenario was like this. On March 18 was like this. And on April the 30, we had this massive spread of the variant throughout north and central of Portugal. And so we estimate that between March 14 and April 9th, because we used mathematical modeling and to speculate and to perform these estimations, and this corresponds to the exponential phase of the epidemics in Portugal. This variant likely caused about 4,000 COVID-19 cases in Portugal, meaning that 1 in each 4 cases during this period of COVID-19 in Portugal was caused by this specific genetic variant that the national health authorities weren't able to control until they applied the measures I talked to you about: closing schools, closing restaurants, closing the airports. And it resulted in the largest transmission chain ever reported worldwide. Here, I use different colors for you to better visualize the genetic variants. All these yellow dots represent the coronavirus sequences that correspond to that specific variant in the spike protein. And all of them, as you can see, come from the same branch, this branch that divides in 2 and so on. So all of them came from the same common ancestor, because we speculate that everything came from Italy, from that trade fair, from a single introduction in our country. And here is the spread by the end of April. And finally, what's ongoing now? We are now finalizing the data by analyzing this phylogenetic tree that we have, and we ask for a huge amount of data from the public health authorities regarding the travel history, the date of contacts, number of contacts, the date of symptoms, and so on, to identify all introductions that occurred in Portugal during the early epidemics, meaning during the month of March. And then when we have this information, we will also search the international databases to search for virus for which we have a matching genetic sequence with ours. And then we will check the travel history of our patients, and we'll try to figure out which of these errors are true and how many introductions we have and present the data in a chronological order to learn not only with these epidemics, but also to apply new measures in future epidemics. Thanks a lot for your attention. I'm sorry, really sorry for what happened during this screen share. And thanks, a special thanks to all consortium members that provided us with all the samples. Thank you.
Thank you, João Paulo. Superb work. So now we are going to enter the discussion at once. And I'm going to introduce the panel members, the other panel members, and the moderator of the discussion. So one of the panel members is Pedro Simas, Professor Pedro Simas, my colleague at the University of Lisbon, Universidade de Lisboa. He's a virologist, so he is affiliated to the Faculty of Medicine and also to the Institute of Molecular Medicine, João Lobantunes. Well, he is currently a very well-known person throughout Portugal on account of his constant and important and of course very well-received presence in Portuguese television news, explaining the science behind the virus and guiding everyone during this new coronavirus pandemic. So the other member of the panel, because it's important, the experience of Brazil. Brazil is a country with which Portugal has strong ties, but unfortunately the situation, the health situation in both countries is not similar. Fernando Spilki is president of the Brazilian Society of Virology. He's affiliated with the Universidade Fival Porto Alegre in Brazil. Thank you for accepting our invitation, Fernando. And to moderate the session, we have Miguel Viveiros. Miguel Viveiros is Vice President of the Portuguese Society of Microbiology. He is Deputy Director of the Institute of Hygiene and Tropical Medicine of Universidade Nova de Lisboa in Lisbon. His institution is also involved in fighting the pandemic, and considering the mission of the institution, he has knowledge about the situation in Africa, in particular in countries that speak Portuguese, like we all in this panel. So, Miguel, I will leave now the discussion with you.
Thank you, Professor Isabel, and President of the Portuguese Society of Microbiology. Thank you all for the presence. Congratulations to fans and to the other societies involved in this long journey celebrating the Microorganism Day. And unfortunately, the entire world is under severe constraint due to a microorganism. So it's a new era of challenges. And we have seen here a good example of how science can help medicine, science, and in particular molecular biology, molecular epidemiology, can be of extreme importance, not only to diagnose, but to follow and to monitor and trace chains of transmission of coronaviruses. So congratulations, João Paulo. We have been leading this project in this consortium, and it's not only a consortium involving health authorities, but it's important to acknowledge the involvement of the academia and of all the academic laboratories. And here in Portugal, we had this fortune in the early days of the pandemic. Our government was, let's say, clever enough to bring together public sector, private sector, academia, to bring enough strength for early diagnosis of coronavirus through molecular biology. And this made a huge difference, not only on early detection, but also in contact tracing. So part of the results that João Paulo have presented. They have been possible only because this consortium was created among all these identities, and it was a free exchange of results and samples. So my first question goes to Fernando Spirki. Let's see the perspective in Brazil nowadays and how things are going in terms of the impact of molecular biology and the connection between public health, health control, coronavirus control in Brazil, laboratory network, and the struggle to control the dissemination of the viruses, and of course connecting with the molecular biology and the early detection of the viruses. Fernando, the floor is yours.
Well, first of all, I'd like to thank to Dr. Isabel and to you, Miguel, for the invitation, and to fans to be part of this session. It is a pleasure, and of course it's an honor to be part of it. And I would like also to congratulate João Paulo. Your work is amazing. I'm in charge of a huge Brazilian network for the sequencing of the virus, and we have a lot of sequences made by many institutions, but now we are starting with a huge network of 10 institutions in Brazil and more than 100 researchers that will perform something very similar to what you have shown us. And I think that many of the hints, and especially about the website, the way that you propose this to show to the people, I think that I will copy some of your ideas, because that was very good. We are studying this work right now because the money is coming now from the government for it. You know that bureaucracy is another thing that we have heard from our Portuguese relatives, but we are now starting this work. But a lot of efforts have been done until the beginning of the pandemics in Brazil, and many of our groups are working together with the state governments and the municipalities to make both the diagnosis and the genomic characterization of the virus. But from the part of the federal government, we have these mixed positions about the pandemics. As you know, the Ministry of Science in Brazil is giving us a lot of support in terms of financial support, and also you know that Brazilian universities are likely in Portugal. We are doing a lot of efforts for the diagnosis. Actually, in some states of Brazil, we have much more samples analyzed in universities than in the public laboratories nowadays. So this happened also in Brazil. But of course, as you know, Brazil is a continent, and in some moments, the Brazilian government did not give the right instructions for the people, and given the huge social inequality and a lot of other problems that we have right now, we are now passing through this apocalypse in terms of COVID compared to many other countries. But from the last month, the situation is now flattening a little, and we are seeing not so many new cases every day as we saw in the middle of May or until the middle of July. And but we are a little concerned about the possibility of a second wave. And that's one of the things that I would like to ask to João Paulo and to you after. How are you seeing the situation in Portugal? Do you believe that you're gonna have a new waves of the pandemics, or what can you tell us about that from the diagnosis, the routine diagnosis that we are doing on a daily basis for the future?
Thank you, Fernando. João Paulo, just a short comment, and then let's listen to the expert opinion of Pedro that has been following all this process from the beginning. João Paulo, as a health authority, how do you look at the current situation? Nowadays, we had 700 and something new cases. The 9th worst day in Portugal. Clearly, it seems the beginning of a second wave, or at least a severe increase in new cases.
Yes, I would say that there's no doubt that we are starting to face a new wave, probably a giant wave. But I will like to listen to Pedro, because I believe he has basically the same opinion as I do. I'm not as worried as probably most of the people regarding this new wave, because I truly believe that we are nicely prepared, not for everything, but we learned an important lesson to protect the ones that we must protect, and to give some degree of freedom to the ones that, for the ones we must do that. And I'm talking about the opening the schools and so on. So there's no doubt the second wave will be there. And I believe that Portugal will be prepared to that. At this stage, taking a look at the hospitals and the intensive care units and so on, the situation is not worrying so far. But, well, let's have our fingers crossed. And please, Pedro.
Pedro, should we relax? You agree or not?
Well, hello, everybody, and thank you, Professor Isabel. Professor is my colleague for a long time, for the opportunity to be here. And thank you, Miguel and João, and our colleague in Brazil. I think inevitably we will see an increase in the new infections, a substantial increase. And as Miguel and João have just said, I think we are prepared to have that increase in a controlled fashion. And what that means is that our high-risk groups are protected. In particular, the care home, people in the care homes are quite well prepared. I must remind you that the lethality rate in care homes was about 40%, which is in the lower side of the average for Europe, and so that's an unequivocal demonstration that Portugal did a good job protecting our high-risk groups in the care homes. What I'm worried about now is the people that are outside the care homes, because if you look at the data, people above 70 years of age, up to now, 55% of the lethality came from there. So there are lots of people in a society that are potentially exposed to this new rise of infections. My hesitation of calling it a wave is because my understanding, that's my personal understanding of a wave, is that there is a connotation with something which is not controlled. It's just a wave that comes and is huge and causes devastation. And I don't think that will happen. I agree with Miguel and with João. And why? As Miguel and João were saying, the Portuguese, they call it the Portuguese miracle, which I don't like, but they call it the Portuguese success in the first wave, was due to two things mainly, in my opinion. One is that the size of our wave was very small because we were very prompt at introducing the lockdown, so we didn't linger on that, and so the seeding was very low in the country, and we are benefiting from all these months that we are being relaxing the measures. We have benefited from a small seeding of the virus, but as you know, viruses disseminate in an exponential way, and above a certain threshold, things can get completely wild and exponential. So we have to be careful that we are still in this lag phase of seeding, and that worries me a little bit. And the other thing that was crucial for the success of Portugal was the fact that we, as Miguel said, we brought there was a huge spontaneous initiative from the Portuguese academia and scientists to bring about the capacity of Portugal to test. And test, as you know, is crucial to do the tracing and everything. I mean, as it has been for the last so many years in infectious diseases. So otherwise we are blind. And so now I'm very hopeful that we are about to change the game in Portugal in terms of testing. And I'm working actively on that, which is we have to go from a situation where a strategy where we were testing people that had clinical signs, people that were supposed or are suspected to be infected, and isolating them and doing the contact tracing, for a situation where, and sending them home into isolation and quarantine, to a situation where we want to do surveillance testing, to send people, negative people, back to work, back to school, okay? And we also want to do surveillance testing to absolutely try to guarantee that health workers that work in care homes do not introduce the virus into the care homes. It's not just like looking after running after the damage. Once it's there, then you have to do all the things, and so I think I'm very hopeful also that the Portuguese people have understood that this is not a joke, in inverted commas. We are at the beginning, not at the very beginning of the pandemic, as I said before, but now at the beginning, because we already have a degree of herd immunity, but we're still at the beginning. And so I was very pleased with the WHO yesterday that they announced that the world is at the beginning of the pandemic, because there is this, what we are seeing now in this rise of infections, which are still within the value or a range that is still safe epidemiological, because we have about 40 new infections per week per 100,000 people. It is still on the safe side. Look what happened in Israel. There are over 300 new infections per week per 100,000 people, and they had to go back to confinement, to lockdown, which is something that we do not want and we cannot afford in our country. So I'm hopeful that the Portuguese people will understand that. The Portuguese people will look back in the summer and say, well, look, well, we know what we did. We know that we had more movement, and that's why the cases are rising. We have to follow the rules, and we have the physical distancing rules that the Portuguese people have integrated very well. We have the mask, which is in Portugal, for Portuguese culture and structure, is absolutely vital that we wear a mask, and children wear masks in school. And then we have the tests, and we are trying to change the tests and bring back academia. And now, as we speak, there are a number of institutes, about 21 institutes in Portugal, academic, not from the National Health Service, that are going to help again the National Health Service to change this game of the testing and increase the capacity and introduce rapid testing. And I'm sorry if I was too long.
No, thank you. It was a great perspective, and yes, we are at the bridge of a new era for this pandemics, at least in Portugal, and massive testing. It's going to be essential to control, mainly to prevent transmission. Now that we are opening the schools, going back to what we call the new normality. And yes, the instruments we have is testing, it's social distancing, and the masks. unfortunately, other countries, other perspectives have been discussing the useless or the use of the mask and how it prevents the transmission. I think nowadays there are enough evidence that this is crucial. But I would like to go back to Fernando, and just besides just one question coming from the audience and from the thousands of viewers of this session, we have picked up two questions. This one goes to Brazil. If you have to choose one main reason besides the political reason for why the pandemics, the virus is not under control in Brazil, exactly which one would be? Social aspect, difficulties in the healthcare system to provide healthcare, or everything goes down to the lack of political support for what is considered the control measures implemented by the public health. Fernando,
I think that there is a mix of things that could brought us to the situation. Not only the political stuff, but I think that the social inequality is one of the problems, because it's truly difficult for poor people to remain in social distancing. So there are many people living in houses that have no more than one room, and 3 to 4 to 10 people living in these places. So it's impossible to talk about social distancing for people that lives with less than 100 euros per month. So that won't happen, actually. People have to go to the streets to fight for survival. That's the way it goes. Another thing that could have some importance is something that we are studying right now, is that we have this B1.1 lineage in Brazil that was also the major genetic lineage of the virus that was in the United States. And we know that this lineage could be more transmissible. And that's one of the things that could be there. And another thing that I think that could have an importance is that that Brazil has many countries inside of it. So we have some states in which you could have a very good healthcare system, or one of the main cities could have very good healthcare and others don't. So people are moving and transmitting the virus from one city in which the situation was controlled, and you have all the ICU beds that we need, and people move from this city to another city that yet did not have the virus, but and then an outbreak explodes, and the city has no chance to fight this. And the other stuff that I think that we have to deal with it is that a part of the population, I would say that something like 30% until now do deny the importance of the pandemic. So it's not only in the federal government, in some people from the federal government, it's also in the population. There are people that do not want to use masks, that do not believe about the struggle that we are living through. So it's a complex mix of social and economic related stuff, and maybe some biological background from the virus. And in our network, we are also studying some hypotheses about the genetic variability of the hosts. But from that part, I have no results to talk about that until now. So But I think that social economics and maybe some of the characteristics of the virus could be enrolled in this. And our response was too late, too delayed in some of the states. That's true. And the social distance was also too short in many places, no more than 10 days. So you won't fight the virus this way. That's the way it goes.
Thank you, Fernando. We have 5 minutes to go because the schedule is tight. But one last question comes from our audience directly to João Paulo. And if you still have time, the other members of the panel might comment, is coming from the genetic sequencing and detecting specific mutations. There is any notion, any evidence how specific mutations might influence disease progression versus asymptomatic colonization and the severity of the disease?
Unfortunately, there's no evidence. There's no enough data to support that kind of conclusions. I pointed out that point as one of the major goals of multiple group scientific research group spread, worldwide spread, because that's a very important point. If we could find a way to have an association between some genetic profiles and disease severity, and then use diagnostic tests that rapidly inform you about the presence of those specific mutations, that would be a priority, because then we will give a priority to those patients, to those infected people. That would be great. But I would like to say that people probably have no idea that it's very, very, very difficult to access metadata regarding clinics, severity, and so on. In Portugal, I would say that it's almost impossible to do that, unless the clinicians inside the hospitals want to do the job and contact you. But the reverse order is not possible. So due to data protection and so on, so it's very, very difficult to perform those kind of studies. I have no idea if in other countries will be more easy to do that, but that's a research line that would be tremendously useful for the progress of the medicine and to control the pandemic.
Thank you, João Paulo. Just one last question to our two moderators, and I'll go back to Professor Isabel to close the session.
May I ask a question myself?
Oh, please do.
Well, to the,
but let's
to the
close
two virologists,
with the hope,
not to close.
a message of hope.
to both virologists, in particular, our colleague from Brazil, about the florestation of Amazonia and other forests, and the coronavirus or other pandemic problems, let's say, because there have been long warnings for the inevitability of a global pandemic at this scale. What do you think about this?
That's one of our major concerns regarding Amazon. Many of our groups are studying new viruses there, and we have found a lot of new flaviviruses, and we have also paramyxal viruses very similar to Nipah circulating in animal hosts there. So the type of destruction of bad use of the forest that's being done right now from the last years, I think that's a threat to the world. To human beings, actually, because there is a recent expansion of minery and of agricultural and animal husbandry in the Amazon, that I think that will pose a threat for the next years. We are wondering that we will have a lot of new viruses being introduced in the next decades from this place, because we have already found these viruses as you know, Brazil has a lot of virologists.
Yeah.
That's one of the wealth of the country. And we have found a lot of new viruses, not only coronaviruses, but as I said, flaviviruses and paramyxoviruses that are somehow new, but very similar to very dangerous viruses that were found in Malaysia and other parts of the world.
Pedro, do you want to comment or to add?
Just I agree with it in principle, but I'm still hopeful that we can somehow revert things, and it's not that easy to break the species barrier. As you know, that many of these viruses break the species barrier, but are not adapted to humans. So let's hope that that doesn't happen very, very frequently. But we need to respect nature for many reasons, not only due to pandemics. And I would like to see it on that perspective, rather than just because we had a pandemic, now we have to save Amazon. We have to save it because we have to save it for obvious reasons.
Difficult, but not impossible. Okay, so we have to close the session. Thank you very much. The session will be online, so you can watch the session again or for the first time soon. Thank you very much. Thank you, all the speakers.
Thank you.
And
Bye.
the moderator.
Bye-bye.
And FAM's office. Thank you
very much.
Bye. Thank you.
Bye-bye.
Thanks a lot, everyone. Right, so our next speaker is also talking about COVID-19 and specifically the SARS-CoV-2 virus and antibodies and immunity. So we'll just see if he can enter the room. Aha. We do have Alfredo.
Hello.
Can I hear you? Yes, can you hear me? Yeah. Yes, I can.
Excellent.
Great. So yeah, we have Alfredo Garzino Demo, professor at the Institute of Human Virology and the Department of Microbiology and Immunology at the University of Maryland School of Medicine in America, and also at the University of Padova in Italy. Yeah, I'm just going to let you begin because we've got 40 minutes and I'm sure you've got plenty of things to tell us. And then we might get a bit of time for questions at the end, but let's just check if your screen share works fine and we can commence.
Yes, let me do that. And first of all, thank you for inviting me. I'd like to thank the organizer for this wonderful opportunity. And let me get to that share screen thing. Let me see. You see my slides?
I can, perfectly.
Excellent.
Excellent. I'll just disappear to avoid being distracting and I'll let you begin.
Okay, I'll try not to distract myself. And let's say thank you. I really like to thank the organizer for giving me this opportunity. And so I'll try to give an overview of COVID and SARS-CoV-2, the causative agent. A couple of disclaimers is one is
sorry, Alfredo, 1 second. Do you mind just switching the display settings so that we have the full slides, because we've currently got the presenter view.
Oh, sorry. Oh, it's
no
a
problem.
computer. Sorry.
No, you can go back into the slideshow, but if you go to presenter view, you can switch it.
Let me share it right, because I shared the wrong screen, obviously. And I think it should be the right screen. Let's try it again.
Ah, perfect. That's perfect.
Okay, my apologies. So, let's say a couple of disclaimers. One is that I'm not a trained corona virologist. I have 30 years of experience on HIV, and I switched entirely to SARS-CoV-2 when the pandemic started affecting Italy and the US. Obviously, I have ties in both countries, so it became really important. And so I'm a newcomer to the field, but I am a microbiologist and a virologist. And I also have a very nice mask with the coronavirus theme, so I think that adds to it. Now, the other disclaimer that I need to make is that the University of Maryland, with which I have an affiliation, is one of the testing sites for one of the vaccine that I will be discussing, but I have no tie whatsoever with that of other clinical trials. And so, you know, we know the phase of COVID-19. This is data from well, in Europe, it would be at 3:32 p.m. You know, but we are approaching 30 million people infected. The US, India, Brazil, and Russia are the countries most affected by the virus. These numbers are probably all underestimates, you know, because in some places you don't have, you know, you cannot test all cases, but these are good reference numbers as well for the number of deaths. The US is approaching 200,000 deaths, and hopefully there will be some, you know, control of the infection sometimes. Now, somebody mentioned, you know, there has been a lot of information about coronavirus, COVID-19, etc. In fact, ever since the beginning of the year, from January 1st until last night, we had an average of 200 publications per day, including Saturday and Sundays. So there is a huge output of information. Some of it is likely to be completely irrelevant. Some of it is bad information, and there is some good information. But the information part has really been critical. In fact, everybody has become a little bit of an expert on coronavirus, including folks like this one. And I hope you can hear it.
Because there's no COVID. It's a fake pandemic created to destroy the United States of America.
But the president said to Bob Woodward that there is a virus, the coronavirus, and that it is deadly. That's his opinion. The truth is that the CDC said there's only less than 10,000 people die from COVID. The other 190,000 have 2.6 or 2.8 other mortalities.
So I don't know how much you heard of this, of what this fellow said. You know that this person was referring to the fact that a lot of people, a lot of the mortality of COVID-19 is observed in people that have pre-existing condition, especially cardiovascular and diabetes, and some people have numerous conditions, and that's for sure a risk factor for deaths of coronavirus. But it doesn't mean that people that have pre-existing condition would have died the day after it had not, if they had not contracted the virus. In fact, this graph that I took from The Economist shows that pretty elegantly, you know, how even when you are getting pretty old and you have pre-existing conditions, and this is the interval of which the person was referring, between 2 and 3 existing conditions. But even when you are above 90, you are really losing 2.5 years of life that you could have lived. And the more you go into the younger population, you know, even with 3 existing conditions, between 80 and 89 years of age, you're losing 5 years of your life, and the toll becomes more and more heavy as you look at younger people. So it is not correct to say that these people have not died of COVID. They have, and there have been a significant number of years lost. So the arguments that we are losing just the weaker, no, it's not true. We are losing them much earlier. We could have them around for much longer. So the SARS-CoV-2, as we very well know by now, is the causative agent of COVID-19. And here is the virus is shown imaged as it attacks cells, lung cells kept in culture. Now, we are very familiar now with the course of the disease. You have an initial phase of the infection where you can have mild or even no symptoms at all, you know. in this phase, our immune system reacts to the viral infection, mostly interferons, which are very good at fighting the virus. In fact, people with good interferon response are probably the people that do not show much symptomatology. So some people have mild symptoms and fever, and they may progress in a second stage, which is where the virus is taking over the respiratory system, the lung. And this is when things start getting hairy, so to speak, because the immune response now, just like a good army, can create damage even to the people that is supposed to defend. Our immune system can contribute to the pathology of SARS-CoV-2, as well as many other microbial diseases, as probably a lot of people on this call know. So, and the disease can evolve further. The cytokine storm and the invasion of the lung tissue by activated cells may progress further. And now we are in what is called the hyperinflammation phase, which is the phase where you may actually, there may be loss of life due to cardiac failure, shock, etc. So, and you have severe respiratory impairments. Now, the one important thing is that the disease is not just while you are in the hospital. A significant number of patients do not have damage only while they are in the hospital, and the damage goes away after they clear the virus. In red here, you see pulmonary damage up to 12 weeks after discharge from the hospital. And in fact, people complain of shortness of breath, weakness, week after being dismissed from the hospital. Not everybody, but still is not necessarily a disease that you clear as you clear the virus. And this is the virus, probably many of us are familiar with this type of figure by now. This is an envelope virus, so it carries with itself part of the viral membrane, the cellular membrane. The spike protein is the one that makes contact with the receptor and causes entry of the virus into the cell. And the genome of the virus is a plus-stranded RNA, complex with nucleocapsid protein, and is 30 kb long. And this is a pretty big RNA virus. So the receptor has an angiotensin converting enzyme 2, which is part of the angiotensin-renin system, which controls several physiological parameters, including blood pressure. And there are proteolytic cleavage that activate the fusion process. Proteases like TMPR, SS2, and furin have been calling into the action of entry. So they are necessary for entry, which is mediated by endosomes, after which the virus genome can be transcribed into protein, and the protein can now the RNA-dependent RNA polymerase can start making copies of the genomic RNA into a minor strand, which then can be copied back into plus-stranded, as well as subgenomic RNAs that are those that allow transcription of proteins, which then are assembled at the endoplasmic reticulum interface and released by exocytosis. And these are some of the drugs that or approaches that we have to treat the infection, including antibodies, which we will talk a little bit. And there are remdesivir interferes with the polymerase. And there are other approaches, including anti-inflammatory approaches and approaches to increase the interferon response. And we can talk about it. The furin cleavage site is a distinctive feature of this virus. And the reason why I highlight it is that furin, unlike other proteases that are important for other viruses like the SARS coronavirus, of SARS, of 2003 SARS, have a furin cleavage site And furin is a protease which is pretty ubiquitous. So besides having a receptor, which is the ACE2, which is pretty ubiquitous, in fact, it is expressed in the gastrointestinal tract, in the brain, and there are gastrointestinal and central nervous system. Also, furin is pretty ubiquitous. So this could be one of the factors in the infectivity of the virus. Like with all RNA viruses, we wonder about the mutations that can occur, and the pretty famous mutation at this point, and is the D614G mutation, which seems to impart more infectivity in at least in tissue culture experiments. And this variant seemingly has taken over, become the prevailing variant in several countries. Whether this is a founder effect of the virus that first made it there, or whether this is truly due to a higher infectivity is still under discussion. The data that I've seen in humans talk about potentially maybe a 20% additional infectivity, but is still being debated. There are many reports about that viruses getting weaker or getting more problematic, particularly potentially dangerous mutation ones that seem to make the virus escape antibodies. Thankfully, that mutation is not being seen circulating a lot. And the good news about even the D614G mutation is that it's still this virus is still susceptible to neutralizing antibodies. A quick word on how the virus causes pathology. You know, as I said, most of it is mediated in the immune system. As the virus enters the alveoli in the lung, it starts becoming cytopathic, and the cell death signal can activate macrophages. There are many other signals that can activate the macrophage, and some of them come from NK and activated T cells, which further activate the macrophage. The macrophage itself has the ability to identify viral infection using specific receptors. And there is another potential component to this, which is against the antibodies, okay? So antibodies that are non-neutralizing may bind to a specific receptor on the macrophage, so-called Fc receptors. So this is a property of antibodies. You know, typically we see antibodies as blocking the pathogen, and this is what we call neutralization. But antibodies have a number of wonderful reagents that have a number of effector functions, so-called, by binding to so-called Fc receptors. So the macrophage is one of the cells that expresses Fc receptor, and binding Fc receptor of antibodies may activate further the macrophage. And there is some question whether macrophages in the lung may be activated to actually express ACE2 themselves, and that's been debated. But, so there are several mechanisms that activate the macrophage, and these causes the macrophage to become activated, start producing cytokines, and that is part of the induction on the cytokine storm. So IL-6, TNF, and other cytokines are really prominent in COVID. Another part of the equation is together with the macrophages, there are other cells that are involved. One is the neutrophil, and there is also a role of the endothelial cells. Endothelial cells express ACE2, so they can be infected. And some people are thinking that this may actually be a very strong participant to the pathogenesis. So the endothelial cells can activate the neutrophil to release so-called neutrophil extra traps, you know, extracellular traps. So these are when the neutrophil starts spewing out its own DNA. Part of the activity is to stop bugs like bacteria, you know, by making everything denser, but some of it can trigger coagulation pathways. Other coagulation pathways are triggered by the macrophage. So this is probably part of the reason why you have massive coagulation in some of the patients. And these are the nets, these neutrophil extracellular traps, which are revealed in this micrograph. When they are revealed, specifically when serum of COVID-19 patients is added to the tissue culture. But going back to the vaccine and neutralizing antibodies, but that's the type of antibodies that we want to induce. We don't want to induce potentially other antibodies that can activate the macrophage. So it's very important in the context of vaccines to make sure that we have the right antibodies. That's part of the safety. So of course, the question is, can antibody protect? Thankfully, there is strong evidence, clear evidence that they do. And this is, I'm not going over all of the evidence, but this is one study that is very informative. It is one of the earlier studies with an efficient vessel where they studied 122 people before and after a significant COVID outbreak. So out of the 122 people that were studied, 104, 82.5% became contracted COVID during the study. So some of since there were data from before the outbreak, the antibodies were detected in 6 of the people that were studied, and in 3 of them, the antibodies were actually of the neutralizing type. And this is the activity or the neutralization activity of the antibodies. Non-neutralizing antibodies don't have that kind of activity. So, and this seropositive with the neutralizing antibodies prior to the outbreak remained negative. They did not have COVID symptoms during or presence of virus during the outbreak, showing that they were strongly protected from the infection. Also in animal models, there are several animal models for COVID. One of them is the macaque, which has some level of symptomatology. So when the macaques get infected, they do have virus. But if you take the same macaques and try to re-challenge them with virus, now you do not see presence of virus. So this is very clear evidence that they get protected. Whether it is only antibody or whether this is a very short term between the first and the second challenge. So there may be something else working here, which we can talk about it after I'm done with this segment of the presentation. So these macaques have very good titers of antibodies, and there is some titers of neutralizing antibodies. So this is good news. This is good news for the development of vaccine. There are a lot of different approaches, and we can talk about some of them, but in the data that I'm going to talk today, I'm going to talk about RNA vaccines, which are very easy to produce, and in fact, they are currently at the forefront of vaccine production. And as you probably all know, there are several phases in the clinical trials, from safety, phase 1, extended safety and efficacy, phase 2, and efficacy, phase 3. And this is the current number of phase 1, phase 2, and phase 3 clinical trials that are ongoing in the world. Some vaccines have been given limited approval, mostly in Russia and in China, meaning that these vaccines are given to segment of the population while they are doing phase 3 trials. I'm not sure that that is the most expedient way to do things, but that's what's going on. There will be terrible logistics problems in deploying these vaccines, but that's not something that we can do much about it as scientists. So there is currently an ongoing phase 3 trial of as I say, of mRNA vaccine, this so-called mRNA 1273. there have been already a phase 1 trial, which is still ongoing, so on 120 patients to test different doses of the viruses. And then there is an ongoing phase 2 clinical trial, which has shown that overall this vaccine is safe. And this was divided into 4 arms, different doses, and also different age groups. And so far, the news are all positive, and that's why this is progressing to a phase 3 trial with 30,000 subjects being enrolled, 1 dose, experimental or placebo. And there will be 2 injections at day 1 and day 29. So looking at the data that have been published, this is again in the macaque model, which is limited disease, but these macaques, as you see at the higher dose, produce very nice titers of neutralizing antibodies. At the lower dose less, and as you see, individual monkeys, as is expected, express different levels of neutralizing antibodies. After the second vaccination, the titers become very good in the monkeys. They don't give absolute protection. You still seem to detect a virus in the monkeys after challenge, at least in some animals. So it's not 100% sterilizing protection, but the monkeys have no symptoms, so they are effectively protected from disease. This is a part of the data instead from phase 1 clinical trial in humans, where there are very good concentration titers, as they are called, of antibodies that binds to the receptor binding domain of the spike protein. And when you test them in a viral infectivity assay, they do neutralize very well the infection. So the data for now are very good, and that's why the vaccine is progressing into a phase 3 trial, and which will take some time to perform, and to get some information on the level of antibodies and potential outbreak infections. One word of caution is that we'll have to keep monitoring for 2 things. One, as I said, is the antibodies that may be not so good, the antibodies that may activate the immune response, and we don't want these antibodies to be induced by the vaccine. But also, this is an experiment done by the Barrick group at UNC, where, and this is with SARS vaccine. This is not, so this is many years ago, about 10 years ago, where they tested the efficacy of vaccines in young versus old mice. And the vaccines seem to be losing efficacy in the old mice. So another word of caution is we'll have to make sure that we are protecting the people that needs to be protected, which is the most vulnerable are people with pre-existing conditions and elderly people. So have we learned something from COVID? I would like to say so, but I'm not sure. And the previous speakers also touched on some of these aspects. One is, are we going to have more pandemic readiness? That would be really nice. And I was really thrilled by one of the previous speakers that was telling us how in Brazil they are seeing a lot of viruses trying to, in the animal population that may be ready to come onto humans. So before to this pandemic, we were really concerned about influenza, and we should still be concerned about influenza. But we should also keep an eye on all the emerging viruses that are out there. Hygiene obviously plays a big role in prevention, and I hope that we are learning to impose stringent rules for agriculture, keeping livestock and food markets, because the more humans and animals come together, the more you have the chance of viral transmission. More attention to environmental issues, because the more we change the climate, the more species that normally are not in an area move to that area. And so now you have increased chances of transmission. you may have more favorable conditions for transmission, etc. And finally, safety versus speed. We'll have to have a debate, and we're not going to have it today, but how fast do we want to go with releasing, decreasing the level of attention to and preventative measures like masks, etc., lockdowns, in order to recover, have the economy recovering, in order, and how fast do we want vaccines to proceed to deliver them to the population. So these are things that we need to think about. And I took longer than I wanted to, but this is what I wanted to say to introduce the topic. And I'm happy to discuss more any of the aspects that I touched upon. And
thank you very much. That was fascinating and has taught me a lot in these last 30 minutes. But you've also been helping us to keep our community up to date, you know, for the last 9 months, to be honest, because I remember we were drafting statements back in January to put on the FEMS website. But before we go into that, I just wanted to ask a couple of questions. So in this case, we're looking, you had some nice data about how rhesus macaques were able to, what's the word, kind of resist infection.
Yes.
Yeah, after being, I guess, inoculated.
This was one of the best news of the year. You know?
Yeah. But has since this been published, anyone repeated it to see if, you know, the immunity can hold over time, or, you know, so I mean, one of the things I remember reading was that coronaviruses are quite hard to make a vaccine for that gives you a long span of immunity.
Okay, very good point. Duration of effect. How long do these antibodies stay around? So a couple of considerations. One is, as I showed, you know, some evidence of protection in monkeys has been shown also with the vaccine candidate. I showed this data in another slide. If I did not highlight it enough, you know, this is the vaccine that is being tested in one of the phase 3 clinical trial. These are data not from human, but from the macaques, you know, and so you see protection, maybe not sterilizing protection from infection, but you see protection from symptoms, which is nice. And so, I think this data is being replicated. The other thing is that one thing is the natural infection, and the other thing is the vaccine. Okay, so how long is the antibody protection in a natural infection? People these days are willing to say months. Okay, but they're not. The fact is, we have not even had 1 year of a complete cycle of this pandemic. So we do not. The only way to know the final answer, how long the antibodies hang around in a natural infection, is actually to study natural infection for several years and in a big enough sample of patients. Yes.
Because that's one of the things, is, you know, it's impossible to answer the question, do you retain immunity 4 years down the line? Because we haven't had 4 years since people knew of the virus to be able to do any test like that. And I still find it bizarre to think it's only September of this year. I mean, it feels like an endless amount of time, but yeah, it's been probably less than a year since anyone really would have been discussing this virus, much less than a year that the public would have known about it. So there just hasn't been the time to learn everything we need to know.
Exactly. And there are proper ways to do these things, but the only proper way to assess the half-life of an antibody response is to assemble a cohort of patients and follow them in time. Bear in mind the fact that some of them may get re-exposed to the virus, you know, so there may be confounding factors. And so, and then then again, we already know that some people got infected the second time. So what happened there? Well, in one of the cases, we don't know because we don't have enough data on the patient that got reinfected. On the second patient that I know about that got reinfected, it appears that this patient did not make a lot of antibodies. So, and we know that, we know that people, probably the reason why people get severe disease is because from the onset, they don't make enough interferon. And then when they get into the disease, there is a lot of lymphocyte death, so probably some people are not able to mount an effective antibody response.
Because one of the questions I also had was, back in March, I was ill with coronavirus-like symptoms, and I was off work for about 10 days, and it took me 2 weeks to recover. And at that point in the Netherlands, they weren't testing anyone who wasn't in a vulnerable category because it was right near the beginning. They didn't have the testing capacity. Is there any way that I can get a test of my antibodies? Or there's no real procedure for this, but would an antibody test of a certain kind be able to inform me if it had been real?
Right now there is a plethora of antibody tests, So I do not know what is the policy in the Netherlands specifically. If you can ask your doctor to be tested, I would have to look. I mean, you can probably find that information much faster. But yes, the tests are widely available. And of course, there is a prioritization of if you suspect that you've been infected, you might still have antibodies if you mount a good response, and they may be measurable, and I suspect they should, and in fact, it's like in the case of the fishing vessel that I spoke about, these people were studied retrospectively, and they find out, no, these people had antibodies, so you can find them for sure.
Okay. One other thing I want to do is just, if I can share my screen, which is to show you, I think I can do it here, a little bit of data on how you changed FEMS's website statistics. So, I mean, you helped us put together this expert statement for kind of more of a lay audience back in March. But one of the things that it did was this spike here, and if you look at this, this is all normal web traffic, is entirely down to this expert statement. So it kind of completely changed the entire year's worth of website traffic to our web, to the FEMS website by drawing in so many people across all the different languages. And this spike here is International Microorganism Day, and still hasn't quite reached the peak of the coronavirus information frenzy that was the middle of March and the beginning of April. So there's now, I find this quite bizarre in the sense it's like a record of human behavior caught in website statistics. And then if you look at here, all the physical events in the world were cancelled, and so actually traffic to the website went down. But this is a spike due to your your writing, so I think that's down to you.
I mean, it is impressive, and in fact, that spike corresponded also to a number of invitations to interviews, by different news outlets. Obviously, people wanted to know.
Yeah, and the different languages you can see kind of ranked here in the different translations we had. But this is the interesting slide in terms of where people were coming from to access this information. And we were putting it out in different languages. So the ones that were written in Serbian and Italian and Portuguese and German were actually the most well viewed, because I think, lots of stuff's going out in English, but if your English isn't your first language, and complicated scientific information also isn't your forte, then having something in your local language is super helpful. So yeah, lots of good information being delivered here at a crucial time.
Yeah, well, happy to contribute a new statement if you think is.
Yeah, I think the antibodies would be an interesting extra element. Do you know what I mean? So that was so unknown at the time, and a bit more work has happened now.
Okay.
Oh, I think we've lost your webcam.
Well, actually, I think you turned me off.
Oh, did I turn you off? Sorry about that. There we go. I think I tried stop sharing and I ended up stopping all your video. But my apologies.
Okay. I don't think that handsome, that people should look at me. Maybe it's better they don't. But anyway, so yes, I mean the fact is, I shot that news clip, on purpose, because I think it really delivers the message that, by the way, other speakers were talking about before, the fact that you have all this bad information out there, these beliefs, and these one-sided, like, I think really the point about the mortality is really a key one, because there are these conspiracy theories websites that tell you, oh, people die of other causes. No, they were not supposed to die.
Yeah.
You lost your grandparents way before you should have. And, I lost my dad when he was 91, a few years back, and God knows, I could have used another year with my dad, even if it had been one,
Yeah. And I think if you're the one who's suffering, there's no point in which you're like, oh, well, I only had 3 years to go. That doesn't.
Sorry. And the point I raised about the symptoms that don't go away when the virus goes away, these are things that one of my relatives, she's 30 years old, she's a cardiologist, and she contracted the COVID in the hospital, and she was tired for weeks after the onset of disease and after clearing the virus. So these things need to be out there, and we should publicize them, because it is important that people have good information.
Yeah. Well, I hope this can be part of that effort. And this is all being recorded, so we're going to cut these long live streams into the individual videos. So you have the name of the speaker and the topic of the talk, and they'll all go up on the IMD YouTube channel.
Well, thank you for organizing these. I think we should have more days like these. We should make people more aware of microorganisms. I also take FAMS and the Portuguese Society for Microbiology. Great job. Thank you.
Awesome. Well, thanks for joining us. Our next speakers are on the way, but it's been great, and you've taught me many new things. And over the next year, all the videos will be going out across to try and fill the gap between today and the next International Microorganism Day. So hopefully we can keep content going throughout the year. So great to chat to you, and enjoy the rest of your day. It's the morning.
Good evening.
Yeah, it's the 6:41 in the evening here. But enjoy your day.
Okay. Bye-bye.
Thank you. Bye. Right. Next we have our intern Priyanka, actually, back to introduce and chat to our next speaker. So let's just make sure everyone is on board. Yes. Hi, Priyanka again. I'm going to get out the way and let you do the talking. So yeah, see you at the end of this talk.
Yeah. See you, Joe. It was really nice hearing from Mr. Gazzino right now. But transitioning into our next phase, into our next segment of Science Talks, joining us to talk about biofilm from India, from Amity University, India. She's Emin Fatima, who's doing her master's in microbiology from Amity University, star in her own right. So she has over 20,000 followers. She communicates about the interesting topics that she covers on her channels. But without further ado, I hand it over to you to talk about biofilms. Yeah. Emin, I think you're muted. If you could mute yourself, that would be great. Yeah, hi, Emin. How are you?
Hi, I'm fine.
Yeah, it's been really nice looking at all the so her account is Microbiotechnia on Instagram, where she has an incredible amount of followers, matching her incredible resource of information that she rolls out. But yeah, Emin, if when you'd like to start, the floor.
Okay.
So while she shares her slides, yeah. Perfect.
Okay, I'm sharing my slides.
Perfect, Emin. So those of us who are those of you who are joining us from India, it's already quite late there, but thank you for staying online with us. And the previous talk, as you saw, was on COVID and about the effects of how COVID is developing, and there'll be more talks on the way. But in this session, the next one is from Santos, I guess. But in the meanwhile, Eamon, if you'd like, we could put up the slides as well.
Just a minute.
No problem, Eamon. Take your time. It's fine. Well, while you do, have you been catching up with the stream right now, or are you tuning in right now for your talk?
Actually, I'm not able to connect the slides. Can you put the slides for me?
Yes, no problem. I can do that for you. Just give me a moment. Yeah, then introduce yourself. I'll just, while I draw up your slides in the moment. Okay. Just 1 second. What time is it in India right now?
It's 10:15 p.m.
It's 10:15. Yeah. So sorry we had to keep you up so late, but yeah, I hope you aren't too tired already.
No, no, no.
Yeah. So are you doing your master's second year, or like is this the final year, or are you done with the master's?
No, it's like it's about to complete 1 year left just.
That's nice. And are you doing a specific sort of specialization, or is it general microbiology?
It's general microbiology.
That's interesting. So how do you manage your time while maintaining that account? I saw there was a lot of information that's rolled out on a regular basis in your Instagram account. How do you manage the time for that?
Because I'm interested, so I'll manage. It's not very hard to manage actually. So I get time for searching for new things and post the familiar topics which people like to learn and to know about the basics first. So it will clear the concept and then you can start your things and everything. So it started like that.
How long ago did you start it though?
I actually don't remember, but I was working in a lab. And all of a sudden, I just got this idea to put a page on Instagram. And I was working on a project. So that's how it started and it's going on.
That's great. So can now look at your look at the slides that you put out and learn a lot about it. I do have your slides here. I'd like to get started with the introduction so that I can just while I start the sharing, or no problem. And you can just give me some cues when you want to change the slide. Is that all right?
Okay.
Great. Just give me a second. Did the idea for biofilms also come from there, or was it something that you were covering as one of your projects in class, maybe?
No, I mean, I was just writing a review paper. So on that side, there was a there was a mentioning of the biofilm very much a lot, and it's a major concern also.
Okay, that's interesting, and I'm sure you can tell us more about it in detail now. Can you see the screen?
Yes, yes.
Can you see your slide?
Yes, I can see it.
Perfect. Is it okay if I leave it on this mode instead of a presentation? It'll just be easier for me to change the slides. Is that fine?
Yeah, yeah.
Perfect. Great. Over to you, Evan.
Okay. First of all, I'm very thankful to International Microbiology Day that they are connected with me and allow me to have this opportunity to present my topic in front of you all. So I'm starting with the topic biofilm because it's a major concern and it can found everywhere in the world and on every nature on every particular thing. The biofilm can form by many bacteria. So we're starting with the introduction. Next slide, please. Okay, so what is biofilm? So biofilm are the collective type of microorganism which can grow on a different surfaces like metal, food, human body, any biological tissue, anything. It can grow on any different surface. These biofilm can form by bacteria, fungi, and protists. So we can see that these biofilms are present everywhere in the environment. And it was seen that the fossil evidence of biofilms are found 3.25 billion years ago, according to the 2004 article published in journal Nature Reviews Microbiology. Like for example, 3.2 billion years old deep sea hydrothermal rock of Pilbara in Australia is found, and these biofilms are also found in hydrothermal environment like springs and deep sea events. So according to the life science, biofilms are a collective of one or more type of microorganisms and can grow on different surfaces. That's the basic concept of biofilm to grow on different surfaces. Next slide. Next slide is about how its formation occur. So this diagrammatic representation is showing the formation of biofilm on any surface. So first the planktonic bacteria, which is a free-living bacteria, attaches to the surface. So the first stage is attachment, second is addition, third is growth, and the fourth is release. These bacteria have high mobility, faster growth, higher virulence, ABX susceptibility, and demand for nutrients. So now we go for the deep study of these steps of formation of biofilm. Next slide. Okay, so the first step is biofilm formation is attachment. The very first step is attachment of the free-floating bacteria on a surface by their roots, or you can say the bacteria has fimbriae, pili, and flagella. They get attached to the surface by the use of this locomotor activity by the use of locomotor organs and attached to the surface to form a biofilm. Now these attachment occurs when microorganisms produce a gooey substance which is known as extracellular polymeric substance. This is the main component of the biofilm which allows the stickiness to the biofilm and make the bacteria stick together for a very long time. You can say it acts as a glue. So this EPS is a network of sugar, protein, and nucleic acid. Now, when this attachment occurs, the second step is the growth of the bacteria. and they form layers and layers, and EPS build up on the first layer, that is a microcolony formation. So the second step is microcolony formation. This process occurs on a rough hydrophilic and coated surface will provide better environment because it has the critical level in flow of velocity, temperature, water, and nutrient concentration. And as I told that a locomotor structure on the cell surface is like flagella, pili, fimbriae, proteins, and polysaccharides are very important to provide the advantage in biofilm formation. So this microcolony formation takes place in bacteria when the attachment, after the attachment occurs to the physical surface. It can be any physical surface, biological tissue, your food items, your rocks under the sea, everywhere you can see the biofilm formation. So in this microcolony formation, the multiplication of bacteria started due to the release of chemical signals. We can come to this chemical signals part later. So these chemical signals, are very important for connecting the, please move to the fifth slide only. These chemical signals are important for the communication of bacteria between each other. So to activate the production of this biofilm formation colony, the genetic mechanism exopolysaccharide starts to activate when the intensity of signal cross certain threshold. By this way, and using of the chemical signaling process, bacterial cell divides and forms the microcolony formation with the exopolysaccharide matrix. It has a surface of a first layer, that is a microcolony formation. Next slide. Yeah. So the third step is formation of a 3D structure or maturation, or you can say a growth of biofilm. After this microcolony formation, there is some expression of certain biofilm related gene take place. These genes products are needed for the EPS formation, which is the main structural material of biofilm. This EPS is a very important structure for the biofilm formation, and it has some genes which make up these biofilm formation. I could not go into the deep of genes as of a shortage of time, so I am just explaining it to the basic. It is reported that bacterial attachment by itself, when a bacteria attaches to by itself, also triggers the formation of extracellular matrix. This matrix formation is allowed by water-filled channels, and it can transport the nutrients within the biofilm. Because to mature the biofilm, nutrients are very important. So they get these nutrients from the water channels, and the transporting of nutrients occurs through this. So researchers have proposed that these water channels are like a circulatory system, as we have a circulatory system. So for the biofilm, the water channels are the circulatory system, which is distributing different nutrients and removing waste material from the communities of microcolonies in the biofilm. Next slide. Next slide, please. Now, the very last step is detachment of planktonic bacteria. All this, after all the formation, attachment, adhesion, growth, and maturation, the last step is detachment. When the bacterial cell from the biofilm is programmed, detachment is a very natural pattern to detach from the surface. It occurs due to the mechanical stress of bacteria, and also in some cases it can occur when a bacteria stop producing the extracellular polysaccharide substance. So it allows the bacteria to detach into the environment, and again these bacteria come into its free floating phase, or you can say they come into their planktonic phenotype stage. So disposing of biofilm occurs either by detachment of new formed cells from the growing cell, means the detachment of new formed cells from the growing cells and the new formed cells will separate from each other and the detachment occurs. The disposition of biofilm aggregates due to the flowing effects or due to the quorum sensing. By these steps also the detachment can occur. So we are moving to the quorum sensing part, which is very important for the formation of biofilm. As I told you, some chemical signals. So this is the communication action of bacteria to communicate between each other and form a strong biofilm formation on the surfaces. So it's a system of stimulus to coordinate gene expression with other cells. So the gene expression with other cells, the one bacteria releases the quorum sensing to the other bacteria by some gene expression and allows to attach to them to form a biofilm. These quorum sensing can occur within the single species of bacteria or as well as different bacterial species. It can enable us to communicate between intra-species or interspecies. Which involves in terms of biofilm formation like food shortage, and environmental stress conditions such as disinfectants, antibiotics, bacterial colonization, and the identification of annoying species. These bacterial colonization have, the establishment of normal intestinal flora as well as many prevention of harmful intestinal flora also shows the quorum sensing formation, quorum sensing signaling to form a bacterial biofilm formation. Some clinically associated bacteria also use this quorum sensing for the regulation of collective production of virulence factor. When a biofilm forms, it has a very high virulence factors. That's why they become antibiotic resistant. So quorum sensing in a gram-positive bacteria occurs through a series of events. First is production, detection, and response to AIS. And the oligopeptide auto-including peptides in many gram-positive bacteria are detected by membrane-bound two components signal transduction system. So now we come to the bacteria part, which almost all the bacteria causes the formation of biofilms. So we are discussing here the biofilm formation bacteria.
Eamon, if I might just interrupt you there, sorry. It's been really interesting, the quorum sensing. I was just wondering, because we've kind of run out of time, would it be possible if you could summarize the last slide for us? And that would be, because we've run over time and the next speakers are already waiting. Would it be possible?
Okay.
Yeah, thank you.
I am doing in a very fast mode. So the biofilm forming bacteria, there are many like E. coli, Staphylococcus, and all the bacteria, they form biofilm and they become resistant to the antibiotic by some few conditions. We see how the antibiotic resistant biofilm occurs. We can move to the 11th slide.
Okay, and yeah. Yeah,
I mean, so this diagram.
Sorry, go on. Yeah, go on. Yeah, if you could just summarize this.
This diagrammatic representation shows the antibiotic resistance of bacteria, which occurs through many stages. First is the proliferation and adaptation of bacteria to ensure the survival. And the second is when the adaptation occurs in a particular environment, these bacteria adapt themselves in such a way that they become resistant to antibiotic by some mutation and enables them to become resistant to antibiotic. Now the proliferation of antibiotic resistance bacteria shown in the third picture, which is blue in color, these all are antibiotic resistant bacteria. Now antibiotic resistant bacteria can transfer its gene from one to another by some kind of a connection like conjugation, transformation, transduction. So other bacteria can also take up these antibiotic resistance capability and become antibiotic resistant. So how the biofilm is very hard to destroy by the antibiotics as they become resistant. Next slide.
I think
Hey,
that's
I'm
a good note to end
yeah.
on there on how biofilms can develop antibiotic resistance. Yeah. So no, but thank you,
Yeah.
Emin, for joining us and explaining all of that. It's
Thank
been
you so
really
much.
great.
Thank
Yeah,
you.
and now I'll hand it over then to welcome our next guest, I believe is
Yeah.
Ricardo Santos, right?
Thanks a lot. And I will let them in for talk. Right. So yeah. Apologies to having to cut that short.
Okay.
We unfortunately have to stick to time in this linear call. But our next guests, Mario and Sylvia. No, not Sylvia yet, but we have Claudia. Okay, good. Right. Mario, are you ready to begin with fungal diversity in thermal waters?
Yes, I'm ready. Thank you.
Perfect. Right, I will.
Yeah,
I can hear you fine.
Okay, perfect.
Great.
Hello.
Away.
Good afternoon to all.
Well, are you seeing my presentation?
Yes.
Yeah, absolutely.
Yeah.
Okay. Well, let us begin then to save some time. First and foremost, I would like to thank the organizing committee, especially to Professor Isabel Sacuré for the opportunity to participate in this special day. I'm going to talk to you about fungal diversity in thermal water. Water, source of life. Every one of us have already heard this expression before. However, most of us, not in the field, of course, not microbiologists, but in general most people still think of water as being sterile, at least in clean waters. However, as you can see in this slide, this is just a drop of water. So as you can see, lots and lots of life. You can find protozoa, algae, bacteria, viruses, fungi, and protozoa. In this particular case, what we are interested in is in fungi, of course. It is divided in two main groups, which is yeasts, as you can see in this slide. In this case, particularly, we are talking about Candida albicans in the image, which causes, as you probably know, vaginal candidiasis. And we have another major group of yeasts, which is Saccharomyces, which is also part of the mascots of the International Microorganism Day. So it's dedicated to that mascot. The other group is filamentous fungi, also commonly known as molds, which are multicellular, opposite to the yeasts, which are unicellular. In this case, you have an Aspergillus formigatus, just to know. In this case, we have a biofilm. I was going to talk about biofilms a little bit, but the previous presentation has already did that, so I'll move forward. In here is just pure curiosity. You find a drinking water pipe, and as you can see, regarding biofilms, in the outside of the pipe, you find lots and lots of life: bacteria, viruses, fungi. So this is just for you to know that really water is the source of life. Now, concerning fungi, there are a lot of health problems related to fungi, all documented in literature, as you can see some examples here. Mainly two types, respiratory problems and also skin and hair and nails problems, namely mycosis. So we think about thermal waters because of that, because in thermal waters we get a lot of contact, direct contact with water. So dermatophytes, which are fungi that grows on the skin, which need keratin, of course, to grow, it might be a problem, and also essentially through aerosol treatments concerning the other fungi that can cause respiratory problems. So we did a work that showed us the prevalence and diversity of fungal isolated from thermal waters. And just for curiosity, in the background of this slide, you have a Paeocylomyces lelacinum, which is the fungi that we isolated the most out of this study. Well, how do we do this job? We collected directly from the springs, different springs around Portugal, and then we filtered the samples, 1 liter, and put in direct contact, the membrane in direct contact with the agar plates, both for dermatophytes and general fungi. We then did the identification through two methods. The first one through microscopy. In the background, you can see a fungal growth. It has radial growth, so the inside of the colony is always the oldest part of the colony. And we also did molecular methods such as PCR and also DNA sequencing to identify other fungi that we isolated. Concerning results, but again, a little bit of curiosity in the middle. What you see in the background is a penicillium, in this case Penicillium notatum, which is the most famous fungi of all, probably because it's the fungi that started the entire thing. It's the one that exudates the famous penicillin, which is found quite by accident by Alexander Fleming. Now the results. This is the prevalence that we found on thermal waters. As you can see, there is a big group concerning Purpureocilium lelacinum or Paeocylomyces lelacinum, the pink one I showed you before, and also Fusariums, Penicilliums, Cladosporiums. All of them are isolated from soil. In this case, in the next slide, you see exactly the same species, but now an average of the plate forming units we found. I'm going to talk just really quickly about some of them, because the first one, as you can see, are penicilliums. The first one is a penicillium that causes rot in strawberries. The second one, like the name indicates, it causes rot in citrus fruits like oranges and lemons. And Cytospora is famous for being a plant pathogen, one of the most important plant pathogens. The next. Are you listening to me? Yeah. Okay.
Yeah. Sorry.
Exophialia, like I was telling you about, it's an important fish pathogen. As you can see, the one that had the most prevalence only appears in the 5th place, Purpureocilium lelacinum, which is that it means it appears most, but in less quantities. Again, Botrytis cinerea is another fungi that causes rottenness in fruits. Small problem here. Okay, it's done. So as a conclusion, no dermatophytes were found. So the main problem regarding direct contact with water doesn't apply. We had a low number of isolates per sample. And like I mentioned before, all of the isolated species are originated from soil, which it makes sense because we were taking the samples from springs. So it makes lots of sense. And as I mentioned earlier, most of them are plant and fish pathogens. And the last but not least, mostly of these fungi, most of them can cause problems, mainly respiratory problems. However, only to people who have their immune system compromised. So they are mostly innocuous to immunocompetent persons. However, there are still a lot of research to do. So it's an ongoing investigation. So we hope to have more information in the future. Thank you very much. I hope you enjoyed the presentation. I tried to be a little bit quicker to catch up some time. So
thank you, Mario. So just to clarify, we just had Mario Freire talking about fungal diversity in thermal waters. Mario works in the analysis lab from Instituto Superior Técnico. And so I will leave the questions for later if we have the time. So the next speaker will be Silvia Monteiro, also from the analysis lab at Instituto Superior Técnico, telling us about wastewater-based epidemiology of SARS-CoV-2. Silvia, can you hear me?
Yes. Hello. Hello, everybody.
Mario, I think maybe you have to unshare your screen.
Sorry. Wait, just.
Thank you.
Okay. Wait, I'm looking for my presentation. I think it ran away. I'm so sorry.
No problem.
I don't seem to be able to share my screen. Here it is. Okay. Can you see my screen?
Yes.
My presentation?
Yes, perfect now. Okay.
Sorry for this small delay. Good afternoon. First of all, I would like to thank to the organizers and particularly to Professor Isabel Sá Correia for giving me the opportunity to talk about the trending organism in a more environmental rather than clinical point of view. So I'm going to be talking about SARS-CoV-2 in wastewaters, and just a small, oops. Just a small background information. By the end of last year, 24 cases of pneumonia of an unknown etiological agent was reported by China. And in January, the disease, later called COVID-19, was known to be caused by a novel coronavirus named severe acute respiratory syndrome coronavirus 2. And by March, a couple of months later, the WHO already declared COVID-19 as a global pandemic. As of today, we have worldwide close to 30 million reported cases, which amounted to almost 1 million deaths worldwide. SARS-CoV-2, as I said, is the etiologic agent of COVID-19. It is a member of the coronaviridae family, and as all viruses, it's very simple structure. It's an enveloped virus with single-stranded positive sense RNA, so a very simple structure. The current testing for SARS-CoV-2 is based on the monitoring of respiratory system samples and saliva by RT-qPCR, which only gives us syndromic surveillance, meaning it needs report from symptomatic persons. What do we do with the asymptomatic population, which is estimated to be between 20 and 45% of the total infectious cases, or with the pre-symptomatic population? This kind of testing does not give us any information about that. It also doesn't give a almost real-time information on the trends on the population. If it's increasing the number of cases, if it's decreasing, it's not almost real-time. So what can we do about this? There has to be a better way. And we have not a better way, but a complementary way, which is called environmental surveillance, which has been used for various reasons throughout the years in tracing contaminated water sources causing outbreaks, in testing particularly, which is what interests us, the pathogen and disease surveillance complementary. And the best known case and the most successful case is the poliovirus and the poliomyelitis. So with the global polio eradication effort, we have two different surveillance indicators. We have the first one, which is the gold standard, which is the acute flaccid paralysis reporting. And complementary to it, we have the environmental surveillance by testing sewage and other environmental samples for the presence of the wild type poliovirus or the vaccine type virus. Monitoring this type of samples allow us to do the monitorization of the vaccine and how it's working. Also to track silent circulation of the virus, because if we don't have any symptoms, we can have the virus circulating either way. We can also better access the eradication of possible existing outbreaks or zones where it's becoming it's circulating again, and also to document the transmission and the importation of new cases. There's a very interesting work on the surveillance of vaccinal poliovirus in Kenya. This was done during 2015 to 2017. And the last case of wild type poliovirus in Kenya was in 2013, the same as the last time they detected it in the environment. And we can see that by having the vaccine, the trivalent vaccine, we can see that we had a lot of positive detections here in orange for the type 2 circulating vaccine-derived poliovirus. It emerged after the eradication of the wild type virus. And in 2016, at a given time, they decided to change, given the emergence, they decided to change the trivalent, removing the wild type, the vaccine type 2 poliovirus, and just giving the people the bivalent vaccine. And we can see that from then on, we don't have a lot of, here in the 16th month, we don't have, from then forward, we don't have a lot of positive detections of type 2 vaccine poliovirus, which means that the virus stopped circulating within the population. So it's a very successful way of using water in complement, wastewater in this case, in complement to the clinical surveillance. How about SARS-CoV-2? How can we use it? So, for instance, the name changed. It's not environmental surveillance, it's wastewater-based epidemiology. And these methods have never, ever been applied to assess and predict the disease in the population in a systematic way. What do we know that makes us think that it can be good to use it? To start with, we know that patients infected with SARS-CoV-2 have virus excreted in their feces, and they are in approximately 50% of the samples, regardless of gastrointestinal illnesses or the severity of the disease. Even asymptomatic people can have viruses shed in their stools. One study has detected and was able to culture SARS-CoV-2 virus from the stools, whereas another one failed to do it. So we still don't know if there's SARS-CoV-2 infectious virus particles in the stools and in the wastewater, of course. Another important aspect is that when we have a place where we don't have any cases and we start looking at the wastewater and we find SARS-CoV-2 RNA in the wastewater, then it means that we have either asymptomatic people or even pre-symptomatic, which means people that have the virus will develop symptoms but still don't have it. What can we see in the wastewater looking at SARS-CoV-2? We can see trends, which is really important to see if the numbers are increasing in the wastewater, implying that the numbers are increasing also in the population. We can see the decrease of the curve when we have the phase out of the epidemiologic curve. We can track the disease prevalence in the community and try and identify hotspots of infection. And so far, we don't know much about the risk to the wastewater treatment plant workers because it's not easy to isolate this virus from wastewater. And from what we know now, from what we know now so far, there are no the virus in what we detect in the wastewater is mainly RNA rather than infectious virus. We don't think that there are a lot of infectious viral particles in the wastewater. So what are the implications of looking at SARS-CoV-2 in wastewater? Well, we have an early warning system because we know that viruses are shed, or RNA is shed in the feces of infected people 4 to 7 days before the development of the first symptoms, if we have the symptoms. So, and it was proven that there is a correlation between the patient reports and positive results in sewage of about 4 to 1 week, which means that we have at least 4 days to anticipate and prepare for, in this case, a new wave of viral infection, or in the case of possible future outbreaks. We can look at the wastewater and have in advance an idea that we have the virus circulating in the population. For instance, it is known, and it was on the news, that in Barcelona, the virus was detected in wastewater samples from March 2019, so really early. And this could give us an heads up so that we can put in place all of the instruments we have to combat these outbreaks and these pandemics. As a result, several groups throughout the world have been working and applying a wastewater-based epidemiology for SARS-CoV-2, and the first report was in the Netherlands. As you can see in this figure, there are a lot of blue balloons throughout the world, which shows you that which means that these groups are working in the detection of SARS-CoV-2 in wastewater. And it's been a really enriching experience because people are sharing their results, sharing their problems, sharing their work, sharing their materials. It's been really rewarding to work in these difficult times doing this kind of work, because people have gone far and beyond. I'm going to present you 2 cases where there are a lot more, as I showed you, where SARS-CoV-2 wastewater-based epidemiology was used. And as you can see, this one is in the Netherlands. And you can see in orange the graphic with the concentration of SARS-CoV-2 per milliliter in wastewater. And you can see that it follows really well the epidemiological curve of prevalence in the Netherlands, where we have an increase in the number of cases. We have an increase in the concentration of SARS-CoV-2 RNA in the wastewater, where we have a decrease. It follows simultaneously the decrease on the concentration of virus in the wastewater. And we can even see here in July, at the end of the graph, that we have an increase in the number of cases of the prevalence of SARS in the population. And immediately we see also an increase in the concentration of SARS-CoV-2 in the wastewater. Another good example is in the US, which was vastly affected by this pandemic. Here, this one is in Virginia. And the graphic on the left, you can see here where we have at the beginning until June, we have the lockdown. So the number of infected people is really steady and low. So are the numbers of the viral loading in wastewater. But then when we have the opening of the state, we can see a deep increase in the number of cases in the population, followed with a huge correlation by the concentration of virus in the sewage. And here on the right, you can see it's another graphic with different wastewater treatment plants in the state. And we can see that from June forward, there's basically an increase in all of them, which was reported to small hotspots of infection in the service areas of these wastewater treatment plants. So it really mimics with days ahead the impact that we see of the pandemic in the population. In Portugal, we also have a project ongoing that we're a part of, which is called COVID Detect. And basically it looks into the wastewater for the detection, quantification, and modeling of SARS-CoV-2 in wastewater as a watchdog for the dissemination of the virus in the community. So what we have been analyzing wastewater from 5 different wastewater treatment plants in Portugal on a daily basis, covering around 20% of the Portuguese population, and also daily analysis of 3 COVID-19 hospital wastewaters since April. Unfortunately, I cannot show any results because this data isn't published yet. So, but you can see from the previous examples I gave you that it works quite well and in advance to the increase or decrease of the levels of infection in the population. So what from these few months, it's been 5-6 months since the beginning of the environmental surveillance or wastewater-based epidemiology of SARS-CoV-2, what have we learned from it? We know that testing wastewater, we test thousands or millions of people, of infected persons at the same time, rather than just one person. And we test not only symptomatic, but asymptomatic, pre-symptomatic, and post-symptomatic population. We can see trends in the virus spread over time and even go to small hotspots of infection. Wastewater-based epidemiology should be complementary to the health surveillance because it gives us a heads up of what's coming. And since it is scalable, it's cost-effective, and it is a rapid way of doing analysis of a large population. It can be really useful in countries with limited resources in helping them with policy making, particularly in the case of pathogens such as the polio that worked really, really well, and also for antimicrobial resistance. So that was it. Thank you.
Thank you, Sylvia. Thank you for your presentation, very interesting. So I have just a couple of questions. So the first would go to Mario. Are you there, Mario?
Yes, I am.
Okay, so the question for you would be, so you showed us what species you have been getting in the analysis of thermal waters. So my question would be, how do you see the application of your work? Are you having some collaboration to study the mechanisms behind the survival of these organisms, or is it known?
No, not quite. We're just at the preliminary study, so we are just seeing what species we find, because nowadays only bacteria are searched in thermal waters. So this was a preliminary study to see what appears and to see if it's necessary to check for other microorganisms.
Okay. So now changing to Sylvia. The question would be, so you said that you cannot comment much on the results, but can you maybe make some comments on how does the situation in Lisbon compares to the examples you gave us? Just some no detail information.
Well, in our work, I can tell you that we can detect SARS-CoV-2 in the wastewaters. So, and we have been detecting in Lisbon. So I cannot go any further, but we find it in the wastewater. Yeah.
Okay. Nice work, both of you.
Thank
you. I guess I will go back to Joe. Thank you very much, Sylvia and Mario, for your contribution to the day.
Yeah, thank you very much. Very interesting. Combination of talk.
Bye-bye.
Yeah, thank you for your efforts and information. I mean, I've learned so many things this day.
That's true.
Yeah.
It's both tiring but interesting, yes.
So informative. It's just like a stream of knowledge. So we have a few minutes before our next speaker is meant to arrive. So I just thought I would, because we haven't covered it in the live stream, although we have tweeted about it, but we have all the winners now for the microbe art contest. And art is a visual medium, so it might be quite fun to get this up on the live stream. So let's have a little look here. If I just start sharing my screen, we can see visually all the winners. So is that working okay? Can you see everything here? Oh, you're muted, but you nodded.
Now we can, sorry.
Yeah, okay, wonderful. So yeah, so the microbe art competition of 2020 has several categories. We had agar art, cartoons, illustration, painting, best photography, best physical piece, and best writing. So let's jump straight into it. If you haven't seen the winners already, although I'm sure many people have, it's just nice to recap them all. So for best agar art, we had this wonderful Vitruvian Man by Naomi Mathy. And I just think that the detail here is just amazing, that these are all made from microbes. It's quite outstanding, really. She said she uses a toothpick to make these, but even with a toothpick, I think I would still struggle to draw these circles and squares out of microbes. So well done to her. All of our winners are going to be receiving prizes. Exactly what the prizes are is still under decision, but they will all be microbe themed, and we're trying to pick prizes that are suitable for each winner. Then we had best cartoon, which was this beautiful kind of illustration of the microbiota diversity. I think we just loved the colors and the form and all the beautiful microbial shapes that they managed to get into this quite simple but still quite complicated image. And the bubbles just add a nice airy and beautiful feel to it all. So that is Joana Carvalho.
Portuguese name.
Yeah.
Carvalho,
Portuguese day. I mean, I've learned or been, forced to come to terms with much more Portuguese as a result of this Femmes Portuguese Society collaboration. So it's been a fun thing. Then we had best illustration and painting, which was this kind of mixed graphic of so it had Pathogenicity Island. So it has elements of agar art, but then it's got graphic design layering it all in together and then kind of like confronting it with the human condition. And we have kind of like, for me at least, it's the juxtaposition between the good and the bad sides of the microbes coming together with kind of the anthropomorphization of the human face. But yeah, beautiful. Then we had best photography. Now this was really difficult, and we had a vote in the end, so the vote decided the winner, but we had lots of mixed opinions for best photography. It was a hard one, to be honest, but in the end we have this beautiful macro view of Phytophthora infestans growing on some kind of medium. But the fact you can see all the individual filaments and the structure of the microbe world here is just so nice. So Mons de Vreza won our best photography prize. Then we have our best physical piece. I think this one was quite unanimous.
Oh, that one I saw, and it's very impressive. Yeah,
it really is. So I mean, it reminds me of Micropia a bit because they have these beautiful petri dishes with molds going in, but then kind of assembled into this. And it kind of just indicates the interaction between plants and microbes, which is so important.
Is there any relation between the species that we see on the plates and the tree itself?
I'm not sure. Should we see the original post?
Where did this person do this?
Endophytic and rhizospheric fungi associated with the grapevine. So they are fungi that grow with grapes. And quickly before we run out of time, best writing was this poem by Chris for Christina Garlada. So we have this beautiful poem here, which of all of the ones submitted by IBBS, we voted as our favorite. So again, you can find it on the blog on our website, and we'll also put one on the IMD website. Then overall, the best art, and perhaps even the most accomplished artist, just from the sheer numerousness of the things she submitted, was Anna Dumitriou. So just so many cool conceptual pieces here. Too many to really run through today in this short segment, but yeah, just so many amazing, creative and scientific and artistic pieces here. So these are all grown out of bacteria on the lattice structure to make this lace. Really cool. So all the winners are here. And just a final thing is that the art contest with the hashtag has had over 2 million impressions, and nearly 1 million people have seen it. And yeah, hundreds of people have posted about it, and we've had thousands of posts. So massive
improvement since last year, right?
I think so. Well, I mean, I can't remember exactly what it was for the microbe art contest, so I'll dig into the data and see if it's bigger than last year. But let's move back into our series of talks after our brief interlude, because we have Karen Lloyd joining us.
Hello.
Hello. Hi, Karen. How are you doing?
Good. How are you?
Good. So you were joining us from University of Tennessee, or not from it, I guess you're at home, but that's where you're currently affiliated. And yeah, you have your full 20-minute slot unused to deliver your wonderful talk, What Microbes Live Below Our Feet.
All right, cool.
So I'm going to just get out of the way and let you begin, but we'll just make sure, screen sharing is all okay.
Okay.
It's a nice busy desktop. Good. Cool.
Yeah, I know. We go in each other's homes and see each other's desktops now, which is what we do. So I'd like to tell you guys a little bit about the microbes that live beneath our feet. And this is a picture that I took while studying deep subsurface microbes in Argentina. When you think about life on Earth, you can't just stop at what we can see on the surface. My colleagues have added up the total estimates for the amount of microbes that live in the mud that's buried underneath the oceans all over the Earth. And it turns out that there's more living microbial cells there than there are estimated stars in the universe. So this is actually a huge ecosystem, and we know very little about it relative to the other ecosystems. More recently, folks have added up the total number of microbes that are present under our feet on land. And this number actually increased the total number by quite a bit. It more than doubled it, because there's potentially even more microbes that are living underneath the continental surface than there are under the oceans. So what this adds up to is, how do we do this? How do we study this vast biosphere under our feet? Well, I like to think about the Earth like this. When you take all the water away, the oceans look pretty fascinating. And it sort of emphasizes how the subsurface, thinking about the subsurface, you can see all these interesting, cool features on the seafloor. In this map, one reason I also like this map for historical reasons, because it was published in 1977, which is a long time ago. And it's not that far off from the much better estimates that we have today. And all of this data was put together by individual seismic data that came back from many, many cruises all over the world by this woman, Marie Tharp, who studiously put all the data together. And she's the first person to actually figure out that there were seams running through the seafloor. So I sort of went through recently and put dots on every place that I could figure out had really good microbial characterizations of the deep subsurface at some depth. And it's not too bad, except that if you imagine that you're trying to talk about this entire ecosystem from just these dots, suddenly you really miss a lot of complexity. Like if we were all in an alien spaceship trying to describe the subsurface, and this is all, or trying to describe the surface of Earth, and these are all the dots that we had, we would never be able to come up with the whole picture that would be accurate. So what we need to do is we need to just keep going forth and studying this massive, mysterious deep subsurface. And the normal way that microbiologists such as myself study things are that, we grow things, we put them on petri dishes, but there's a couple of problems with this. And the biggest problem with just approaching things through culture is that now that we can get DNA sequences from life in lots of different places, we have discovered that if you look at the total tree of life, so this is, you can imagine this like a family tree. So sticks that are closer together would be more like siblings, and then farther apart are more distantly related items. Every single major branch that has a red dot on it is something that no one has ever grown in culture. And what this means is that as you look, this is just the archaea, but the bacteria look pretty similar. most of what's out there, most of the deep branches on the tree of life have never grown in culture by anybody ever. And so that means that for now, we're kind of stuck with just looking at their genes and trying to infer what they're doing from their genes. But another reason why we can't depend on cultures is that how something acts when it's on a petri dish, touched by our gloved hands, is probably going to be a little different than how it acts actually out in nature when it's doing its own natural thing. So for these reasons, my lab and the way I approach things is often to just try to figure out as much as I can about a microbial subsurface ecosystem without messing with it. So I try to disturb it as little as possible and just gather all the information sort of passively that I can about it. So today I want to tell you about some really cool recent work that we've been doing in Svalbard, which if you've never heard of this amazing place in the Arctic, this is, I just took this off of Google Maps, but this is the archipelago called Spitsbergen. Svalbard is the name of the biggest island of that archipelago, and it is very, very far north of the northernmost part of Norway. It's pretty much co-equal with the upper tip of Greenland. So this is very, very high latitudes. It's about 79 degrees north. It takes a long time to get there. And this is the work that I'll be showing you was mostly done by two of my students, Joy Bongiorno, who's now a professor at Maryville College, and Katie Sipes, who is still working with me. And this was largely funded by the Simons Foundation, although we got much needed help also from the French and German research station in Svalbard, AVIPEV, and the NSF, the National Science Foundation in the US, and the Center for Dark Energy Biosphere Investigations. So what they did was they went up to Svalbard and they sampled marine sediments in these fjords. So this is a close-up of just one of the fjords of Svalbard. And you can see all the glaciers are sort of emptying in to this fjord, and they went out on a small boat, dropped cores off the back of it, and took some samples and brought them back up. And from this, Joy was able to put together a vast, diverse ecosystem of largely organisms that are eating and or breathing iron and sulfur compounds. So despite the fact that there's all these strange organisms in Arctic sediments that we don't necessarily know so much about, it turns out that many of them, we can actually infer their physiology to be using iron and sulfur compounds. And it's not simply that they're there sort of dormant and waiting in the subsurface. They are actively doing lots of things with iron and sulfur, and they're pretty active microbes. And the reason why we think we saw so many iron and sulfur cycling microbes is that anytime you fly over Svalbard, to get around to your field stations, in front of the glaciers are these massive just pushes of iron. You can see this red stuff coming out of the glaciers. So this is really specific to Svalbard because it happens to be hosted by a lot of iron-rich rocks. And there's red Devonian sandstones that are getting dredged over and glacially dredged and spewing into the fjords. And so our hypothesis is that this is what's feeding the microbes. So you can kind of see it as a subsurface glacial fed ecosystem where the microbes are eating the iron and the sulfur that's getting pushed off by the glaciers themselves. And this is just another picture I took from a plane where you can actually see all this iron streaming off the edge of an iceberg. So the icebergs sort of float this stuff out into the middle of the fjords. But this work really was just kind of a presence absence thing. The really big challenge is to figure out how to study the actual activity of microbes as they're out in nature. If we have a pure culture, we can watch it make a certain chemical, or we can watch it eat another chemical, and we can say, yep, it is making methane, or it is eating iron. But it's hard to do that when you just collect a mud sample from the subsurface in the Arctic, as we did. So one thing you can do to study the activity of microbes out in nature is that you can look for not just the DNA, you can look for the RNA, which gives you a measure of how active those genes are, how much they are turned on into messenger RNA to eventually get turned into proteins. So here's an example that is not from the subsurface. This is sort of the opposite of that. This is Bordetella pertussis, which is the bacterium that causes whooping cough. This is its genome stretched out end to end. So each little gene has its own little dot here. And the y-axis tells you whether that particular gene was upregulated or downregulated. And this is cool. I mean, this technology, which is often called RNA-seq technology, if you're working with cultures, can tell you so much about the physiological state of an organism. You can say, oh, yep, this gene region, those are getting turned on. This gene region, those are getting turned off. And you get this really high resolution data about the activity of your cultures. Well, we wanted to do that, not with cultures, but with Arctic mud. And as far as we know, people have done this in a couple of other environmental samples, but nobody had done this with Arctic mud. We were gonna have to pick apart the signals from the RNA coming from one organism versus another organism. And we just kind of crossed our fingers and made the measurements and hoped that we could see something as pretty as you can see with whooping cough. So what does such a transcriptional genomic map look like if you make it on a natural Arctic microbe that's living in the wild? Well, the first thing we did was we wanted to get the actual genome to stretch out like that and look at the transcripts. And we happened to catch one that, these are not photographs, microphotographs taken from my lab. These are from another group, but this is a uncultured group called the Wosiales. It's within the gamma proteobacteria, but it does not have a close relative in pure culture. So it is one of these microbes that is kind of mysterious to us. And one reason why we find it so interesting is that it tends to be the dominant microbe in sort of the upper layers of marine sediments. So we kind of have this feeling like it must be doing something important. We just kind of can't quite figure out what it is. We know from work that other teams have done that this is, it probably can fix carbon. So it's probably an autotroph on some level. And this group made lovely pictures of it where the RNA is in green and its DNA, it's wrapped around a DNA core that you can see in blue. And it has these interesting morphologies. So we were lucky enough and we caught one of these genomes. We got it pretty complete and we said, okay, now when we extract all our RNA from this environment and we make that pretty map like we did a whooping cough, remember that one that had lots of genes upregulated and downregulated, what's this gonna look like? It didn't look like that at all. It blew our minds. We did not expect to see this at all. Look at these genes. If you line up all the genes across the genome, certainly some of them are going up and down. We kind of, if we zoomed in on this, it would look like the whooping cough genome. But look at this gene. That is one gene and it went crazy. There's tons and tons of transcripts of just this one gene. And it didn't matter whether we went to one station in one fjord or a different station in that fjord, or if we went to a different fjord entirely, like drove a boat around and took a sample from another place. It didn't matter where we went or what depth we looked at. We got huge quantities of just this one gene. This pretty much, you know, this is one reason, this is my plug for doing exploratory science. I never would have written the hypothesis, hey, is there only one gene that gets highly transcribed in this organism? I wouldn't have enough information to even ask that hypothesis, but here we have it. So I can't tell you exactly what's going on with this. It's still somewhat of a mystery to us, but one possibility is that this gene looks a lot like this gene, which is present in this cultured organism called Stigmatella aurantiaca. And this group put gold particles so that you could see them on where this gene is getting expressed into proteins. And 6 hours after this organism starts turning into a spore, so a spore is a dormant phase. It's a very highly specialized dormant type of microbe, and only certain types of microbes are capable of making spores. It starts to form these different types, just this one protein, and expressing lots and lots of this protein. And then a full day after it starts making spores, this protein sort of clusters up on itself. So that's our best hypothesis right now, is that maybe this uncultured Arctic microbe is actually turning into spores as they're buried in the Arctic sediments. And this would make sense because if it's dependent on oxygen, or if it works great with oxygen, then as it sort of gets buried under marine sediments, it can harden down and become dormant, and then maybe wait until, you know, an iceberg dredges across the bottom of the fjord and pushes it back up to the surface again, where it can become a vegetative cell again. But that's just a hypothesis. Now we have a hypothesis. So we will definitely be doing more work to see if it's actually doing this out in nature. So to finish up, which I guess I blew through that very quick. I was overly worried about being too long. So I always end up going too short when I do those things. Maybe people will have questions. There really are many, many mysteries yet to solve about the organisms living in Earth's crust. I find this fascinating. It doesn't matter where we go or what questions we ask, something cool is going to be down there. And now, sort of as microbiology and molecular biology has progressed, we really have the molecular tools to do the same sort of high-level molecular work that we can do on cultures, but we can do it actually out in the wild on natural samples and start piecing together what these things are actually doing out in nature. So I will stop my screen share, and we can either have...
Thank you. Yeah, no, I have a couple of questions. If I'm visible again. I am, cool, yeah. So yeah, you're looking at microbes that are living in the crust.
Yeah.
But how deep down can we find these microbes?
Yeah, the deepest that anybody is on record of finding a microbe right now is about 5 kilometers.
Okay, and is that purely because you can't drill much further without it costing too much?
Yeah, and it's also, we think that there's going to be an ultimate limit because there's a geothermal gradient. As you go deeper and deeper in the Earth's crust, it gets hotter and hotter. Currently, the known upper temperature limit for life is 122 degrees Celsius, which that usually hits somewhere between the surface, if you're at a really hot spot, and it can dive down to maybe 20 kilometers, would maybe be the deepest, maybe a little deeper. Or that may not be the upper temperature limit for life.
Because I mean, even with that kind of thickness, if we think about a sphere and the depth, that's a huge amount of space that you can find microbes. So even if they're not beyond 20 kilometers, I mean, are we looking at most of the microbes being deeper than the surface? If you just look at them, yeah, okay.
Yeah, if you like picked the height of the center of mass of microbial life on Earth, it would not be at our feet. It would be slightly below it.
Yeah, okay. So they're beneath us more than around us. I mean, they are everywhere. But I mean, you know, there's been a lot of hype in recent media about finding phosphine in Venus. And here you have, you know, you'd have to have microbes living at high temperatures, perhaps high pressures. So are the microbes that live in the mantle or the crust perhaps places to look for kind of life that perhaps would be similar? I don't know. But they would be fairly extreme to be living deep and hot and under high pressure.
Yeah, absolutely. These make awesome analogs for life on Venus or any other planetary body. And there's a cool thing about high temperature and high pressure. High pressure has a tendency to make lipids more ordered, and high temperature causes them to be disordered. So they can kind of work against each other, and you might be able to get them down deeper at both high pressure and high temperature, which is kind of cool.
Okay, so it kind of feels more normal for the bacterium.
They
They love
know they're extremophiles.
It's true. They think we're extremophiles, I guess, living in weirdly cold environments, or there's just nowhere near enough acid. Cool.
Yeah.
Awesome. Well, thank you very much for your talk and joining us on International Microorganism Day, all the way from Tennessee, and representing, the US in this mass international live stream of talks.
Well, I'm glad you guys are doing this. This is pretty darn cool.
Yeah. Well, so we're recording everything. So hopefully within the next couple of weeks, we'll get them all on YouTube. So all the videos will be watchable because, you know, there's been so many. I mean, it's impossible to watch them all just because of the time constraints and the time zones. So hopefully we can all then watch the ones which take our fancy if they weren't there when we were awake.
Maybe you'll discover that somebody was been staying awake on 5-hour energy and coffee and did all 24 hours.
Yeah, I mean, that's our freelancer, Ben. He's basically been doing that. And apart from 3 hours of sleep, I've kind of been doing that as well. So yeah, it's been a long one, but it's been great fun. Well, thanks for joining and submitting your contribution. And well, hopefully people can see you again on YouTube. But thank you very much.
Thanks for having me. Bye.
Cool. Okay. Ooh, do we have Ben? Or is he just muted? Okay, nice. Right, we've got a few minutes before the beginning of our next talk, but we might as well get it started because we've got a few long talks coming afterwards. So let's see if we're ready. Cool. Okay. I think, yeah, great. So joining me now, we have Bamidele Odumosu, who is the publicity secretary of the Nigerian Society for Microbiology and is also a researcher at the University of Lagos in Nigeria. So let me just check audio and the camera's all working. Let's have a look. Can you hear me? Am I coming through okay? Ooh, it's still muted at the moment. Let's just fix that. Okay, cool. Can we hear you? Can you just check?
Yeah, I can hear you. Can you hear me?
Yeah, now we can hear you. Perfect. Okay, great.
First of all, how are you, Joseph?
Yeah, not bad. We're towards the latter end of the live stream, so it's been a long haul, but it's all been very informative, and I've learned a lot.
You must be exhausted.
A little bit, maybe.
That's nice. Well, I'd like to appreciate the Federation for European Microbiology Society for affording me this opportunity. I'm not taking it for granted. Thank you for providing me this opportunity. In my presentation today, I'm going to be talking about the activities of microbiology in Nigeria and also talk about microorganism of interest in my own area of research. I hope you'll go along with me.
Cool. So you've got a presentation. So I guess if you want to begin,
Yeah, just something to guide my talks. Yeah, I don't want to miss the line. I want everything to go sequentially. Yeah. So the Society for Microbiology in Nigeria was established in 1972. So our main agenda has been promoting science, precisely microbiology. We are the biggest society for microbiology in Nigeria. And as a matter of fact, many other associations related to microbiology are like, it's like a parent body for these smaller organizations, societies. And we have zonal coordinations because we are widespread in the geopolitical zone of Nigeria. So we have the Southwest, Southeast, Northeast, North Central, and Northwest zones, which are all coordinated, all towards the central executive. We have a central executive, which I'm privileged to be the publicity secretary. So our activities include symposium, workshops, conferences, as well as mentoring our younger microbiologists.
One second. Is the presentation on your screen? Because I don't think it's registering on us just yet. I think it's got the wrong window.
It's not showing on my screen?
So we've just got the folder, not the full presentation. It might just be you have to share the different window.
Okay, let me share my screen properly.
I think it will work.
I can go on without the screen, without the presentation if my folder is not showing. Hold on. Is my screen showing now?
Yeah, but just the Explorer window, not the presentation yet.
Well, I don't know what's happening.
This has happened before today. It's one of the possibilities with having so many calls.
Has that happened today before?
A couple of times. I think it just might be the window you select. But if you don't need it,
okay. I'll just go ahead without the sharing of the screen. I'll stop sharing screen. Let's just go one-on-one like that. Okay.
Cool.
So like I was saying, we have our activities, which is where was I? We mentor young microbiologists. So we have the younger arm of microbiology we call Nigerian Association of Microbiology Students. So the idea is to bring them up, to induce them with microbiology, and when they graduate, they also become microbiologists, and they also get to practice and efficiently participate in development of science in the community. So what we have our major challenge regarding that. Our major challenge as a society in Nigeria is professionalization of microbiology in Nigeria. Our society is having this huge challenge. Our graduates are not having a say in practice. What I'm saying is that they're not being given opportunity to function whenever they graduate. We have some professionals who are intruding into our space. They are preventing graduates of microbiology from practicing, especially in the hospitals and medical facilities. And this is a huge problem.
So you mentioned
because
I said you mentioned to me that you've been working quite hard though on this problem as a society and helping to get legislation passed within your own.
Yes, that is the professionalization. Yeah, so we have been pushing a bill so that microbiology as a profession in Nigeria will be professionalized. We don't want to take the back seat. We are in charge of microorganisms, from crying out loud. Take, for instance, COVID-19. This is a virus. Nobody knows about virus more than a microbiologist. Do you agree with me, Joseph?
I would say, yeah, virologists are probably the best people to go to.
Yeah, but you find out most people who are first responders to this COVID-19 are not even microbiologists. Most of them get contaminated, or doctors get infected with COVID during the treatment, despite their protective gears. And you as a microbiologist, you know how to handle this organism better than anyone else because you were trained to handle microorganisms. But we were not given a say. We're pushed at the back seat. That is a recent issue about the pandemic. Previously, when our students graduate, even currently, they don't allow them to work in the hospitals. We have some society that is called Medical Lab Society, Medical Lab Sciences, which are paramedicals, which takes a seat because they got professionalized before us. So they prevent the real microbiologists from practicing, and this is a big issue. So
I was going to say, especially microbiology seems to have been in Nigeria for a very long time because, the Nigerian society is older than our society, FEMS. So it seems to have been.
How old is your society?
1974. Yeah.
Well, I would say your society, we are older than you, but you came from a more developed country. So you get a lot of things done easily than we do. So as a matter of fact, I'm kind of using this platform to also plead with international organizations to lend a voice to our society so that we can take the rightful place. Most of our students are getting discouraged by the day. You know the pain. Most graduates of microbiologists, they leave the profession and go elsewhere because they can't get job placement. And this is a burden we are trying to, get off our shoulder. We are pushing hard to get this professionalization agenda.
So actually, I had a question because one of the things that lots of societies do is the thing you're trying to do, which is to affect policy and policymakers. So I was wondering if you had any advice, because you said that you were, approaching a point where perhaps the legislation is going to be passed for professionalizing microbiologists, but it's been a long job. So do you have any advice for how to do this kind of project? What kind of things could we tell other societies or similar societies to get this kind of legislation passed?
You mean similar societies so they can get this?
Well, yeah. So working with policymakers and politicians is, famously difficult. So did you have any advice for any other societies who are trying to do something similar?
Well, the advice I would give other society I'm not really in a good place to give advice because we are still battling with this problem. You know, if we have been able to overcome this problem, I'll be in the right place to give advice. But if I have to be in the right place to give advice, I would say they should not allow every other society to intrude into their space. They should take their rightful place, lend their voice, speak out, make their presence known before any other smaller organization take their place. I think that's the problem with us. We have allowed smaller organizations, smaller society make much noise than we do. So they get recognition before we got recognition. Not that we are not recognized as a society, but practically we are not recognized. So that is my advice. But like I told you, I'm not in the right position to give an advice because we are still battling with this challenge.
Okay. I think, yeah, microbiologists have often found themselves on kind of the back foot, and, you know, sort of other voices have come into the conversation who are perhaps less expert. So it is something that I think is, yeah, faced by many societies. But I wanted to also ask, you mentioned in a previous call that you also managed to organize lots of conferences for your members across Nigeria. So do these conferences help you to invigorate your members to perhaps have a stronger voice?
As a matter of fact, our annual conferences is where we have more members. You know, this gives us more publicity. Like this year, we couldn't have our conference because of COVID. Likewise, everyone is having this issue. So this gives us opportunity to have more members come to join, and they get to know about the society, and also we generate income. That's one of the problems we have as a society. We don't have sufficient income because one, poor membership subscription. So members don't pay annual dues regularly. I know this is also a problem which, like you told me the other time, in some smaller society in Europe, they also experience this, but it's affecting us more because our income is poor. Most members come to us because they need a certification to belong to a society. Once they get certified, they abandon us. They don't even renew their membership. I'm a member of ASM, SFAM. I renew my membership. I'm a member of FMs. So, you know, we try to be committed, but commitment with Nigerian Society for Microbiologists in Nigeria is poor.
Okay. Some interesting problems here. I think you mentioned in our last call as well that at that point you were hopefully confident that the legislation was going to be passed soonish. Has there been any change to this since we last spoken, or?
Yeah, the first reading to the bill passage was done last year, November. Now the bill has to be read twice. I don't know much about legislation, but I know I have to read the bill twice. Then there'll be a public hearing. Then the president will sign it into law. So the first reading has been done last year. Now we are pushing for the second reading to be done now. The second reading, we are hoping maybe this year or early next year, because we are at the point of getting the second bill to be read. But you know, politicians, we have to lobby. You have to make some leverages. If you don't have financial muscles, you have to do a lot of things. And we are a scientific body, for crying out loud, to do all these things. And we have a poor income, is an Herculean tax. So we are hoping that based on the promise given to us by our representative at the National Assembly, because we have notable people who are microbiologists. We have more than 4 vice chancellors of the Nigerian University that are microbiologists. We also have a sitting governor. We have a deputy governor. We have many people. We even have senators who are microbiologists. You know, because politics have made some people to abandon their profession, but then they have their first passion. So people like that are helping us push the bill at the floor of the House. Now, when such bill is not having some political, you know, how will I put it, some political energy, it gets to be watered down. So that's why I'm emphasizing that international organizations and society like FEMS, ASM should lend a voice to Nigerian Society for Microbiology and save microbiology in Nigeria. I'm saying it passionately because our youths are losing interest.
So it's, yeah, sort of, because I mean, you said you relied on the politicians and the vice chancellors, you know, the powerful members who have a microbiological background to push this thing into, I guess, is it Parliament or the Senate? What's the name of the legislation body in Nigeria? I guess that, yeah, whatever the bill is read. But you need them to be replaced in the future, right? By new people coming up who are young scientists and eventually getting to positions of power. So what do you think would be the most helpful way to engage young Nigerians in microbiology? What do you think is the format or the medium that will help bring them into the subject?
If we talk about professionalization, it's going to encourage them because they will be trained. We will have Microbiology Council of Nigeria. And when they graduate from their first degree, they go into training into real aspect of microbiology they are chosen. You know, this training is going to be licensed at the end of the training. And you know, it will encourage students to apply to be a microbiologist because you are going to be licensed to practice microbiology. Then a lot of young microbiologists with ideas into how they can fashion microbes into economical value. In food microbiology, in pharmaceutical microbiology, we have several microorganisms of economic value that can be used to generate income. You don't have to depend on the government.
Yeah. Okay. So it's the way of incentivizing it for the youth. You know, this is a great profession. You can make it something valuable and productive.
Exactly.
Yeah. Okay.
I'm speaking on that. Okay.
I was going to say, we've only got 6 minutes. So I wanted to make sure we had time to cover bits of your research before we go.
Like I discussed that previously with you, I'm interested now in marine environments, in discovering of antimicrobial agents. By profession, I'm a pharmaceutical microbiologist. I'm interested in antimicrobial agents. So I'm looking at actinomycetes as the microorganism of the day, which is, you know, known to produce a lot of metabolites. They represent 3/4 of microbial products for chemotherapy nowadays. Now, my current interest is into marine environments. Nigerian marine environment has been underexplored for antimicrobial agent bioprospecting. I wish my slide could have been displayed. We have a preliminary study we have carried out. We have published 2 articles which have identified the Lagos Marine as a rich environment with potent organisms with activities. We have seen anti-cancer, antivirus, antifungal producing actinomycetes, which have been isolated and published. So that's what I'm working on currently now.
So have there been any, you know, previous studies into all of these things, or is it quite an untapped area then? You said it was underexplored, but has anyone explored it, or is this some of the first research of the microbes that live in the lake?
I'm not the first researcher, but we have few people who have published on this aspect. That means it is untapped. It's an opportunity for international scientists to collaborate with Nigerian scientists. We have a lot of untapped environment waiting to be explored in Nigeria. Our problem is facilities and infrastructure for novel scientific work. So if we have good collaboration with good, you know, ideas, I think we can have a lot of breakthrough using my Nigerian sample as a case study. I'm telling you a reality.
Okay. So, I mean, I remember in our journal, Fem's Microbiology Ecology, there was a thematic issue on the environmental dimension of antibiotic resistance. So some of the papers here were looking at, how antimicrobial resistance genes and the environment are interacting. And they did some research in different parts of the world. So it might be worth having a look and see if these authors are interested in looking at parts of Lagos for perhaps new research. But if I'll send you a link after this and you can have a look at the papers, but they sound similar in the kind of research that they were doing. But so have you ever then spent some time having to dig for your own samples in the Lagos Bay, or do you have?
Yes, I have. I took a photograph of a marine sediment that I had previously. I wish I can show you. Should I try?
Let's try again, because if you go share screen, it gives you some options on a little menu. See if perhaps you can select a screen or a different window, because it might have been, because it's just showing one window, but is there a possibility of showing a different screen? Or if you double click on the presentation, does it open up in this window?
I'm trying to double click now, it's not coming up. I don't know what's happening. Okay, let me try again. How about now?
No, it's just focusing on that little window. It's not giving you the full screen.
Hold on. Let me try a different folder.
Yeah, okay.
Oh God, I have to show you this. What about now?
No, shame. It's still giving just the little Explorer window.
I don't know.
But if you send me the pictures in an email, I can at least show them at some point.
I can send you the pictures. Do you want me to send it to you now?
Yeah, just send it quickly. We can go a couple of minutes over before we bring Jason in. And then if you send it in an email, I can get it up on the screen, because it'd be a shame not to show it, because I have this brilliant imaginary image of you digging up microbes in the Lagos Bay, and I really want to see it in real life.
Let me see.
Cool. Ooh. So is the main focus for the University of Lagos microbiology researchers medical microbiology, or do you have like quite a spread of
general
research?
Microbiology?
Yeah,
it's general microbiology.
General, so you have a whole range.
Yeah, it's general microbiology we do, but at a postgraduate level, then you can focus on some other aspects. You can focus on probably medical microbiology or pharmaceutical microbiology. No, we don't do pharmaceutical microbiology there. We do food microbiology, environmental microbiology, and so on. Oh my God, I don't know why I'm trying to send you the picture too. What's going on now? Oh gosh, what's happening? Okay.
Cool. Hopefully, in my inbox in a minute. And cool.
You should see it now. It's sent to you.
Okay, cool. It will hopefully come in a sec. Let's just see. I always wondered how long an email takes.
Maybe you can transmit it.
Yeah. Cool. Here we go. Great. Right. I do. So hopefully this will work if I just open the image. Ooh, my computer's getting a bit frozen. I think it's just a little bit tired after today's long video call. Okay. Great, there we go. And I just share my screen and we have it here. Right, cool. Nice. So have you managed to study any of the composition of this sediment yet?
Yeah.
Yeah. So what did you find on analysis?
Where I didn't do a deep analysis, what we try to do is to isolate the microorganism of interest from these sediments. So we just basically do some zero dilutions and get the sediments into a broth. Then we decant it. You know, we take some loopful, you know, the basic preliminary method. So we have it spread on the plate, depending on the protocol you want to use. We don't do deep analysis on these sediments, which is not impossible, but sometimes major focus is just to isolate the organism from the sediments.
Okay. Cool. Well, I hope you find some interesting things in here.
Yeah.
And perhaps some novel genes or anything, or novel species.
Yeah, we currently, I have a publication that was published in ASM Microbial Announcement. Is a new species of Ocrobatrum, a new species. So we are in the process of naming it now.
Okay, cool.
It was isolated from these sediments. It's a new species. Has not been given a name, so probably we can name it Ocrobatrum legoensis.
Sounds cool to me. Cool. Well, we've gone a bit over, so I need to finish the call up now so we can move on to our next speaker. But I'm glad we managed to fit all this in. And thank you so much for joining me on International Microorganism Day. And very much enjoyed having you on the call again, as before. And enjoy the rest of International Microorganism Day. And all of this will be on YouTube at one point, and we can make sure that everything is recorded. But thank you very much, and hopefully we'll be in contact again after this.
Yeah,
it's great to continue this.
Thank you very much.
Thanks very much.
Have a nice day.
Yeah, you too. Thanks for joining us on this Christmas for microbiologists. Thank you very much. Cool. Okay, where's our next speaker? We have Jason. Hi, Jason. Can you hear me? Ooh, let's just unmute you.
Wait myself. There we go. Now I'm unmuted.
Perfect. Great.
Excellent.
Sorry with a little bit of time, but it's
no
okay.
Worries.
We can run on a little bit longer into the break after with Elizabeth's talk. So I'm just going to let you begin because you're here to talk to us about our relationship with microbes.
Yeah, so if I just share the screen here, let me just get this put together so I can share. Let's just make sure you all can see what I'm seeing. And can you see that?
Yes, we can.
All righty. And I'm just going to do a very quick movement, right? That's good. You saw that?
Yes.
Good. All right. So I've given away the next slide, but that's fine. First off, I want to say thank you very much for having me here. I've been a fan of microbes. And as well, I'm just so pleased that I'm being interviewed by Tormund Giantsbane. So that just, I mean, he's my favorite character from Game of Thrones. So thank you so much, Tormund.
Well, thank
you. Oh, and I'm still on. Yay. Okay, good. So when we, as you know, when it comes to microbes, when it comes to germs, a lot of people may not be thinking that we want to be doing any kind of relationship with them. And I mean, all you have to do is look at people who happen to be germophobic, much like Howie Mandel, to basically say, you know, you can't be serious that I would want people to have a relationship with germs. And of course, this year it's kind of gotten to a point where it's now a little bit, I don't know, what's the word, toxic to even suggest that having a relationship with microbes is a good thing. However, one thing that we have to realize is that when we're actually talking about germs, we're not just simply talking about one or two different species. And if you actually look at this slide from 1979 to 2012, you know, the number of identified microbial species has been going up dramatically. And even since 2012, it's just been going up exponentially. We seem to be finding our way to understanding the diversity that exists out there. And more importantly, we're realizing how those numerous different species actually work with us as human beings. And what is really interesting is that a concept that was developed many, many, many years ago for just general ecology in general, which happens to be the idea of commensalism, mutualism, and parasitism. Now, commensalism happens to be, you kind of get along, everything's all right, nobody really notices one another. You got mutualism, and what this means is that there are people who are helping one another out, or in this particular case, conspecifics if you're talking about ecology. And then there's parasitism, which essentially is pathogens. They're out to live and survive off of you. So how does this relate to us as humans? Well, I think the best way to approach this is to take an example that we all can understand. Now, I'm in Canada, and our largest city happens to be Toronto. And if you were to look at Toronto, we have a population somewhere above 2.5 to 3 million, something along those lines. And when you look at the potential number of species that we have of microbes essentially on Earth, that's about the same. So when we take a look at all of this, the majority of people, the majority of microbial species are commensal. They just go about their business. They don't really do anything. They don't say anything. And they don't have very much impact on us. And that's fine. I mean, it's good for people to just go about their business like we see here on this street. By the way, this isn't a regular day in Toronto. This is actually during one of the parade routes. I believe it was the Pride Parade. And honestly, if you ever get to Toronto around Pride Month, and especially in Pride Week, you will have the best time of your life, guaranteed. Anyways, then we have essentially the mutualism. And these are the people, or in this case, the conspecifics, that are really aiming to improve our lives. And so we think about it from the perspective of who is in our world who's helping us. Well, I mean, to some extent, we have law enforcement, and again, that's debatable these days, but firefighters, absolutely. Teachers, you can never go wrong. Nurses, I mean, nurses are essentially the best thing for us. And of course, doctors. Oh, wait, sorry, wrong one. There we go, there's the doctor. So the fact is that when we have these mutualism candidates in species form, by getting close to them, it can actually help improve our lives. And then finally, we have the parasites. I mean, these are North American specific, but I'm sure that you have your own in wherever you happen to be across the world. Although I have to admit, Thanos is definitely one of the worst villains across the universe, so I think we can all relate. Now, when we start talking about microbial species that possibly could have the same effect, well, then we're talking about something like Ebola. I mean, how many of us have heard about Ebola, what it can do? We now know about the epidemic that happened. It's one of the really nasty ones, and we need to avoid it. And of course, we now have the coronavirus, SARS-CoV-2, COVID-19 is the disease. And so at that stage, you start thinking, okay, well, we really do need to avoid these. But the fact is how much of that should we actually be giving our attention? All right, how many of those species are there? And you know, when I give these talks in lecture halls and schools and stuff, I always like to ask certain questions, and this one is very important. You know, how many species are out there that actually do harm humans? And if you happen to be a gambler based on what you're hearing out in the real world these days, you'd probably go at number 3. But at the end of the day, the actual number is 1,450. And out of that, there's really only about a dozen of those species that you yourself are ever going to see, the sort of dirty dozen. The rest of them happen to be just good old-fashioned microbes that we see under a microscope. And honestly, we've had such great microscopic imagery that we've seen over the course of this day. If you haven't, I really recommend that after it's over, you go back and take a look. So when we first started seeing microbes under a microscope, it really was more along the lines of, oh, that's very interesting. I don't know what's really happening, but it's kind of cool. We hadn't yet made that link to pathogens and to those that are harmful. And unfortunately, really didn't happen until something really bad occurred. And that was, we realized that certain types of bacteria were actually making our beer bad. They were spoiling our beer. And quite honestly, this means war, because honestly, in Canada, if you ruin our beer, we're going to go up against you. You're going to go down. And I'm sure that there are other places around the world that would agree with me. And the problem is that this idea of war against microbes, whether it be against beer, whether it be against human health, whether it be against the environment, it has really taken over the mindset of humanity. And so a number of years ago, I was approached as what I'm still called now, the germ guy, to sort of find a way to change that perspective. Was there a way that I could develop not war, but peace, possibly even love with the microbes? And so I took upon my task about 7 or 8 years ago, and eventually it came out in the form of the Germ Code, which was a Canadian bestseller and was also nominated for Best Science Book of the Year back in 2013. And essentially what had happened is, even though the book says how to stop worrying and love the microbes, what it really came to be, as I found out, was sort of a discussion about our actual dysfunctional relationship with microbes. And so when we talk about relationships, it doesn't matter if you're talking about a rom-com, it doesn't really matter if you're talking about a novel, or just simply a talk like we're having today. There are 3 different acts that are involved. And the first act is always about how we set the stage for that dysfunction. And one of the best ways to approach it is just to say that when it comes to microbes, we really don't have much of a comparison as humans. Here's the easiest way of doing it. If you take a microbe and you give it the unlimited amount of resources, all right, how long will it take before it covers the planet? And we're not just talking like one layer. We're talking about, several layers, so that when you step in it, it's goopy. Well, the answer to that is 36 hours. Yeah. Essentially, these things can multiply within 20 minutes for the most part. Sometimes they can be faster, a lot of times they're slower. But at the end of the day, if you give an E. coli, which is pictured here, Escherichia coli, unlimited resources, and you give it unlimited space, 36 hours, it'll cover the planet. That's really hard for us to understand, because even at 7 billion, we haven't even come close to being able to cover the planet entirely. What's even more interesting, and again, we've already seen some of these amazing talks, so I'm just going to highlight it. They can be found, these microbes, in some of the most incredibly extreme environments. And Ben, much like you were saying, it's kind of funny that, the microbes that live here probably look at us and say, holy crap, you guys are extreme. How the heck can you live in these conditions? And it's absolutely fascinating that we can find them in areas where, for the most part, we would think happen to be completely uninhabitable. And then, of course, we do have those pathogens like I was talking about. And I mean, I've got a couple of them listed here. We've got the noroviruses, the little blue dots. We've got Giardia, which looks really cool as a tattoo. I've got a few friends who have that as a tattoo. We have the nematodes that can do nasty things. And of course, we got the bacterial species. Up here, we've got in the middle Listeria. And of course, we've got some of the gram-negatives that are sharing their pili, and we'll get into that in a minute. So how do we get from a place where we essentially started off as being at war and get to a place where we feel like we're at peace? And in my research, what I found was that the best place happens to be a book that was written many, many, many years ago, Sun Tzu's The Art of War. And honestly, it's a very small book. I would really recommend that you pick it up, you read it, and you get a good feel. But when it comes to microbes, I've already done some of the work for you. So you want to know your potential enemies. You want to know what your weaknesses are. And I've already mentioned a few of them here. I mean, we only multiply about once every 20 years and compared to 20 minutes. We don't evolve anywhere near as fast as microbes in that case. we essentially are very large organisms that require certain types of conditions to stay alive. They can pretty much go anywhere, and they'll evolve to do that. You want to know what your weapons are. And over time, we've actually figured out that we do have weapons against microbes, but the ones that we have been using may not necessarily be the best ones. You want to always act in a concerted way, and always, that's always a good thing. I don't really have to go into that. But one that I really do want to bring up happens to be this idea of beware of lengthy campaigns. And this is one of those things where you start realizing, even if we started the war on microbes back in 1912, which is one of the first times I've ever seen the term used, we haven't really gone very far. In fact, anybody who may have actually believed that we have had a mission accomplished against microbes, especially pathogens, has been proven obviously wrong. So the question then becomes, what do we need to know in order for us to be able to find that harmony as opposed to the war? And even if we do happen to be having a war against those small number of pathogens, 450, what are we going to do? Well, the first thing we need to realize is that microbes can only get into us in a number of ways. We call these the portals of entry. And it's the 5 I's: the inhalation, the ingestion, the ingress, the injury, intimacy, intercourse. That's really the only way that you can really get into a human body these days. And the fact of the matter is that if a microbe can get in there, well, what can end up happening is that you get a shift. And essentially, that shift can be what would normally be a nice, diverse environment comprised of microbes in some cases, maybe your cells, and eventually it gets to a point where you see that one entity coming in. We're just going to highlight him here. And then over time, it's essentially going to develop to this. That's very dangerous. And one thing people have to understand is that no matter what you're talking about when it comes to commensalism, mutualism, and parasitism, if any kind of bacterial entity and even viral entity happens to get into your bloodstream, there's always a risk of this happening. They're not thinking per se. All of this is based on thermodynamics. All of this is based on the way that molecules interact with one another. And if you give microbes the opportunity to grow, they're going to grow. And if that comes at your expense, well, then that's really up to you. And I think one of the best examples of that happens to be one where you've seen people do surgeries in order to try and improve people's lives, but inadvertently, what ends up happening is that a microbe actually gets in there. And sometimes it doesn't even have to be a pathogen. It can be something as simple as a bacterium that's always on your skin called Staphylococcus epidermidis, and it can cause significant problems. So at the end of the day, what we have to realize is that we want to be sure that it's not getting inside of us. And if it's not getting inside of us, we're safe. But here's the problem. If we happen to have something that is pathogenic and has the capability of being able to spread, we have to figure out how it spreads and how fast it can spread. And we have a term for that, and it's called the R0, or the basic reproductive number. And essentially what this is telling us, despite all the math that you see here, is that we can be susceptible to an infection or some kind of colonization. We may be able to be recovered. In other words, we've seen it, we've moved on. Or we could be infected. And that means that we're currently in the process of dealing with this particular colonization. Now, again, we talk about this in the terms of pathogens, but this actually can happen in terms of a number of other possible entities. And one that happens to be really important in this particular case is a bacterium that we tend to see all the time, no matter where we go. It's called Pseudomonas aeruginosa. Now, the fact of the matter is that Pseudomonas really doesn't do a heck of a lot to most of us. But if it gets in your wounds, it can cause a major problem. And for those people who suffer from something like a cystic fibrosis, it can get into their lungs. So even if we're talking about regular average bacteria that normally are not considered to be pathogenic, it can lead to a problem of transmission, and the basic reproductive number takes over. And what we see here is just how complicated it is when it comes to the idea of an R0. It's not something for you to read, not something for you to understand. We're not doing a final exam at the end of this, but it gives you an idea that this is not something that you do easily. And another thing that I also want to point out is that during the COVID crisis that we've been going through, you probably heard numerous months ago this argument over the reproductive number, the R naught. What is it? Is it 2.5? Is it 3.5? Is it 15? You know what? At the end of the day, it's not really that big of a deal. And I'm going to show you why. First off, though, I'm just going to show you a few of the R naughts for some of the diseases that we have out there. You'll actually notice that when it comes to measles, which can be airborne, it's somewhere between 12 and 18. For something like HIV/AIDS, which of course is sexual contact, it's about 2 to 5. But you know, we can do the R naught for practically anything. And a friend of mine out of the University of Ottawa, Robert Smith, ? You got to Google him, he's one of the best guys out there. He actually did something very similar, but it was for how fast Justin Bieber could transmit in terms of that fever that kids were having from person to person to person. What they found was that the actual R naught for that was 24. So the whole idea of a meme, something going viral, et cetera, actually does have scientific validity, and in some cases may actually be far larger than any of the transmissible organisms that we talk about. Now, when we talk about COVID-19, all right, we have to sort of take a look at that R naught with respect to others. So as we saw, you know, Ebola 1.6 to 2.0, COVID-19 2 to 2.5, MERS 2.5 to 7.2, okay? Here's the thing. The reason I say don't worry about that is because this is all situational based. Because when you start looking at Ebola, you actually realize that that 1.5 to 2.5 only exists if people are happening to be alive and interacting with one another. What we found in West Africa was that after death, that R naught went up to 12 to 13 because the virus didn't die, unfortunately, when someone did. And so without the proper burial protocols, and thankfully the WHO put those out there, we saw lots and lots of people getting infected. So again, when we start talking about being able to prevent these pathogens from getting into us, and not just the pathogens, we're talking about all those different types of organisms that potentially could cause a problem, you have to realize something. All the numbers in the world, the R naughts and all of the epidemiology that we're seeing, only relates to one particular factor at any given time. But if you want to start looking at a way of being able to forget about the threat and start looking at how you can have that relationship with them, then you need to start looking at behavior change. And one of the most important things that you need to realize is that we have to go into some dark areas of human behavior in order for us to be able to understand how we can start getting a better relationship with microbes. And another question that I tend to ask people happens to be this: what aspect of lifestyle has changed the most? In other words, how much has your life today changed from life 2, 3, 4 generations ago because of a bad germ or some kind of germ that has led to a problem, all right? And you might think eating habits. A lot of people tell me travel because that's the one thing that really has changed a lot. Mass gatherings, Olympics, going to the Hajj. But at the end of the day, the actual answer happens to be intimacy. And this is one of the things, a lot of people refuse to get into the idea of intimacy. However, if you actually look at the way that germs have affected or changed the way that we get close to one another, you actually notice that, thanks to syphilis, we got modest in the Middle Ages. Gonorrhea led to the criminalization of prostitution. Herpes changed the way that we looked at promiscuity. I mean, the whole idea, and I'm in North America, so I was born a little bit too late for the love generation. But when herpes came around, all of a sudden the love generation kind of dropped off. HIV, no glove, no love. I mean, the whole idea of using barrier protection became a norm. And then, of course, the human papillomavirus, which has what we call the Michael Douglas effect, because he came out and said something along the lines of intimate behavior actually led to him having a throat cancer based on a human papillomavirus. Of course, that was completely bogus. It was not true, but it did actually have an effect on how people started looking at different ways of intimacy. So now that we've set the stage, we've got all these things put together, we understand what it is that we need to be looking out for in terms of that relationship. Now we have to face the big challenge. And no matter what you're watching or reading, the rom-coms, the novels, that type of thing, Act 2 is always when everything gets blown out of proportion. And when it comes to our relationship with microbes, I like to call it the pandemic state of mind. Now, we are currently in the midst of COVID. So most people would actually think that the state of mind of panic with respect to COVID is probably worthwhile. The thing is that when you start looking at our history, COVID is just one aspect of what we've gone through. And if you were to look at something a little bit more prominent in our history in order to understand how we've sort of misinterpreted microbes, you kind of need to go to influenza, which is what you have here, and what I call the master of the germ code. And what this is doing is it's showing us that influenza is a very, very preventable illness. However, it always tends to come back, and usually it comes back every year, but every now and then we end up getting these pandemics. The most recent happened to be back in 2009. And one of the very interesting things about a pandemic is that it differs from an epidemic in the way that our social behavior takes us. Now, most of the time when you actually see a regular influenza virus and look at the dashed line here, it really doesn't affect the majority of people. It's only those who happen to be very young and very old. But when you get into a pandemic, what ends up happening is that those middle ages from around 15 to 50, and when it comes to COVID, we're now looking at around 19 to 50, you start to see a rise in the number of cases and also a rise in the number of severe cases and possibly deaths. Now, H1N1, the cause of the Spanish flu and even the flu from 2009, it was causing a lot more damage and death in these age groups than COVID is at the moment. And so when you hear people saying, well, it's no worse than the flu, or it's no bad, it's not as bad as the flu, it does have some perspective to it. However, we are in the midst of a pandemic of a virus we've never seen before. So that makes it much more troublesome than the flu ever would be, because we just simply don't have that lasting partial or heterotypic immunity. So it's an argument that a lot of people are having. What people are not arguing about, though, is this. Travel actually does have the opportunity to be able to change not the way we live, but the way that we interact with microbes. And when you actually start looking at the number of international tourist arrivals, you start to realize that, oh my goodness, we had to somehow find a way to be able to stop the traveling before we ended up with something that could lead to a pandemic such as COVID. Unfortunately, that really didn't happen. And so what could have happened, which was on January 17th, when it was pretty much known in Wuhan that there could have been that human transmissibility, they probably could have started the lockdown then. They didn't. They waited another 4 days. And with air travel, that was enough. And that's how it managed to spread across the world, as we've seen here. But is that unique? No, because we saw the same thing with HIV. And I'm not talking about HIV with patient 0 or patient O, if you want, and that spreading. This was spreading around long beforehand. It's just it was unrecognized. It was just simply under the radar. And another one is the prevalence of measles. And this is very important, especially in North America, where we have a huge majority of the population who already happen to be vaccinated. So they have protective immunity. But then you get somebody who doesn't have that protective immunity. They've never been vaccinated. They go to one of the hot spots, as you can probably see here, and then they travel back through air, and then they bring it with them, and other people end up getting infected. Normally, it doesn't really get much attention, but then a couple of years ago, it ended up in one of the Disneylands. It also ended up in state fairs, and all of a sudden, you start to really see how important that travel ends up becoming. So we've got intimacy, as I mentioned. We have travel. And now we have another factor, and that's brought on by this, which sort of is the epitome. Now, these are not loaves of bread. This is a colored electron microscopic image of C. difficile. Now, we used to call it Clostridium difficile. It's now been called Clostridioides, and I'm pronouncing that horribly because I haven't memorized it yet, but C. difficile will work for now. And this actually is a major, major, major problem when it comes to infections that essentially cannot be treated. Now, normally Clostridium difficile would not have been a problem. However, it has developed what is known as antibiotic resistance. And if you're wondering how that happens, and the second part of the slide here will actually show you. Essentially, when we're treating different types of organisms, we use antibiotics. But if we don't have a strong enough concentration, then what may end up happening is that the Clostridium or C. difficile is going to develop an evolution that's going to lead to the inability of that antibiotic to have an effect. Now, this isn't something that's only with C. difficile. This actually happens with a large number of different types of organisms. And there's several different mechanisms that we now know of. So we could have efflux systems, inhibition systems. We could essentially inactivate whatever the drug happens to be. All of these are now being seen. And another thing that's happening is that they're spreading from one particular bacterium to another. Now, when this is in a very small environment, yeah, okay, that happens, and very little we can do about that. But what we can do is we can actually stop the macro spread of antibiotics by essentially reducing their use in all sorts of different environments. Now, when it comes to humans, well, we can reduce the amount of antibiotics that are used when we're treating. When it comes to animals, well, we can use either drugs or treatments that have absolutely nothing to do with antibiotics. We can go natural if you want, or we can simply use drugs that are simply not seen or found within the human population, which is one of the directions that a lot of places are taking at the moment. Now, when you look at it from that perspective, now all of a sudden you're realizing that we're also going back to what we were seeing before with all sorts of different types of intimacies. The difference, of course, is that now we've gone from person-to-person intimacy inside of a bedroom to intimacy between people who we care about, in other words, close contacts. And now that leads us to a point where we start to realize that in everything that we do that happens to be close to another individual, we're risking the chance of having some kind of spread. And if we're not careful, then those types of bacteria and viruses, whether they happen to be, parasitic, whether they happen to be commensal, opportunistic, or quite simply, they just happen to be bacteria we're not particularly used to, can actually spread from one individual to another. So what's the answer in this case? Obviously, it seems like the answer would be you have to just simply stay away from one another, lock down and isolate yourselves. Of course, we've already tried that. And I mean, how many of us are suffering from COVID fatigue, right? It's not particularly the best option. So what is the best way to be able to deal with this climax in the issue between us microbes and that relationship? And that takes us into Act 3, where we have to sort of find the love in all the right places. Now, before I get into some of the things that show that microbes really are good for us, the first thing I actually do want to tell you is that when it comes to the microbiome, which a lot of you probably want to be talking about, that's going to be in my next talk in about 2 hours. So we'll see you in actually almost 2 hours exactly from now. As for what we can be doing with microbes to actually help us, we can use them to develop medications or medicines. And if you actually look at Banting and Best, they actually created human insulin, and then eventually it became something that could be done through the use of bacteria to be able to mass produce it. So in that sense, we can actually use bacteria to be able to help us develop medicines. We can also use bacteria to be able to develop products that can be good for our health, but also possibly good for the environment. And in this particular case, astaxanthin, which is a really, really good supplement, can be made in algae. What's really funny, and this was actually featured in a show called Dirty Jobs many, many years ago, was in order to get from that green to that red, you just simply had to make them angry. And the way you did that was you actually put them under sunlight. It's a fascinating thing, and I would definitely recommend that you learn more about that. We can use responsible genetic engineering, RGE. And what this means is that we can start developing ways to be able to change our world, our environment, so that we can make things better. And so let's just say that people are deficient in a certain type of vitamin, or maybe even omega-3 fatty acids. And we know that those can actually lead to chronic disease problems. We can actually use the bacteria to be able to help them, help plants develop these particular types of supplements. And when that happens, we can then start providing that to people in a way that will increase their nutritional I guess, safety, if you will, and prevent the likelihood of chronic diseases, both caused by bacteria as well and viruses, as well as chronic ones, NCDs, non-communicable diseases. The other thing is that germs actually do help the environment. So the more that we have them around, especially the ones that produce oxygen, the better off we're going to be. I know that we talk about trees and they're incredibly useful and necessary, but at the end of the day, algae and photoplankton are really the ones that are making up about 70% of the oxygen production on Earth. And something that I had brought up a number of years ago was this idea of being able to develop these almost monolithic algae and phytoplankton towers in the middle of the ocean, where they can just essentially be oxygen pumpers. And this is something that has been looked at and is currently in development. Finally, looking at how we can use bacteria to be able to, or even yeast, and to be able to make ethanol. And this is very interesting because we're now at a situation where we are starting to look at how we can do a better job of being able to make these biofuels, because that can actually take us out of the oil and gas industry. But at the same time, we have to do it in such a way that it balances out with our need for certain types of agricultural products, which would be the initial yield or substrate for development of the ethanol. We're getting there. For an example, the idea of using hops. We use it to make beer, but what we can do is take whatever's not used from the hops and put that in to make cellulosic ethanol. So there are ways that we can approach this and actually improve our world. So at the end of the day, what I want you to understand is that our relationship with microbes is pretty good. I don't want you to panic. But what I do want you to understand is that if you are panicking, or if you are feeling like in a panic situation, it's okay. Because at the end of the day, as we go through that 3-act process, all right, we start to realize that it's going to take us some time to get from that despondency and depression at the end of Act 2, which is really where we are thanks to COVID, and start getting back into hope and relief and optimism. And the idea behind that isn't necessarily having a vaccine or making sure that you wash your hands 30 times a day. These are all going to be great in order to be able to help reduce the transmission. But at the same time, it's not going to give you the sense of confidence that we're going to be able to move forward in a way that is safe. We need that behavior change. We need to start looking at our relationship with microbes in a way that we are getting away from the bad ones and we are welcoming in the good ones. And that, you know, is sort of basically what trust is all about. So when you start talking about the germ code and our relationship with microbes, the first rule is the same rule that you see pretty much in all types of ecology, and that is eat and be eaten. You know, microbes live by the pili and die by the pili, and if you happen to be microbiology, you know what that means, and yay. Otherwise, if you don't know what that means, essentially, it's all about being around others. And then we evolve and adapt, which is really important. And then we have all good things must come from your end. And then finally, germs are everywhere and get used to it, all right? And at the end of the day, even though you have to sort of take all of these into consideration, the one thing that you have to realize as we're moving forward is that the answer to the germ code, if I can get my screen back, there, is to evolve and adapt. And if we adapt together, we're going to be able to have that relationship and find that love that we need. And I will thank you at the moment because we're now moving into the next segment. But I do want to say, if you're not going to come back for my second talk, season 2 of my show, The Super Awesome Science Show, starts September 28th. And you can listen to season 1 right now on Apple Podcasts, Spotify, Google Podcasts, or wherever you get your streaming audio. Thank you.
Awesome. Thanks a lot, Jason. Now, I'm glad we got you coming back again for a second talk. So that's been fantastic.
Excellent.
Right. Let me just double check. And yeah, we'll see you there again then in a couple of hours for your next talk on journey through microbial world.
Excellent.
Can't wait.
Looking forward to it. Take care.
Yeah, you too.
Hello.
Hi, Elizabeth. Sorry for a couple minutes behind.
No worries. No worries.
Mostly on time. And yeah, I think we will basically just hand over to you. So you're here to give us a talk on research integrity, which is a very important topic, not just for microbiologists, I guess for all scientists, but certainly for microbiologists. And while we celebrate microbiology today and microorganisms, we must make sure that we're studying them in a way that actually gives us real knowledge, right? So I'm going to let you begin, and I'll get out your way. Just make sure you share your screen, and I think it will all be fairly straightforward.
All right, let me get that going. And let's see, I find my right screen. All right, there we go. Maybe, can you give me a heads up if you see the right screen that looks like a presentation? It's still loading. I might be stopping my video to give myself a little bit more bandwidth.
Okay.
Wait, it's in the way. Let's see, present. There we go. Is that loading for you?
It is loading, and I can see the first
slide. Awesome. Okay. No, so I'll let myself stay on, and so you guys can see my face. But give me a heads up if there's a little bit of connection issue, because I'm presenting from my parents' home in the Netherlands, so my connection is a little bit slower than I'm used to. So just making sure that you guys can see and hear me fine. All right. So let me start by saying that I sort of have this dual personality in science. I am from training, I'm a microbiologist. I did my PhD in the Netherlands, which is where I'm presenting from now. And I worked 15 years in academia at Stanford, and worked 2 years in industry. And it always had to do with microorganisms or the microbiome. And so I started a blog called Microbiome Digest, which is still running, not now by a team of volunteers. And it's just a little plug here. We have at that website, we have a daily compilation of papers that came out in the microbiome field. So it's everything about microbial communities, not only in humans, but also in plants, in animals, in the environment, and sometimes some COVID papers or some microbiology in art. So we try to make it interesting for a lot of people with a lot of different backgrounds, but it's all about the microbiome. And it's basically a list of daily papers, but we sort of sort them into categories. So if you're interested in plant microbiology, you can find your favorite papers there. And we try to post things that are less than a week old. So we try to be up to date. But I've also became interested in science integrity. This started sort of as a strange hobby. I started reading about science integrity and specifically misconduct. And as anybody who likes a good detective story, I got hooked because some of these stories are very juicy. They're about fraud. They're about lying and cheating. And, that's very interesting. But I also became interested in the sadness about these cases and just in sort of finding these cases. I started by searching for plagiarism, And I gradually became more interested in finding duplicated images, as you see a couple here on this starter slide. But also it got me to think in broader terms about why do people do misconduct, and what, how harmful is it for science? And I got more and more passionate about it. So at some point I just decided to quit my job and do this full time. And so it's an unpaid job. I do some occasional consultancy, but most of the work that I do online, you might know me from Twitter. I'm at Microbiome Digest without the E. And the work I present there and on my blog Science Integrity Digest is most of that work is unpaid. But I feel now that I have the freedom of not being employed, that I can do a lot more than when I was employed. And I hope to make a difference for science, even though I'm not employed by a university or a company. So that's a sort of an introduction on the work I'm doing. And if you think about why science misconduct is important, for me, the central sort of theme that I see is that scientific publications are the way that we're measured. That's sort of our output. We see science papers as sort of a measurement for our success. And that is true when whether you're a graduate student or a postdoc or a professor, you will always be held accountable for the numbers of papers that you publish. Maybe you have to publish roughly a paper a year, or maybe your group has to publish 10 papers a year. Usually you'll feel the pressure that you have to publish an X amount of papers per time unit. And if you don't do that, if you don't publish papers, that means you might not get a letter of recommendation, you might not get an appointment as a professor, you might not move on in your career, you might not take that next step. And so all of us in at least in academia will feel this pressure to publish. So it's called publish or perish. That's the motto. If you follow the university motto, you'll do fine, but else. There's always a threat that you might lose your job or not get that promotion if you don't publish. And so even though all of us feel this pressure to publish, not all of us will move into the dark side of science, into doing misconduct, into faking results. Most of us can handle this pressure, and even though we have to put in crazy amount of hours, which is not good, but we do that because we're passionate about science. So we can mostly handle that without resorting to doing misconduct, but some people choose to cheat and to fake results in order to get that promotion or that letter of recommendation. So that is sort of the setting the tone. And I, even though I don't obviously approve of misconduct, I do feel a little bit of the pain and the sad stories that are sometimes behind these things. I'll maybe touch a little bit more upon that in the rest of my talk. But I very much realize, even though the work I'm doing might seem fun and juicy and very interesting and all those good vibrations, there's very sad stories about each of these, behind each of these cases. There's people who wanted to thought they had a great idea and were convinced that they were going to get a beautiful paper, and then they were scooped. Or there's people who were had some very promising results and had a very nice position. They became professor, and now suddenly the experiments don't work anymore. Or they might work in a lab where the professor is maybe a bully and really asks that of them that they publish, and or else they will lose their job, and the PI will hire another postdoc. And so people are sometimes in very desperate situations, and that is the motivation why they do science misconduct. And again, I'm not approving science misconduct at all, but I'm sort of sympathetic in a sort of sense of the motivations that are behind these cases. But I will not stop and I will not look the other way. I will keep on finding these papers and flagging them on websites or reporting them to journals. So as I said before, why am I so motivated to do that? Because I do see publications as the building block of science. When we are working in science, when we are thinking about our next project, or when we are writing a paper, we'll do a literature search and we'll look at papers and we will see what other people have published before us. We base our research on these previous publications. And if you think of science sort of as a hundreds of years old continuation of knowledge and building upon the previous generation, you can think of science publications as a big wall built of bricks, in which each brick is a publication, and every generation of scientists lays these foundations of bricks, and then the next generation lays another foundations of it. And if you sort of pictured that in front of you, then you can also imagine that if one of those bricks, one of those publications contains falsified or fake data that is not true, then the next people who are building their work upon that publication might actually not be able to replicate it. In other words, if one brick is not really strong, the layers of the wall on top of that might actually also tumble down. And so the reason I'm passionate about science publications and science misconduct is because I realize how much time people can waste trying to replicate results that are fake. And science should be about finding truth, not about finding fake papers. And fake papers sort of pollute science and hinder science and its progression. So having said that, I know that most scientists are hardworking and honest, and I'm convinced that all of you are in that category. I'm obviously working on finding people who are maybe not honest and might not do hard lab work, but might actually fabricate results. And I don't want you to walk away from my talk and think, oh, all science is flawed and all scientists are dishonest and I cannot trust any paper anymore. That is really not what you should be thinking. I'm just focusing on a very small section of science, which I think is should be cleaned up, or should be flagged, or should be we should make others aware that this is going on. But it's not applicable for all science. But science is also not immune to dishonesty and fraud, like in any other field that you can think of. If you think of banking and construction, or any other type of profession or field, there will be dishonesty and fraud. That's not to say that that everybody in that profession is fraudulent. And similarly, science is not immune for that either. So I've worked on many different types of science fraud. Like I've dealt with predatory journals and fake authors, fake peer reviews. There's conflicts of interest. There's lack of patient consent. And then there's really the dark side, where we're talking about the true definition of science misconduct, which is one of three things: plagiarism, falsification, and fabrication. And this is according to, for example, the definition by the Office of Research Integrity, which in the United States oversees all research that has been funded by the National Institute of Health. So ORI, the Office of Research Integrity, defines science misconduct as one of these three things. But you can also extend science misconduct to the things I've mentioned in the previous bullet points, like conflict of interest, where, for example, a paper gets published in, or a paper gets published on a particular result or a particular drug, but the writers, the authors of that paper are working at that company. And of course, they will tell you that their product is great. And if then the peer reviewer is also working at that company, you can see there's a clear conflict of interest and you should not trust that paper, or at least that it's of high concern. And so there's all kinds of things, and where they technically don't necessarily fall under the definition of science misconduct as defined by ORI, but I will also work on some of those cases. So plagiarism, obviously you are probably familiar with that. That's copy pasting somebody else's text, but it can also be including copy pasting somebody else's ideas, which are much harder to prove than textual similarities. Falsification is where somebody does an experiment, but changes the results a little bit, or maybe leaves out an outlier, like you can see on the cartoon on the bottom left, where, well, fabrication is completely making up results. So a way of looking at it is where falsification is an experiment happened, but you change the results. And fabrication is no experiment happened, you just made up the results. So that's sort of a working definition. And sometimes these two are not easy to distinguish, because you can do one experiment and also pretend it's a second experiment, which sort of seems to fall in between these two definitions. So science misconduct is not a black and white distinction, because basically science misconduct, research misconduct is sort of this slippery slope. And I've put some things here on this slippery slope, sort of roughly going from not so bad to very bad on the right. And that's not to say that one is, I just put them sort of in a particular order. But these are some things you can think of, and you might think, that's not too bad. That's okay, that's acceptable. But then the next step might be a little bit, seems to be less scary to then move a little bit farther. And before you know it, if you don't talk to yourself and sort of slap you back up on the ramp, you might actually end up at the bottom, because it becomes easier to do fraud. Every time you can push the boundary a little bit farther. And I want to make everybody aware that most of us might have done a couple of things at the top and then move back to the sort of the zone of honesty. But it becomes sometimes easier to move farther to the right. And just as some examples, you can think of changing contrast, or I've done an experiment where I did I made a calibration curve for some dilution, maybe some PCR, quantitative PCR, for example. And then you make this beautiful calibration curve, and there's this one outlier that just doesn't fit. And I don't know what I did wrong. Maybe I made it wrong dilution or so. So I redid my calibration curve, and then it looked fine. That's not misconduct, obviously, but it's sort of this starting the slippery slope of like, I didn't like the experiment. Let's redo it. But I did write it down that I redid it and why. And I just was not sure if I had made the right dilution. Maybe I was just not paying attention. And one of the dilutions was just completely off. And so I redid it, and then it was fine. So I for myself, I sort of justify this because I wrote it down. I didn't hide it. I didn't try to make it go away. I was very honest about it. But it did feel a little bit bad, and I was worrying I was going down the slippery slope. And so here I am working on research integrity. And I think all of us are in this position where we do little things that are, you hide some bad experiments. And I think as long as we realize that this is bad and we don't hide it completely, we take notes in our lab books, and we maybe discuss it with somebody else who is more experienced, then it's okay. But you can sort of see where it might actually also become easier to then move to the next level. So I just want to point that out, that there's a gray zone sometimes where all of us sometimes go into, but as long as we go back into the lines, then it's okay. But you can think about things like where you have a blot and you cover up an aspecific band by a patch of gray. That's maybe not a good thing. Or where you don't want to do a control and you just take the photo or the results from a previous experiment, or you change the value to make it a little bit higher or lower so that it either falls below the cutoff or above the cutoff, and your experiment now becomes statistically significant, for example. Or to the really, really bad things where people have a microscopy photo and just copy paste a couple of cells or bands to make it look like there's more cells or there's more bands, or the bands are at a different height, for example. So that is clearly not good, because that's photoshopping and that is making up data. And then the complete extreme is where you completely make up data. You don't even do the experiment. You just make a graph and say that that is what you measured, but it never happened. So this is the slippery slope of science misconduct. And so I assume that all of you have maybe ventured a little bit on the top left, but hopefully none of you have gone all the way down at the bottom of the slope. But there have been several cases where people have been doing misconduct and have been caught by doing, and that ended up in retractions of their papers. And here are 4 cases that have been in the news a lot. And this is to show you, this is not specifically microbiology, although the first one has some links to that, but just some 4 famous examples that some of you might have heard about or read about. And they are showing that this happens to people all over the world in all kinds of fields. So the 4 examples that I chose are from, the first one is from medicine, where in the UK, Andrew Wakefield wrote a Lancet paper in 1998, in which he claimed that he had 12 patients, 12 young children who received the measles, mumps, rubella vaccine, MMR vaccine. And after receiving the vaccine, they started to become, to have symptoms of autism, as well as some gastric or intestinal complaints. And other people somehow knew that this data was not completely true, that some of these children already had autism-like symptoms before they received the vaccine, or some of the pathology reports of these children had been changed. And so it appeared that Andrew Wakefield or some of his other authors have changed results, so have falsified some of the results. And this paper, after back and forth discussion for 12 years, it got retracted in a couple of years later, 12 years later. And even though it got retracted, and even though many other papers have come out later that showed that there was no correlation between the MMR vaccine and autism, people still believe that vaccines cause autism. And that's based on this paper that got retracted. It's based, in my opinion, on misconduct, and it has far-reaching results. And children have gotten measles and they had not been vaccinated, and maybe that's a result of this paper. And so this is one of the examples that keeps me motivated to find papers like these. And but I realize very much that even though papers get retracted, they still might have far-reaching results. There have been other cases as well. In physics, there was the case of Jan Hendrik Schön, who worked in the US at Bell Labs in the field of semiconductors. And he fabricated results. So he made up data, and he made some mistakes. He published the same graph twice in 2 different papers, and somebody just noticed that. And after a long discussion with his bosses and people at Bell Labs, an investigation was started, and it was found out that this person never did those experiments. And so they retracted several of his papers, and by now he has 32 papers that are retracted. And in Japan, there was the case of Haruka Obakara, and she worked at the Riken Institute, and she claimed that she could make differentiated cells pluripotent again by just dipping them in an acid bath. And that sounded too good to be true, and it was indeed too good to be true, because people noticed that some of her figures had been previously used by her in a lab meeting or some presentation at a conference to represent different experiments. So they recognized the photos and they said, oh, but she used those photos before, and then it was a different cell line. And so her 2 papers in Nature got retracted. This actually happened already, I believe, after 6 months or so, pretty fast. And then she was offered to repeat her experiments by working under supervision for about 9 months, but she was never able to replicate her results when somebody was watching what she was doing. And then in the Netherlands, where I'm presenting from, there's the case of Diederik Stapel, who worked in psychology. He was a professor at Tilburg University, and he made up most of his data. He actually handed his data sets over to graduate students and says, oh, just write a paper. But his graduate students and other people in his lab became suspicious because they never saw their professor go and do research. He had all these beautiful results. He claimed, for example, that he interviewed several school classes, but they never actually were allowed to go with him to do the interviews. And they didn't believe really that the results were true. And so they became suspicious. They reported him. And in the end, he actually admitted that he made up all those studies. And he's in the leaderboard of Retraction Watch. If you like stories like that, I would recommend that you follow retractionwatch.com. They have stories behind retracted scientific papers, and they're very interesting to read. So he is in the leaderboard of that website with 58 retractions. So I was not involved in any of these cases, but I'm sort of doing similar work. I specialize in finding duplicated images. So it would be similar to the Haruko Obokata case, because that is sort of what people found out about her work and how they discovered that the work was falsified, or at least parts of it. And so the work I'm doing is looking at photographic images or figures in biomedical papers. And the reason that I'm doing this is because apparently I think everybody has some talent to seeing that all these images are different from each other. You might see microscopy photos that look sort of similar at first glance. They're black with a green fluorescent cells visible, but all these panels are different. There's photos like these from Western blots where you see dark black bands on a lighter gray background. But you can see that each of these bands has a different shape. There's different shapes, there's different curvatures, different gray values, different thickness of the bands. There's dots and spots and things like that. So you can tell that each of these panels look different from each other. And then there's microscopy, photos of tissues. There's more tissues here that have been stained with monoclonals for certain proteins, or there's other fluorescent photos. And some of these photos might look quite similar, but there's differences. And in this case, it's actually a time series, so they're supposed to look very similar to each other. But the human eye-brain combination is pretty awesome because we can see that all these photos are different, and that's fine. That's what we expect, because in biology, photos and tissues and cells and anything we make photos of is unique. Like faces, like every rock is different, every leaf is different. We don't expect things in nature to be exactly the same. So we can see when they are. That is a very unexpected finding. Here are some of the examples of image duplication that I have found. Here on the top left, you see a bunch of panels these are bacteria that were streaked on a Petri dish, and they're photos. And so you can see whether or not they grow. Some grow very thick, some do not grow very well. And even though these are sort of similar photos, they're expected to all look different because they're all different bacterial streaks. But I found two pairs here, marked by me in red boxes and blue boxes here. These two sets of panels, these four photos look identical to their paired other photo. So I will usually say these are more similar than expected, and that is sort of code for these look identical to me. And yeah, we don't expect that. And you know that these are what I call simple or type 1 duplications. It's the same photo that is used twice. Then here I have two examples of what I call type 2 duplications, in which we see an overlap between photos. For example, here, the two areas marked in green or in blue. These are 4 photos of cells treated with different amounts of radioactivity. And we expect these to be 4 independent experiments, and yet we see overlapping parts of the photos. And here we see something similar, where we see 2 overlapping Western blots that differ in 2 lanes, shifted 2 lanes to the right, but they represent 2 different cellular locations and 2 different proteins. So we would not expect these Western blots to look the same. And yet these two panels marked by me in red are identical to each other as far as I can tell. So these two duplications are not the same as the one I showed previously. They're not the exact same photo. It is the same photo, but it's shifted or rotated or moved a little bit or zoomed in, out, or a little bit like that. And then type 3 duplication is shown here. And these are photos of Western blots again. And you can see that some of the lanes look identical to each other. These two are marked by me in blue. And if you look carefully, you can see all kinds of little spots that are, yeah, those lanes are not expected to look the same. These are photos of a whole blot, and yet they look identical to each other. And here in the bottom row, row D, you can see 3 lanes that look identical to each other. And it appears that this photo has been manipulated. So I don't know why, but these 3 lanes are visible, or this particular lane is visible 3 times, while in reality, that cannot be, that cannot have happened. I cannot think of any other explanation that this has been photoshopped. You can also see a splice here, but that's not even the worst problem. Yeah, so this is what I call a type 3 duplication. And if you, again, recapitulate everything, this is type 1, type 2, and type 3. You can imagine that type 1 is most likely to be an honest error. Type 2 is somewhere in between, and type 3 is most likely to be done intentionally. Because you can think, if you don't label your photos really well, maybe you by accident grabbed the same photo twice. Here, I'm not sure if there's any reason, any accident that could explain this, other than human manipulation after the photo has been taken. So sort of this was a useful distinction, because I turned my searches, which I did sort of in my spare time, I turned this into a scientific project. So in my spare time, after working in the daytime on my research, at night I would search biomedical papers for duplications. So it became a very strange hobby, and I did this for many years. And in the end, I had scanned 20,000 papers because when I do things, I do them sometimes a little bit too intense. And I scanned them in 40 different journals for 14 different publishers. And those papers were published in a period of 20 years. Up with Arturo Casadevall and Ferric Feng, who are both famous microbiologists, and they were in my field, and they had together already published a lot on things like retractions and falsifications, but they had not really heard about image duplications. And at first they didn't quite believe me that I was finding these things, but then I showed them some examples and they became very interested, and they became great mentors, and they helped me a lot, turning my strange hobby into a scientific paper. And so in the end, I found in these 20,000 papers that I scanned, I found 800 papers with duplicated figures. I have to say that these figures were duplicated within the same paper. So it could be, like I showed you in the previous slide, within the same figure, but it could also be that figure 1A looks very much like figure 3B, for example. So the duplications had to be within the paper, and they also had to be inappropriate, as you can see in the title, which means that if a photo represented the same experiment, then that was okay, that's appropriate. But these photos were representing different experiments. So that's not okay, that's inappropriate. So those were sort of the rules. And the 800 papers in 20,000, that's 4%. So 4% of these papers contain these types of photos. And another rule that I applied is that the paper had to have at least one photo. And if a paper only had line graphs or tables or things like that, it didn't count. So all these papers had at least one photo. And because we had these 3 types of duplications defined, simple, type 1, repositioned, type 2, and altered, type 3, that was very helpful to sort of answer the question that how many of these papers, how many of these duplications were done intentionally. So basically the old question that there's no good answer to is, what is the percentage of misconduct in biomedical papers? And then many people have made some guesses, and of course we only made a very rough estimate. But because we had these three types, and we know simple, let's say that's an honest error, altered, let's say that's always intentional, and then repositioned is somewhere in the middle, then roughly, because these things were roughly distributed equally over these three categories, roughly half of these papers, or 2% of these papers, might have been done intentionally. Again, this is a super rough estimate because there's many caveats. For example, I scan these things by eye. I probably have missed a lot, and if it's a good Photoshop, I would not find it. So maybe the percentage is higher. On the other hand, you can say, well, these are only the ones with you enriched for papers with photos. So maybe the percentage of duplicated photos is probably, in the end, if you count all papers, it's much lower. But you could also then again argue for the other side and say, well, it's fairly easy to see a duplicated cell in a photo, but how about an altered value in a table or a line graph? How easy is that to spot? And there the answer would be that's almost impossible to spot unless you sit next to the person doing the work. But just by looking at the paper, you would not be able to see a well fabricated data set. If it's a smart fraudster, you cannot catch them very well. I can only catch the really dumb fraudsters, to be honest. Most of the examples, you're like, they're laughable. Once you point it out, it becomes fairly easy to see. So if I had to make a guess what the real percentage of fraudulent papers would be, I might actually end up between 5 and 10%, like much higher, which is very high if you think about the amount of papers that get published every day. You could also argue then that means that 90% at least of the papers is okay. So it depends on how if you consider the glass half full or half empty. And again, this is a rough estimate. Maybe I'm completely wrong. This is it's really hard. You cannot really ask authors, did you do misconduct? Because who is going to answer that honestly? Nobody. So it's hard to make a guess, but my best guess would be between 5 and 10%. So of course, this is a virtual talk, so I cannot really see you guys or have people raise hands or be in a discussion with you guys. But I want you to look at this photo, which was represented in a scientific paper as these are microscopy tissue sections, so very thin sections of tissues from mice that had been treated with different conditions, and they have been stained with different antibodies to bring out different proteins. And that's a very long description of saying that each of these panels should look unique. There should not be any two panels that look like each other. They're from different mice, they're different proteins, they should all look slightly different from each other. And yet there's lots of overlaps. And so I will typically stare at a photo and I'm like, these photos, I see some blue and I see some brown and I see different colors. But I do notice that this row, all these photos look sort of very similar to each other. Would you expect differently treated mice? So different mice, individual mice, they would not really look that similar to each other. And so if you stare at these photos long enough, you might start to see some overlap, and maybe you have already spotted a couple of overlapping panels. But let me show you what I found after spending some time on this photo. I have marked it like this. So boxes of the same color, for example, these two areas marked here in bright green, they look identical to each other. And these are actually supposed to show different antibodies from differently treated mice. They should never look the same. And you can spot these red panels also show a lot of overlap. And then there's this purple panel. If you look at these two brownish shells here, you can see them here and you can see them here. And they just keep on coming back. There's another overlap in this panel that I even didn't see, and I only saw after giving this presentation a couple of times. I noticed that. So there's tons of overlaps here. And even though this is type 2, I think this has reached a point where I would not believe the authors when they would tell me, oh, sorry, we made a mistake. That's too many mistakes. If you make this many mistakes in your science paper, then I'm not going to trust the rest of your administration. Then you just have not really kept track of your photos very well. So this is your daily reminder that you should label and work on your lab books, and make sure that you can, 10 years from now, you can still understand what you did on that day, and make sure to label everything diligently, and try to not make mistakes like this. But this is there are so many overlaps here. I would not believe anything in this paper anymore. That's like beyond, you have ruined my trust at this point. So, well, I've already showed you this, so I'm just gonna show this again. But here the point I'm trying to make is that, so this paper that I already showed you where the overlaps are, this was published in Clinical Microbiology and Infection. And this to me looks, again, like a type 3 duplication, and with almost no chance that this has been done by error. So I reported this to the journal in 2014, and I sent reminders in 2016 and 17. And the author said, okay, thank you, we'll look into it. And they never did. So we're now 6 years later, this paper is still out there. People will still cite these papers. People will still believe this paper is the truth. And I'm frustrated about this, because I reported this paper the official way, and the journal did not seem to care. And that is frustrating, because we put trust in our journals, and we like the editors-in-chief to take action if a person like me, even though I'm just an outsider and maybe a nobody, but like I report this, like this seems so obviously photoshopped that I'm disappointed that the journal did not respond. And this is the case in many of the papers I've reported, not just this one. I've reported those 800 papers that I told you about previously in around 2014 and 15. So 5 years later, 2020, I have a fairly good idea of what those journals did with the 800 papers that I've reported. And only 1/3 of these papers have been corrected or retracted. In a case like this, I think an appropriate action would be retraction. I would not trust anything in this paper anymore. But if it's an honest error, you could give the author the benefit of the doubt and address it with a correction, which is still good for science, because you'll mark the paper will be marked that there was an error in it, and you should look at the figure like this. Sorry, we made a mistake, it can happen. But 2/3 of the papers that I've reported have not been acted upon. They're still out there, they're still uncorrected. And that is sad. And that is why I'm taking things sometimes to social media and I write blogs about that. Because I'm worried about conflicts of interest and inertia in scientific publishing. And it's good that people discuss these things. So here's another example of a paper in infection and immunity that I reported, and it had lots of duplications. And within a figure or a cross figure, there was also duplication here. And this is one of the rare cases where I feel the journal, which was infection and immunity, responded really well. They retracted the paper. And this was actually also reported to the Office of Research Integrity. And it was recently reported that this was indeed misconduct. And the person, the author who published this, has been accused of recklessly falsification and fabrication images, et cetera, not only in this paper, but also in several NIH R01 grants, which is a big deal, lots of money. And this person is now under 4 years of supervision, so they can only do their research when they're supervised by a person. I'm not sure how strict that is. I don't think somebody will really sit next to them, but this is one of the rare cases where I feel this has had an effect, and this there's a punishment. It's not unreasonable. It's not this person wasn't put in jail or anything. They can still do research, but they will need to be supervised. So this feels fair and justified and the right action. So I've worked on many other cases in microbiology as well. This is a case where that has nothing to do with image duplication, but it's one of the cases where I sort of do a post-publication peer review and write a blog post on this. So this was a paper in which the single author claimed that the HPV vaccine basically caused infertility in women. Now that sounds very serious. So HPV is a virus that some of the serotypes of HPV can cause cervical cancer and also anal cancer and all kinds of other, like throat cancer. It's a sexually transmitted disease, and it's devastating. And most women are checked for that every other year, every 4 or 5 or 6 or 8 years, depending on which country you live. Women are checked when they have a pap smear. They're checked for the presence of cancerous cells in their cervix. And the disease is caused by a virus, and there's now a vaccine against it. And it's actually one of the very rare cases in which a cancer can be prevented with a vaccine because it's caused by a virus. And it's all nice and beautiful. And now young boys and girls are vaccinated against this virus with the hope of decreasing the amount of cervical cancer and saving lives. That's all good, but this woman claimed that the vaccine itself caused infertility, which is alarming, obviously. And it was based on this research she did in this group of 800 people who got like a questionnaire, she claimed, or 700 people. She claimed it was 8 million, but it was actually only 700. But here's the thing that nobody noticed this. So she looked at the women who received the HPV and women who did not receive the HPV vaccine. And she looked at their pregnancy rates. But the thing is that she only looked at women aged 25 to 29. It's even in the title. She's not hiding that. But nobody noticed that you know, a lot of women don't get children until a later age. And that is actually correlated with having a college degree or not. So here's a map of the US, which was not in the paper, in which you see the average age of the first pregnancy or birth, I should say, of women without a college degree, which is much lower than if they have a college degree. Basically, if you go to college, you get children at a later age, which is not surprising. But the average age, if you have a college degree, of your first pregnancy or your first child is 30. So if you only look at these young women, the older women are excluded, and you can see that people with a college degree have children at a later age. So, and that again is then confounded with whether or not they received an HPV shot. So it's this very complicated cluster fork in which having a college degree or not, an age, and having the age at which you have your first child, as well as receiving an HPV shot or not, these things are all correlated. And if you leave one of these out, you can draw the wrong conclusions. So it's called a confounding factor. And it seems that she maybe deliberately left out these older women. And of course, then the HPV vaccinated women who on average have a higher amount of college degrees, they didn't have their first babies yet. And so the vaccine doesn't cause infertility. If anything, it's associated with having a college degree or not. But these women did not have their first baby yet. And so I wrote a whole blog post about how this paper should not have passed peer review, and it's now retracted. But there's so many websites that use this paper as a claim that HPV vaccine causes infertility. It's horrible and it's scary, and you can hear I'm mad about this because this is false information, and it will lead to women who will die of cervical cancer, which is now not completely but partially preventable by getting the HPV vaccine. And then people will accuse me of that I must be paid by the HPV vaccine business. I'm not being paid by anything. I wish I was paid, obviously, by somebody, but I'm not being paid. I'm just here to hopefully save some lives and fight against false information like this. So, of course, now COVID-19 happens and brings along a lot of new papers that have been hastily published and hastily peer reviews, and there's all kinds of things for me to work on, and I'll be, I've been as busy as ever, and busier than ever, I should say. And one of the cases I've worked on is a paper, a very famous paper now, by the group of Didier Raoult in France. And Raoult and his group claimed that hydroxychloroquine resulted in a dramatically decrease of the presence of the COVID-19 virus, SARS, the SARS virus, the coronavirus. And if you look at this graph, it looks great. Like, yeah, all these people who are being treated with hydroxychloroquine and azithromycin, they cleared the virus. But there were several things wrong with the paper, and I wrote a blog post about that. One of the things is he left out 2 patients who died in the treatment group. He didn't include them. And so that seems to be, leaving out the results you don't want, which would count as fabrication if you would do an investigation to see if that is really what has happened. And it's a very small group of patients. There are many other things wrong with it. It was peer reviewed in 1 day. It was published in a paper in which one of the authors was the editor-in-chief. And so there were several things that were not good. And this paper, of course, was heavily discussed, but it was also backed up by the US president, the United States president. And he claimed that this hydroxychloroquine was the new saver and would prevent COVID-19. He also claimed he took it himself, etc. And it was just based on a small study. And then there was, on the other side of the equation, there was a paper that proved supposedly that hydroxychloroquine actually did not help at all, but actually caused more disease, and specifically heart, cardiovascular diseases. And that paper was based on a huge data set that then turned out to also be falsified, and the paper got retracted. And I looked into one of these authors, and I found that he produced this beautiful artwork during his PhD. And in this paper, he claims that he's looking at rodents' inner ear structures, which look remarkably like Melba toast. But yeah, they seem to be photoshopped. There seems to be lots of repetitions going on here. And so there's lots of work for me with hydroxychloroquine, and I've worked on both sides of the hydroxychloroquine, the pros and the cons. There seems to be misconduct on both sides and lots of retractions, and that is eroding the trust of the public in science. I'm going to skip this slide because we're going to be close to the end now. And I'll talk briefly about paper mills, which is another project that I've worked on as part of a team of other people who do similar work like I do. So we're a group of whistleblowers, if you will, but some of us, like me, work under their full name, and some others work under pseudonyms for various reasons. So I didn't do this work by myself, but I'm here just to talk about it, but I do want to give credit to those people who worked on this paper mill, like Spot Clyde, Cheshire, Mordy, and then Jennifer Byrne and several other people, Jana Christopher, and Tiger. So it's a mixture of pseudonyms and real names, but I want to give credit to the work they have done. So we found this set of 400 papers that all had the same background. These have, if you compare these panels, it's a Western blot panel, and I've increased the contrast a little bit. If you focus on particular regions, you can see that the background of each of these panels looks remarkably the same, which is hard to explain because it's noise and it should be random. And then if you look at the bands, they sort of seem to float on this background. The bands are all different, but then these backgrounds are the same. How can you explain that? And then we found not only these similarities between panels in the same paper, but we also found them across papers. So there were multiple papers that had this exact same background, even though they came from labs in different places, in different cities, in different provinces. And that became bigger and bigger and more astonishing by every paper that we found out. So now we are, I think we're actually close to 500 papers now that all have the same background. And the only reason you can think of is that these photos are artificially produced by what we call a paper mill, which is some commercial entity, maybe a company, that sells papers to people who need a paper for their career. And there's another paper mill that I worked on where the same photos were used, or same photos, but slightly differently zoomed or rotated. And if you put them on top of each other, it's all the same photo. So that's a set of 120 papers, also from all different laboratories and different groups of authors. So there's no overlap in the authors, or hardly any overlap. And they were probably fabricated by the same company who is selling these papers for money. So that brings me to the last slide. And I want to point out again how much the cost is of science misconduct. It's not only damaging for scientists who do this, and don't only think of the people who do this themselves, but the other authors on these papers, or the other people who work in these labs, who get dragged into these cases of misconduct and whose credibility might also suffer, even though they didn't do misconduct themselves, but their names are on the paper, on that paper, or they have worked in a lab that did this. But it's also damaging for science as a whole, because if people read about these cases, they might start to think that science is broken and that all science is not to be trusted. And I don't want that to be the message of this talk, because I'm fighting against it. But it's easy for a non-scientist to think that if there's misconduct in science, then we cannot trust anything anymore. And that is I think incorrect, but I can see why people think that. And in a situation where we are now with a pandemic, we want people to trust in science, and it's a scary situation because it seems that scientists are less and less believed, and I think that is going to have great and severe consequences for our health and our environment as well. I've touched a little bit about why do people commit science misconduct. This is a question I've already talked a little bit about. There's sad stories about it behind it, and not involving just them, but their whole environment and labs. And one of the examples I gave is that, for example, the atmosphere in a lab could very much determine whether or not the person commits suicide. I keep on saying it wrong, I apologize. Why they commit science misconduct. And that is, it could drive people to do bad things. And the reason I want to bring up that people have taken their lives is that in some of these cases, people who have investigated these cases of misconduct, or who have been accused of misconduct, or who were part of a team or a mentor of somebody who did misconduct, they did take their own lives. And I'm very aware of how sad these stories are. And I want you to think about that, because it's very easy to yell, oh, fraud and fake news. And it's, in a way, it's juicy and funny, but in reality, it's not. These are very, very horrible stories sometimes. And I want to point it out, and I'm trying to always be aware of that. We can also talk about publications and productivity, and maybe we focus too much on scientific publications as the output of what we do. And are there ways where we can measure our output in different ways, that we can not focus so much on how many publications somebody has and in which papers, which impact factors? Can we measure science productivity in a different way? And can we maybe also focus more on negative results? Because one person can spend years of their lives and only having negative results, and that might be worth publishing, but it's hard to publish such results in journals. We can think about which or whose role is it to detect science misconduct? Is it the peer reviewer who might not be trained to find these things? Is it maybe people who work at journals, at staff at the publisher, for example? Those people might be more trained. Is it their role to detect these things? And some journals and publishers have already taken steps to hire people who are specialized in finding these types of photo duplications, for example. And there have been people who are more trained in making the requirements for submitting a manuscript in a better way. And then conflicts of interest are always very important and not to be avoided. And in the end, the question is science broken or not? So like I said already at the beginning of this slide, I firmly believe in science, and I don't want the message of this talk to be that all science is broken. Science has its flaws, but I firmly believe in science. And I hope you're convinced that there's people fighting this fight with me, not just me. There's many people who are concerned about misconduct and who want to fight it and who want to change some of the ways that these cases are handled. And I hope with that we can make a difference to science. So with that, I thank you. And I don't think there's any way of people who can ask questions to me. I'm on Twitter, Microbiome Digest, without an E. And I have several blogs and you can ask me questions. Thank you.
Hi there, Elizabeth. This is Ben. Thank you very much for fantastic talk. I'm just going to look through Twitter and Facebook and see if we have any questions through the live stream. And I also have a lot of questions written down if you have time to discuss them a little bit. Is that okay?
I have time. I don't think there's somebody scheduled after me, but I wasn't quite sure if there was if the gap had been filled. I have a whole hour that I can talk to you.
Okay, great. Because we have until now, we have 15 minutes basically in the schedule for this. So that's great if we can take advantage of your expertise and all the incredibly important work that you've done. Yeah, one thing I wrote down kind of part way through the talk is kind of, what is the damage, do you think, in your position who has been really digging, kind of looking into this, and someone like me who has been involved in science and research and maybe doesn't know the extent to this. And I'm kind of thinking specifically, if I want to go and do a lit review or I want to go and do an experiment, I go and pick a random paper, should I be scrutinizing it in the way that you are, or is it not as bad as that? What is there any way you can kind of say what the damage might be to the system that we have?
So there's no way I would be able to express that in amounts of money. I can only think it must be enormous, because there's so much money that goes on in science, not just in academia, but also in industry. And I've worked in both. There's damage in the amount of grant money and just time and effort and kits that somebody uses and tips of pipette tips, but just the blood, sweat, and tears that all of us spend in the lab trying to replicate somebody else's results. It's hard to measure that, but it must be enormous. That is one cost. And then there's the cost for, if I think about the cost that a paper such as the Wakefield paper, the Lancet paper that got retracted after fabricating results, and people still believing that paper is true, and that vaccines cause autism, the damage that has done, in my opinion, and I realize there's other opinions about this, but in my opinion, that is enormous, because that might have involved lives of children who could have been vaccinated against measles but have lost their lives. And now with the vaccine discussion on HPV, where the amount of people who claim that HPV vaccines are causing people to be paralyzed or sterile, or I've heard all kinds of horrible stories. That could lead in the end to women and also men. Well, not women. So women will get cervical cancer, but men can get from HPV, can get anal cancer or throat cancer, for example. There's all kinds of nasty cancer that you could have prevented with a vaccine. So the price of that is beyond measure. Like, what is the price of a human life? I have no idea, but it's enormous.
Yes. Yeah, it is. It's a very, very difficult question, a very, very difficult subject, but it's clearly important to address. And I think, you talked a little bit about, Trump endorsing that publication. And then we have,
Right.
we talk a lot as scientists about the media and the press about the fake news and the problem with fake information. So I wonder if you could talk a little bit about where this fits in and maybe what we could do to be proactive about that, because obviously it feels like there's a few different ways of that this could harm science. Like people, it's easy to kind of look at science like being kind of having these problems and saying like none of it's reliable, and then you could have scientists potentially being too defensive and defending science and not owning up and not acknowledging problems. So it's very complicated. But what is your perspective and your point of view on how we can maybe be proactive in this environment?
I would say that a year ago, I was fairly optimistic, and I was sort of the opinion like, we scientists, we know what we're doing, trust us. And there's a couple of people with different opinions, and that's fine, but they're not really harmful. But I'm a little bit more pessimistic today, because I think we have seen several countries with leaders who have no longer supported science. And of course, I live in the US normally, not right now, but normally I'm in the US, and I have been, with many others, so disappointed in how science is now being dismissed. And not just that, but how we're moving towards being less polite on the internet or to each other, where it seems to be now the new normal that it's totally fine to call each other an idiot. And it's hard to remain polite and to be in a discussion when somebody just calls you an idiot or even worse, a retard. I've been called many things on Twitter, and it hurts. I cannot deny that. You sort of try to pretend, and everybody says, "Oh, just move on." But when you try to fight disinformation, it's not an honest discussion. Like I tried to my scientific arguments, but it seems that most of the people who are convinced that it's different, they don't listen to arguments. They just immediately slap you in the face with an insult. And so these discussions are not very fruitful. And I have less and less hope that we can convince people like that. And some of these people are very influential. And it's apparently totally fine on social media, because the moderators don't really seem to care. They find it acceptable that people call each other idiots, and it's just hard, because that's not an honest discussion anymore. It's just throwing names, and I don't know. Maybe I'm very pessimistic because that happened this week to me. I've lots of trolls, and it's harder and harder to go into a fruitful discussion with people who don't believe in science.
Yes, no, I understand, and they're always there, I suppose. So I suppose you use Twitter and social media for the positive things, which presumably are that you can get this kind of information spread very, very quickly as well to people who care about it. Like, I've seen your work on this, and I haven't engaged so much, but it's definitely made me think a lot about it. So is that why you use social media, in spite of the fact that it's you kind of risk kind of being targeted by these trolls?
Yeah, I mean, on the other hand, social media has many good things. So the trolls are bad, but there's also lots of. I feel the scientific community is so wonderful. It's diverse. It's open to lots of ideas. It's exchanging practical tips. It's cat pictures. Did I mention cat pictures? Stuff like that is just, it warms your heart, and you see science like the moment it comes out. It's, you know, the paper is still warm and you can already read it. It's fantastic. It has so many good things. And then of course the trolls are bad. As long as you stick to the circle of science, it's fairly fine. But if you move outside and engage with non-scientists, it can be sometimes very nasty, and sometimes very rewarding also. But it's very dependent on which circle you're in. And I try to stay in that science circle as much as possible, even though I want to make sure it's diverse and representative of all types of voices. But it can be very unpleasant outside of that.
I have some questions, but I just want to pick up on your last point. In my old job, working at the Science Media Center, we would often face lots of antagonistic insults from people who were from outside of mainstream science and had particular opinions about things like perhaps vaccines or, you know, certain diseases or certain treatments for diseases, and they didn't like the currently accepted treatments, and so they, you know, really didn't like anyone who was advocating the science for any of these things, and then it would get very personal. So it's certainly a thing that I've experienced as well.
Right.
But my question for you actually was, in your talk, we had a look at all of these examples of falsified data, mostly images or mostly doctored images or doctored Western blots, that kind of thing. With the advent of, you know, widespread coding and, you know, people able to just train AIs and so on and take a more data focused approach, is there a risk that people can falsify, like, you know, data sets to a point where, and you know, they can come to conclusions erroneously and falsely, and is that harder to detect? Because, you know, our human eyes are really good at detecting visual irregularity. We're not so good at looking at giant spreadsheets of numbers and working out if it's a real data set or not. So I just wondered if you had any comments on that development.
Yeah, and the answer is, I think that happens in all types of science. And if I can spot it in photos where it's relatively, you know, it's somebody left a trace. I can see the photo clearly. I can draw my little colored boxes, and everybody's like, duh, that is photoshopped. It's very obvious. But yeah, like you said, if there's a data set of 1 million reads from a sequencer, who's checking that? I certainly, if I peer review a paper, I never download those data sets. I don't even know how to download from you know, not PubMed, the NCBI SRA file. I have no idea how to open that or do that. So I don't look at these large data set as a peer reviewer, and I imagine that not everybody would do that as well. And so it must be relatively easy to cheat and to not be detected. And so I imagine that happens fairly often, but I have no idea about the frequency, so I don't know. But I worry about it, and I also worry, like the paper mill that I pointed out, where you have these artificially generated bands. In that case, they made the mistake of having the same background, so we could easily recognize them. I'm convinced that the paper mill folks are like, oh, we should also do something, you know, have some artificial intelligence making a background that looks different every time. And that's, if you can make fake bands, you can make fake backgrounds. So how can we ever recognize these things? We can only recognize them when they make a mistake, and we can then catch them. But it must happen a lot. And I'm very worried about paper mills in particular, because the individual fraudster usually will make a mistake, but these professional fraudsters are really scary. And I think as data detectives, even though we might use bioinformatics ourselves to find these things, we will always be one step behind what the fraudsters are doing.
And there's no real systematic way in which science hunts for these things other than the peer review system.
No, the peer review system doesn't hunt for it. I feel the only hunters are people like me.
Yeah, I mean, so
right.
what I mean is,
the peer review, right.
Your approach is unique in the fact that it's not part of the system. Do you know what I mean? It's not like there's a,
right.
You know, ubiquitous way in which science looks for false data. We look for, perhaps experiments that weren't done to the best possible standard, but there isn't the same kind of correction system for falsifying data.
So journals or publishers in general are starting now to be more aware and to scan for these things. So you see more and more journals that have more rigorous guidelines for submitting figures, for example. So they will say splicing is not allowed, and these and these and these things are not allowed. Sometimes you have to spell it out specifically. You are not allowed to slap a rectangle over a band you don't want to see. Not allowed, just a band has to be there. You can play with the contrast, you can make it lighter or darker, but you still need to see background features, and you cannot have oversaturated bands. You can have very specific rules for those type of things. And now more and more will also ask for things like, did you outsource any of your experiments? Because that is now being used as an excuse where the author will say, oh, we have no idea what happened to this Western blot. We actually asked a friendly laboratory to do this experiment for us, and we didn't do it ourselves, so we're not to blame. And we're like, well, if you outsourced it, you should have mentioned that. But then if the journal doesn't specify it, so there's more stricter and stricter rules trying to catch, or to at least make it harder for fraudsters to do these things. But it's still not enough, and journals just are very slow in responding. It's like changing a big cruise ship. It doesn't change very quickly in its direction. I know, I've been doing this for years, so I know I'm still not, the ship is still turning, but we're getting there. I do feel publishers are starting to listen to what people like I'm saying, and they're starting to take us seriously, because I'm not going to go away. I've been doing this for almost 7 years now, and you just have to be very patient in this business, for sure.
Well, very grateful that you and people like you are helping to preserve research integrity and hopefully increase our research integrity. So thank you very much for this talk. Ben, did you have any more questions before we run out of time?
I've got a ton of questions here actually, but I don't think we have time. I mean, it was great to hear from you in person. I think, like everyone else, we should interact with you on Twitter. Is that the best place to get in touch with you, Elizabeth?
Usually, there are. I'm at Microbiome Digest. So it's microbiome without an E at the end. It's because Twitter does Microbiome Digest. That's my handle. Or search for Elizabeth Big. If you hover over my name, that's actually my name. And there's, as far as I know, nobody else with that name on Twitter. So luckily I got a very unique name, so you can find me.
Nice. And we are following you, so you could also hunt through the FEMS followers, or people that FEMS are following as well. So a few ways to find you online.
Sure. Great. Well, thank you so much for having me here. It was my pleasure. And I'm going to listen to a couple more talks before I go to bed.
Awesome. Yeah, we can't wait.
Thank you.
Going to go up online as well eventually, so people can review it and watch it again. Thank you.
All right. Thank you so much. Bye-bye.
Okay, so we have our break now before the last round of talks.
It's our final break, which will lead to our final individual live stream. So that means there'll be 4, on social media, there'll be 4 live stream posts containing all of the webinars that we've done in the last 24 hours.
So we stop for 40 minutes and we come back with Jason Tetro again, if I'm looking at reading the...
Indeed. Jason Tetro.
Great.
And then we'll be jumping back into the daytime to head to California for Carly Grant. And then we will be finishing on Yeast Hour, where we'll be discussing essentially yeasts and brewing with yeasts to kind of bring us to a fitting end. But we'll see everybody at 11 o'clock CET, 10 o'clock London, Lisbon, Lagos time. And see you on the other side.